Hello, good morning, everyone. I'm Tamara Johnson. I'm a lead physician within the Division of Pediatrics and Maternal Health in the Office of New Drugs in FDA's Center for Drug Evaluation and Research. On behalf of the Division of Pediatrics and Maternal Health and the University of Maryland Center of Excellence in Regulatory Science and Innovation, I would like to welcome you to this day-and-a-half public workshop on evaluating immunosuppressive effects of in utero exposure to drugs and biologic products. The purpose of the workshop is to engage stakeholders to discuss the available data on placental transfer of drug and biological products with immunosuppressive effects and the potential clinical impact on infants exposed in utero. The workshop will not discuss topics related to policy and regulation or recommendations on drug use during lactation. Our objectives are to discuss the available data on the placental transfer of drug and biological products with immunosuppressive effects, to discuss the potential clinical impact on infants exposed in utero, to identify gaps in knowledge, to explore innovative and practical approaches for collection of relevant data, and to allow an open dialogue amongst regulators, academia, industry, and patient organizations regarding the potential safety concerns and approaches to data collection. I would like to take this opportunity to thank our speakers, panelists, moderators, and the planning committee for their expertise and efforts in putting together this workshop. We look forward to a productive workshop, and thank you all for joining. Now, a few housekeeping items before we move on. This is a hybrid workshop with in-person and virtual attendees. Please reference the FDA workshop webpage for meeting materials such as the final agenda and speaker biographical summaries to learn more about our distinguished speakers and panelists. For workshop attendees in person, please note that there is a kiosk for purchasing food and beverages in the main entry hall. The kiosk is open from 8 a.m. to 2 p.m. Lunch may be eaten in the room, at the tables in the main hallway, or on the patio outside. Bathrooms are located behind the kiosk. Phones should be placed on silent for the duration of the workshop. If it is necessary to take a phone call, please do so in the main entry hall area. And lastly, note the FDA guest Wi-Fi network. There will be an opportunity for the audience to ask questions. Hold questions until the designated time during the panel discussions. There are 2 mics in the room. Please state your name before asking your question. For our virtual attendees, you will not have access to camera or microphone. There is a Q&A button for asking questions. If you're unable to connect to Zoom, please copy and paste the below hyperlink. Speaker slides and workshop recordings will be posted within 3 weeks following the close of the workshop, and they will be located on the CERSI website. We are honored today to have our commissioner, Dr. Robert M. Califf, present the keynote welcome for the workshop. Dr. Califf was confirmed as the 25th Commissioner of Food and Drugs. He also served in 2016 as the 22nd Commissioner, and immediately prior to that, as the FDA's Deputy Commissioner for Medical Products and Tobacco. Dr. Califf has had a long and distinguished career as a physician, researcher, and leader in the fields of science and medicine. He is a nationally recognized expert in cardiovascular medicine, health outcomes research, healthcare quality, and clinical research, and a leader in the growing field of translational research, which is key to ensuring that advances in science translate into medical care. Prior to joining the FDA in 2022, Dr. Califf was head of medical strategy and senior advisor at Alphabet Inc. contributing to strategy and policy for its health subsidiaries, Verily Life Sciences and Google Health. He joined Alphabet in 2019 after serving as a professor of medicine and vice chancellor for clinical and translational research at Duke University. He also served as director of the Duke Translational Medicine Institute and was the founding director of the Duke Clinical Research Institute. It is my pleasure to introduce Dr. Califf.
And thanks for that kind introduction. It's great to see people here in person at White Oak. Maybe today is the day you could eat outside. I heard that part of it. I'm not quite so sure. It's been like an oven here in the swamp for quite a while now. Today is like the best day we've had in a while. So welcome, and also welcome to the CERSIs. CERSIs are a very important program to us, and across the board, linkage with expertise in academia is a very important part of what we do. I have a few prepared remarks just to hopefully set off the meeting. First of all, I'll just say it's a great time right now, given where science is, for a meeting like this. Identifying these gaps in knowledge will be important as people plan what the research agenda should be, but the ability to look at issues like this is so advanced now compared to where it was recently. Of course, this pertains to 4 million births a year in the United States, with pregnant individuals representing an important segment of the population. Many have chronic or acute conditions that need treatment. Indeed, the published data show that most pregnant individuals take at least one medication, not counting vitamins, during pregnancy. As a public health agency dedicated to applying the best available science to protect and promote consumer and patient health, the FDA is committed to supporting and advancing data collection. The decisions we make every day concerning the effectiveness and safety of new products, as well as most of the other aspects of our work to advance consumer and patient health and safety, rely on the need for high-quality data in the context of well-designed studies to provide the substrate for analyses that produce reliable evidence. Strengthening systems for generating and gathering new and better data is an important priority as we seek to supply increasingly well-tested methods to generate reliable evidence to inform and improve the many decisions that we make as consumers, patients, families, and clinicians. In considering this topic, I want to underscore what may otherwise seem an obvious point. The data we collect must not simply be of high quality, but the data are only useful if they're focused on and responsive to the specific population being studied and the clinical environment in which intervention might be considered. We know that some groups of patients, whether based on their race, sex, or other differences, may respond differently to therapies. To develop effective treatments requires patient-specific data. This connection and focus is particularly essential in the data collected to inform clinical care for those who are pregnant. Unfortunately, it's also all too clear that this is an area where enormous gaps remain. Over the years, the FDA, in conjunction with the clinical community, has made important progress in strengthening our approach to data collection in pregnancy, but significant shortfalls remain in the way we gather and access scientific evidence for determining whether drugs are safe for those who are pregnant and their fetuses, and what the correct dose should be, for example, of a drug to be given. Those who are pregnant historically have been excluded from drug development trials and continue to be underrepresented in research. To help address this shortfall, the agency may require post-marketing safety studies to collect this information, and when appropriate, the FDA encourages enrollment of pregnant individuals in clinical trials. But we continue to face a lack of information about the safety and dosing of many drugs when used during pregnancy. This is an area that I've had some personal experience with. Our daughter Sharon was born with a serious congenital heart defect requiring surgery as an infant. After a pioneering life-saving surgical procedure, a month in the intensive care unit, and assiduous follow-up, She's been able to lead a productive life and, in fact, coordinates multinational clinical trials in her current job. She's given birth to a granddaughter who's a rising junior in college and majoring in statistics. So granddaughter can actually do math after all this. But during her pregnancy, Sharon needed to take medication for her chronic health conditions. And at this time, I was a professor in medicine and cardiology with a focus on the operating characteristics of tests and therapeutic clinical trials. This proximity made me even more aware of the paucity of information to assess clinical status and guide the use of medication during this critical time period when so much was at stake. Decades later, despite all our technological sophistication, we have considerable work to do. The focus of this workshop, the evaluation of immunosuppressive effects of in utero exposure to drug and biologic products, is a particularly important aspect of how we study and treat this population. Some products can be actively or passively transported across the placenta from a pregnant individual to the fetus. When this occurs for products with immunosuppressive properties, it's biologically plausible that these products can exert immunosuppressive effects on the developing fetus and newborn infant. Among other impacts, the exclusion of pregnant individuals from clinical trials has resulted in a lack of data available on transplacental transfer and its potential consequences to the developing fetus and newborn infant. During this meeting, your goals should include an examination of all the available data on the placental transfer of drug and biological products and the potential clinical impact on infants exposed in utero, and exploring innovative and practical approaches for collection of relevant data. The workshop will allow for an open dialogue among regulators, academia, industry, and patient organizations regarding the potential safety concerns of medicines that may need to be used during pregnancy and approaches to data collection in this complex clinical situation. And it offers an important opportunity to seek the evidence to identify and address knowledge gaps about immunosuppressive effects in infants exposed in utero. As FDA Commissioner and in my work as a cardiologist, one of my priority areas of focus has been equity. Today, we understand more clearly than ever just how many of the negative health trends that exist today, from shortened life expectancy to the increase in chronic diseases and, of course, infant and maternal mortality, also including heart disease and mental health. I could go on and on. These are the result, at least in part, of social determinants and inequities, which interact with the biological determinants in ways, in many cases, yet to be defined. For instance, we have a better understanding of how minorities and people with lower education and wealth levels and those who live in rural areas have, on average, a shorter lifespan, been in poorer health, and, of course, a higher maternal and infant mortality. It's not a new issue, but it gained attention during the pandemic when we saw certain segments of the population, including but not limited to racial and ethnic minorities and rural, lower income, and lower education populations affected differently. The FDA increasingly has been focused on the goal of equity throughout our scientific work, and we've worked to strengthen this aspect of science in everything from increasing the enrollment of minorities, women, and elderly patients in clinical trials to bring the voice of underrepresented populations into the world of drug development. This is an especially specific aspect of research on pregnancy, and whether people who are pregnant are included in clinical studies that generate the relevant data, we need to find answers. During pregnancy, it's also essential that we include the spectrum of clinical conditions that require medication to improve the outcome of the fetal-maternal pair. We must also be cognizant of the social determinants that are so important in determining outcomes and the ways in which these factors interact. COVID-19 pandemic highlighted the lack of safety and dosing information in pregnancy for therapeutic products authorized or approved to treat COVID-19. Pregnant individuals were excluded from most clinical trials evaluating initial COVID-19 therapeutics. Pregnant individuals with COVID-19 were demonstrated to have greater risk of severe disease compared to those who are not pregnant and have an increased risk of pregnancy complications. One indirect consequence of the pandemic was that researchers and analysts at the FDA were able to apply more creative approaches to evidence generation, including the design of clinical trials. We're continuing to take a hard look at which approaches have worked and how we can best leverage this information, particularly in areas involving the study of medication use in pregnancy. The agency has recently reinforced its commitment to increase underrepresented groups in clinical trials, including pregnant individuals. In the 2022 draft guidance to industry, diversity plans to improve enrollment of participants from underrepresented racial and ethnic populations in clinical trials. The agency described a mandate to broaden clinical trial eligibility and increase diversity of trial participants to maximize generalizability of trial results and reflect the therapy's benefit-risk profile across the patient population likely to use the drug in clinical practice. Beyond race and ethnicity, diversity in clinical trial participants includes, of course, additional demographics such as gender, identity, age, socioeconomic status, comorbidity, and pregnancy status. The FDA encourages drug manufacturers to submit plans that ensure adequate participation of relevant and underrepresented populations in clinical trials, including enrollment of pregnant individuals. And as you talk about this very refined area of science and a very specific topic, the recent guidance to come out is specifically oriented. The law specifies phase 3 clinical trials, essentially. But I really want to encourage the companies that are here and the academics working with companies to start at the beginning of the development plan and think about the entire development plan. And it's written into the guidance that this is something we really hope will happen. So you may do a specific study that's not relevant to the overall diversity plan. It's a particular biological issue. But taking the whole thing into account, we're very hopeful that we'll get very good plans and much better information about populations that have been left out. In addition, the 21st Century Cures Act established the HHS Task Force on research specific to pregnant women and lactating women to identify and address gaps in knowledge and research on safe and effective therapies for pregnant and lactating individuals. Key recommendations include the need for pharmacokinetic and safety data to be conducted in a timely manner. At this workshop, we look forward to working with you and collaborating with all of our stakeholders in discussions about the available data on placental transfer of drug and biological products within immunosuppressive effects and the potential clinical impact on infants exposed in utero. Before I close, I want to mention one final topic that's a particular priority. It's entirely applicable to the topic we're discussing today, but has a much broader impact, affecting our ability to use the promise and power of science and medicine to protect public health. What I'm speaking about here is the extraordinary, dangerous, and growing dissemination of misinformation about science and medicine. The Internet, along with the digitization of data and other information, has dramatically changed how we communicate and how we incorporate information. This has a particularly significant impact on health, offering enormous opportunities but also posing tremendous risks. Today, the consistent stream of information, opinion, and misinformation is helping to erode trust in experts, including the FDA, as well as the science on which decisions by scientists in the FDA and elsewhere are based. The potential impact of this misinformation is enormous, leading people to disregard proven treatments that will improve their health and reduce the risk of death. And it's an issue that directly relates to the conduct of clinical studies in pregnancy, including pharmacokinetic studies. There's been a great deal of misinformation about the appropriateness of dosing clinical studies in those who are pregnant, as well as about vaccinations and pregnancy. As you address the many issues on your agenda for this workshop, I hope you'll also focus on this aspect of public health. We're looking forward to working with you to elevate reliable, truthful information and counter the efforts to create and promote misinformation and disinformation. It's one aspect of the significant work we have to do to ensure that patients and healthcare professionals have the knowledge they need to make well-informed and safe decisions about the medications needed and actions taken during pregnancy. Another way of saying it, the most elegant science in the world about this topic is going to be of no use if, for example, rural patients don't seek help at the time they need it to reduce the risk of bad pregnancy outcomes. We're sort of, as you know, at the bottom of the heap of high-income countries in these outcomes. So we need to marry this elegant science that you're talking about today in the context of information that will help people make better decisions about their own health. So thanks for your commitment, and look forward to productive meetings. Unfortunately, we have a few other things going on today, so I'll be tuning in from the desk a little bit, multitasking. Some of my meetings are actually quite boring, so I can multitask during those. I won't say which ones. But thanks, and again, it's just great to have in-person meetings. The scientific exchange is essential. I don't want to diss the people who are tuning in remotely, but the more we can have good interaction in topics like this, the more likely we're going to be able to fund and advance the science that's needed to make the progress that will help all of us. Thank you.
Thank you, Dr. Califf, for those introductory remarks. Now I would like to turn to Drs. Catherine Kratz and Sonali McClymont to present the regulatory considerations for in utero exposure to drug and biologic products.
Good morning and welcome. My name is Katie Kratz. My colleague Sonali McClymont and I will be providing a brief overview of the regulatory considerations for in utero exposure to drug and biologic products. And we will also provide an overview of the planned sessions for this two-day workshop. We are medical officers within the Division of Pediatrics and Maternal Health at FDA. I am an obstetrician gynecologist, and Dr. McClymont is a pediatric hospitalist. We're so pleased that you're here with us today. First, a bit of housekeeping. We do not have any financial disclosures, and the views expressed in this talk represent our opinions and do not necessarily represent the views of FDA. The objectives of our presentation are shown here. We will briefly discuss the regulatory framework for inclusion of pregnant individuals, neonates, and infants in drug development trials. We will provide examples of drug and biologic products that have immunosuppressive effects on offspring following in utero exposure. And towards the end of our presentation, we will highlight knowledge gaps. And finally, we will outline the workshop sessions. As Dr. Califf and Dr. Johnson have both said, the reason that we are here today is to focus on in utero exposure to drug and biologic products. It is well known that pregnant individuals take medications during pregnancy. On average, they report taking 2.6 medications at any time during pregnancy. These medications may be transferred across the placenta, thereby exposing the fetus and having effects on the infant. How do we study this exposure of the fetus and the infant? This question will be at the heart of the presentations and panel discussions over the course of the workshop. In the 3 slides that follow, 2 will provide an overview of the current regulatory framework for studying pregnant individuals, neonates, and infants. And 1 slide will provide an overview of the regulations behind labeling of pregnancy safety data. Considering how to study in utero exposure to drug and biologic products begins with considering the inclusion of pregnant individuals in pre-marketing drug development trials. Historically, pregnant individuals have been excluded from research in an effort to protect them from research-related risks. The consequence of this has been limited availability of safety data to inform labeling, which leaves clinicians and patients to conduct their own benefit-risk analysis. The current landscape involves strong support from FDA for the inclusion of pregnant individuals so that they are protected through research. In 2018, FDA published the draft guidance shown here on the right, which is currently under revision. The guidance addresses the ethical and scientific issues surrounding the inclusion of pregnant individuals in drug development clinical trials. To support enrollment of pregnant individuals, exclusion should be justified. When there is no justification for exclusion, developmental and reproductive toxicology studies in relevant non-clinical models should be planned accordingly and completed before enrolling pregnant individuals. We recognize that there are many challenges involved in conducting research in pregnant individuals and that the benefit-risk analysis is complex when the pregnant individual fetal dyad needs to be taken into consideration. However, human data are needed to inform safety and appropriate dosing of drug and biologic products during pregnancy, and we believe that the ethical and logistical challenges can be overcome. Along with FDA, efforts to advance this goal have been made by multiple national and international organizations, some of which include PREGLAC, the National Academies of Science, Engineering, and Medicine, and the International Council for Harmonization. Human data to inform safety of drug and biologic products during pregnancy may be obtained through pre-marketing clinical trials, as was discussed in the last slide, and also through post-marketing studies, which will be discussed in this slide. As many of you know, the Food and Drug Administration Amendments Act of 2007, known as FDA, provides a regulatory framework in the form of a post-marketing requirement, or PMR, to study new safety information at the time of approval or after approval. The application of FDA to pregnancy safety is described in the guidances shown on this slide. New safety information may be data about a serious risk associated with use of a drug or biologic product, or it may be a concern about a risk that FDA believes to be serious but does not know enough about to address in labeling. Any evidence that a drug or biologic product crosses the placenta means that there is a concern for a serious risk, and the regulatory authority described by FDA may be used to study this risk. As pregnant individuals have not been included in pre-marketing clinical trials historically, PMRs such as pregnancy exposure registries, complementary studies, and descriptive pregnancy safety studies have been issued to obtain data related to pregnancy safety. Traditionally, these PMR studies are observational in nature and provide descriptive data related to pregnancy outcomes, congenital malformations, and infant outcomes. Clinical data, such as pharmacokinetic or PK data, and evaluation for abnormalities of immune system development in offspring exposed to drug and biologic products in utero could be obtained from PMRs, as supported in the FDA guidances shown on this slide. However, these data have not been requested in PMRs due to challenges in identifying measures to evaluate immunological effects in neonates and infants following in utero exposure. In this workshop, we are looking to define strategies to conduct these evaluations. Now moving on to labeling. Relevant pregnancy safety data, including immunosuppressive effects of in utero exposure, are summarized in drug and biologic product labeling in subsection 8.1, titled Pregnancy, under the heading Risk Summary, and further interpreted under the heading Clinical Considerations. As outlined in the Code of Federal Regulations, or CFR, the risk summary must contain risk statements based on data from all relevant sources, including human data, animal data, and/or pharmacologic data, that describe for the drug the risk of adverse developmental outcomes, including structural abnormalities, embryo-fetal and/or infant mortality, functional impairment, and alterations to growth. This is where in utero exposure data are summarized. The clinical considerations section must include relevant information to the extent that it is available under 5 different headings, with fetal or neonatal adverse reactions being the most relevant to our discussion in this workshop. If it is known or anticipated that in utero exposure increases or may increase the risk of an adverse reaction in the fetus or neonate, the labeling must describe the potential severity and reversibility of the adverse reaction, and must also describe available information for monitoring or mitigating the reaction. Now that we've discussed the regulatory framework for obtaining pregnancy safety information and its inclusion in labeling, we'll move on to look at 3 examples that we've selected to demonstrate the types of available data on immunosuppressive effects in offspring following in utero exposure and how this information has been included in labeling. Baricitinib is a JAK inhibitor indicated for the treatment of active rheumatoid arthritis, as well as severe alopecia areata and severe COVID-19. Relevant data are from a pre-market non-clinical study called a pre- and postnatal development study, or a PPND study, and we'll learn more about this type of study later today. The PPND study for baricitinib in pregnant rats demonstrated decreased cytotoxic T cells in rat offspring exposed in utero. These pre-market non-clinical data appear on the risk summary in the labeling shown on the right. However, given that there are no clinical data to put these non-clinical data in context for humans, there is no clinical consideration section for fetal or neonatal adverse reactions. Alirocumab is a monoclonal antibody indicated to treat cardiovascular disease and hypercholesterolemia. Pre-market non-clinical data from a PPND study in pregnant monkeys showed suppression of the humoral immune response at 4 to 6 months of age in infant monkeys exposed in utero. These data are included in the risk summary. However, similar to the previous example, in the absence of human-infant outcome data, the labeling does not include a clinical consideration section for fetal or neonatal adverse reactions to aid with clinical decision-making. Infliximab is an IgG1 monoclonal antibody specific for human tumor necrosis factor alpha, indicated for the treatment of inflammatory bowel disease, rheumatoid arthritis, psoriatic arthritis, plaque psoriasis, and ankylosing spondylitis. Post-market clinical data from case reports and pharmacovigilance have reported detectable drug levels in utero exposed infants for many months after birth, and infant deaths due to sepsis and disseminated BCG following BCG vaccination in human infants exposed in utero. Based on these data, a clinical consideration section with fetal or neonatal adverse reactions is included, which describes that these infants may be at increased risk for infection and provides a recommendation for at least a 6-month waiting period following birth before the administration of live attenuated vaccines. These examples illustrate how we're trying to communicate information in labeling with missing knowledge. When a product with immunomodulating properties crosses the placenta and enters the fetal circulation, we don't have a clear understanding of whether it may have short and/or long-term effects on fetal immune system development. Furthermore, if the in utero exposed infants have ongoing detectable drug concentrations following delivery, we don't know whether that detected drug concentration is clinically relevant such that it would actually impact the infant immune function or the infant's ability to adequately respond to infection and/or vaccines. We also recognize that exposure through lactation may have an impact here, and there are knowledge gaps related to lactation as well. However, we will not be discussing lactation during this workshop because that subject could really fill an entirely other workshop. We're holding this workshop to ask what additional knowledge gaps exist related to in utero exposure and how to fill those gaps. We really hope to gain a better understanding of what types of data we should be collecting and when and how to collect those data. So to wrap up, I'll walk through each session of the workshop and briefly describe the key questions that we hope to answer. We'll start with a background session to ask, what do we already know? What data already exists regarding immunosuppressive effects of in utero exposure? Next, in session 1, we'll ask the opposite. What do we not know? What are the clinical and safety concerns that warrant gathering further data? Session 2 is our non-clinical session to ask, how do we maximize the amount of non-clinical data to inform the design of clinical trials? And then we'll close out today with session 3 to frame the ongoing concerns for in utero exposed infants. So based on available data, what concerns are essentially theoretical due to inadequate data, and what, if any, concerns are well understood and potentially don't require further study? Tomorrow, we'll start the day with session 4, and we'll ask what types of data should we be collecting and how do we design studies to collect those data? And lastly, in session 5, we'll synthesize the information from the prior sessions and discuss future directions for the next steps needed to close the current gaps in knowledge.
So that's our plan for the next day and a half. We thank you all very much for being here to work through these very challenging questions together. And I will now turn it over to Dr. Layla Shaheen to start our background session. Thank you.
Good morning, everybody. I'm Layla Shaheen, and I'm the Deputy Director for Safety in the Division of Pediatrics and Maternal Health. Welcome, everybody. It's great to see all of you. It is great to be here in person, especially it's good to see familiar faces, and it's great to meet the people that we've been meeting with on Zoom for the last several months. It's great to meet you in person. So thank you all so much for being here today. I'm pleased to introduce the first session on background and current landscape. All of our speakers have illustrious careers, and I'm not going to take up time from their presentations to provide details about their expertise, but please refer to their biographies on the website. So I'm going to only provide very high-level introductions. The first speaker is Leslie Myatt, who is Professor and Director of Perinatal Research at the Oregon Health and Science University and an international expert on the placenta.
So thank you, and thank you for the invitation to come along and talk today about mechanisms of placental transfer for small molecules and biologics, and I've got no conflicts of interest. I want to start out by proselytizing somewhat and telling you that the placenta is not just a conduit. As a card-carrying placentologist, I'll tell you it's the most important organ you ever had. None of us remember our placenta, unfortunately, but it made us what we are today, for better or for worse, and I'm saying that tongue-in-cheek. But I also say it's the immune barrier between mother and fetus. It also directs maternal metabolism, maternal physiology to ensure the supply of nutrients and oxygen that it needs to then direct fetal growth and development. It's in a constant state of growth and development across gestation, so a placenta in the first trimester is not the same as a placenta at term. So here in this cartoon here at the top, you'll see a cross-section of a terminal villus tree in the first trimester. This is the main exchange unit of the placenta. So we have maternal blood in contact with this multinuclear syncytial trophoblast layer, which is the site of steroid and peptide hormone synthesis, and also the site of many of the transporters. But in the first trimester, it's underlaid by a continuous layer of cytotrophoblast, the blue cells, which constantly proliferate and fuse into this outer syncytial trophoblast layer. And you'll see there is fetal vasculature in the core of the villus tree, but there's a considerable distance, several cell layers between maternal blood and fetal blood. In contrast, at term here, the villus core is much larger. You'll see we still have this outer syncytial trophoblast layer, but we see these nuclear aggregates or syncytial knots, apoptotic nuclei, which are shed constantly into maternal circulation, gram amounts per day. And you'll see very few residual persisting cytotrophoblast cells. You'll see the volume that's occupied by the fetal vasculature is much larger, and now these vessels sometimes move very close to the syncytial trophoblast layer, so the so-called vasculo-syncytial junctions, so there's a much shorter diffusion distance between maternal and fetal blood. So altogether, these improve efficiency and capacity for placental transport. So you've got increased villus volume occupied by vessels, exponential increase in trophoblast surface area, about 13 square meters at term. That's the size of a parking spot. Okay, just to give you some comparison. Mean trophoblast thickness, maternal fetal diffusion distance decreases. Again, all of these to improve efficiency and capacity for placental transfer. The placenta has also evolved a series of transport mechanisms for a range of molecules of various sizes. So small hydrophobic molecules less than 600 molecular weight can diffuse freely down a concentration gradient from maternal circulation to fetal circulation. More recently, there's been some interesting work on nanopores, which have been described between the apical surface and the basal surface of syncytial trophoblast. Again, more work needs to be done on those, but glucose and other molecules can transfer down the nanopores. Then there's a range of transporter-mediated mechanisms, facilitated transport, exchange transport, active transport that requires ATP or energy generation, and some here prototypical molecules transported by these different transporters. And finally, large molecules, proteins such as IgG, are transcytosed from maternal to fetal circulation by endocytosis, exocytosis mechanisms. So long before the pharmaceutical industry came on the scene, the placenta had evolved a range of transporters to move nutrients that it required, that were essential for fetal growth and development, across the placenta, but also to protect the placenta and the fetus from xenobiotics and noxious compounds. So these really are now, as I say, being used by drugs. These same transporters now being used by drugs. They were originally classified, all these transporters, originally classified by their function, but they're now classified by the sequence homology of genes. So two main families, the solute carrier or SLC transporters, mainly involved in uptake of substances, and prototypes would be, say, the SLC 3 and 7 families which transport amino acids, and then the ABC cassette or ABC transporters, mainly involved in efflux of substances, and many groups have worked on these. Their expression has been shown to change across gestation. We know what their localization is. The expression changing with drug-drug interactions, obstetric condition, and gene expression could also be affected by SNPs in the genes as well. So if we look more closely at the SLC transporters, again, involved in secondary active and passive transport, more than 400 transporters in over 60 families have been characterized, some more extensively than others. Quite a number we don't really know absolute specificity, etc. And as more and more drugs come along, we obviously have to investigate which transporter they use. And again, some of the groups of compounds that some of these families transport here. The ABC transporters are highly conserved. They use ATP hydrolysis for energy. So you can imagine maybe that conditions where ATP production by the placenta is decreased might affect activity of ABC transporters. They were originally identified as the multi-drug resistance factor, or P-glycoprotein. That's encoded by the ABCB1 gene, is highly expressed in the syncytia trophoblast apical membrane, so on the surface of the placenta, and transports cationic hydrophobic compounds. Again, 7 different subgroups and characteristic structure. So many people have looked at expression and localization of these families of transporters. So this cartoon from this paper in 2021 shows the localization of the ABC transporters, some of the SLC transporters on the apical membrane of the syncytia trophoblast and the directionality of transport. But you also find them on the basal surface of syncytia trophoblast and cytotrophoblast, again, directionality of transport, and some are also expressed in fetal capillary endothelium, where they can move drugs in and out of the fetal vasculature as well. So what factors might influence placental transport? So obviously molecular weight. If you're less than 600 molecular weight, you're hydrophobic. You can freely move by diffusion. Whether their compounds are free or bound to plasma proteins, and then obviously their binding affinity for these plasma proteins, and the amount of plasma proteins important. Lipid solubility, pH gradients, the diffusion capacity of the placenta overall, maternal and fetal blood flow is important for diffusion, for flow-limited transport. What's the expression and localization of the transport? Does the placenta actually bind the drug or compound and metabolize it? For example, it metabolizes steroid hormones, fatty acids quite well. What's the gestational age of the pregnancy? What, as I say, is placental energy generation? And we and others have shown, for example, with maternal obesity, maternal gestational diabetes, dramatic reductions in ATP production by the placenta, which might influence transport. But also importantly now, fetal sex. So what's the evidence of sexual dimorphism in placental function? So we see differences in gene expression across the entirety of gestation, with immune genes being expressed at higher levels in female placentas compared to male placentas. This sexual dimorphism in gene expression has been linked to failure of X-linked inactivation in the placenta. And we and other groups have shown many differences in inflammation, hypoxic, apoptotic, or autophagy responses, antioxidant defenses, etc., fatty acid transporters, gene expression. And a lot of these differences are now linked to outcomes in male versus female fetuses. And generally speaking, it is much better to be a female fetus in utero, many, many more adverse outcomes with a male fetus. This also relates to sex differences in immune responses in pregnancy, and I refer you to this recent review. Interestingly, if you're a pregnant person carrying a male fetus, the maternal immune response seems to be more inflammatory Th1-like, whereas if there's a female fetus, it seems to be more regulatory Th2-like. So again, difference in what the fetus and the placenta are producing, and this then affects both the innate immune system and the adaptive immune system, shown here differences in TLR expression, different levels of chemokines and cytokines, etc., different levels of HLA. And again, during the SARS-CoV-2 pandemic, people started seeing differences in antibody transfer, depending on whether you had a male or female fetus, and differences in responses of placentas as well. So let's move now to the large molecules, particularly active transport of IgG. So of the 5 antibody classes, only IgG is transferred across the placenta using the neonatal Fc receptor. And this Fc receptor shows pH dependence of binding of IgG, such that at the maternal surface of the placenta, IgGs are not bound to Fc because we're at neutral pH. But the IgG is taken up in endocytotic vesicles. Once internalized, the pH of the endosome drops, and that IgG binds tightly to the Fc receptor. They then get sorted. The non-receptor bound proteins are degraded in lysosomes, whereas the IgG bound to the Fc receptor is recycled to the fetal side of the syncytial trophoblast. It's then exposed to neutral pH, and it dissociates off this Fc receptor, so you get transfer of the IgG from the maternal side to the fetal side in that manner. This is work from a colleague Antoine Malek, done nearly 30 years ago now, where they looked at uptake of IgG and transfer across the placenta. So these are the fetal umbilical vein concentrations of IgG. It increases exponentially across gestation. This is the fetal to maternal ratio, and you see at term, the fetal level exceeds the maternal level by about 20 to 30%. Why does it increase exponentially? So the right-hand panel here shows the surface area of the terminal villi. This increases exponentially across gestation, reaches about 13 square meters at term. So you've got greater surface area for endocytosis, greater transfer of IgG across the placenta. Obviously, the earlier you are in gestation, if you're delivered preterm, you don't have the opportunity for this late exponential transfer. So low levels of IgG seen in preterm infants, low birth weight, often associated with a small placenta. Again, less IgG, perhaps impaired transfer, but maternal age, weight parity, type of delivery don't seem to affect transfer. We know that there's differences in the transfer of the IgG subclasses. So broadly speaking, IgG1 is preferentially transferred greater than IgG4, 3, and 2, although there's a little controversy about 3 and 2 if you look at different papers. What factors might limit efficacy of placental transfer after immunization? So what's the time between the mother being immunized and the delivery of the fetus? So how long do you have for the antibodies to be transferred? What's the gestational age at birth, as I've shown you? What's the total maternal IgG level? So the more you've got on the maternal side, potentially the more you'll get on the fetal side. What's this vaccine-specific IgG subclass concentrations? But then maternal pathologists such as hypergammaglobulinemia.
I'll find her on my, but I mean, she's an autism, like she'll be like.
Can you mute, please, whoever you are?
Like she doesn't wear a bra.
Okay, maternal IgG specific subclass concentrations, then pathologies such as where you've got hypergammaglobulinemia and you get competition amongst the IgGs for uptake, and hypergammaglobulinemia is seen in certain African populations, you might get reduced transfer. And then where you've got infections, placental injuries, for example, malaria or IUGR may affect transfer as well. I want to focus a little bit now on post-translational modifications of IgGs. So all IgGs undergo post-translational modification. So shown here is the structure, the light chain, which is the antigen binding region, the FAB site. You can see a post-translational modification. And in the heavy chain, the FC receptor binding site, there's this characteristic end-linked glycan, it's asparagine 297 here, which affects structure and function of the heavy chain. On the right, you'll see in this cartoon, this is a representation of some of the residues which are found in this glycan modification. They're different between the FAB and the FC region. I draw your attention to sialic acid and galactose. We'll come back to that in a minute. But galactosylated antibodies are preferentially transferred due to enhanced binding to the FcRn receptor. But we now know that things like maternal infection may alter this post-translational modification, the glycosylation, and this was illustrated quite well in SARS-CoV-2 infected individuals. You see different covalent modifications, different affinity for the Fc receptor, differences in transport. This was also the importance of the Fc region also illustrated by transfer of anti-TNF antibodies. Infliximab and adalimumab, IgG1s that show the characteristic exponential increase. See the normal binding, neonatal levels exceed maternal levels, persist for up to 12 months. But if you look at etanercept, which is a fusion protein with a modified Fc portion that binds, that's got low capacity for transfer because this Fc region doesn't bind very well to the receptor. You see minimal levels in the fetus at birth. And then with tolizumab, which is just an FAB conjugated to polyethylene glycol, and there's no Fc portion, that doesn't cross the placenta at all. So showing the importance of the Fc region. And this we can take even further now. More recently, if we start engineering antibodies in different cell types, this was an experiment done with the primate placenta where they used HIV antibodies raised in plant cells versus antibodies, the same antibodies raised in mammalian cells. You see that the antibodies raised in the plant cells do not cross the primate placenta. And that's because plant cells cannot add sialic acid or galactose residues in those post-translational modifications. It doesn't bind to the Fc receptor, whereas if you raise it in a mammalian cell that's then post-translationally modified the correct way, it will transfer. Importantly, if you do these experiments in a mouse placenta, both plant and animal antibodies will cross the mouse placenta just because it's not subject to the same post-translational modification. So again, showing species specificity and the model that you use to test is important. So I just want to summarize by saying the placenta is not simply a conduit. Its function changes across gestation. Importantly, activity changes with fetal sex and with lots of medical conditions. We've evolved a range of transporters to handle molecules of a range of sizes, and these are the transporters used by the many drugs that we use. Obviously, many factors have explained it influence placental transport. Transfer of the small molecules affected obviously by the drug transporters, IgG by the neonatal Fc receptor, and importantly now, glycosylation of IgG antibodies can affect binding to the Fc receptor and transport across the placenta. We can utilize that right now by engineering antibodies to prevent transfer. I just want to leave you with the view out of my laboratory window. This is why I get to work early, particularly in the winter. This is the sun coming up over Mount Hood. Thank you.
All right, thank you so much, Leslie. I'd now like to introduce our next speaker, Raman Venkataramanan, who is Professor and Director of the Clinical Pharmacokinetic Lab at the University of Pittsburgh Medical School and an expert in obstetric clinical pharmacology.
Good morning, everyone. Thank you for the opportunity to be part of this important workshop. My talk will be focusing primarily on small molecules. Over the years, we have received funding from NIH, but the presentation that I'm going to make, there is really no conflict of interest in the current presentation. Up front, I would like to acknowledge the contributions of our pregnancy team, led by Steve Caritas, and also the transplant team led by Thomas Stazel. When we think about drug exposure in pregnancy and postpartum, we need to think in terms of safety, efficacy, and actual exposure of the drug in the mother, safety, exposure, and occasionally efficacy in the fetus or in the neonate. When a pregnant woman takes a medication, the drug, which is free or in the unbound state, has the potential to go across to the fetus. And this can typically happen either by a passive diffusional process or a facilitated diffusion process or an active transport. In addition to these passive diffusion and active uptake processes, one needs to also consider efflux proteins that are expressed, which will prevent the drug from going to the fetal side, such as P-glycoprotein, BCRP, and MRP2. Multiple options are available when we think about looking at exposure, response, and safety in pregnancy. Broadly, we can look at them as in vitro model systems, ex vivo model systems, animals, actual clinical data, and computational approaches. My presentation today is going to focus primarily on the clinical aspect and with a little bit on the computational approach. When we consider in vitro cell culture systems, typically these systems are static systems. We are able to look at drug uptake and permeability parameters from these systems. And we are also able to study the impact of drugs on the placental cell and the function. Placenta on a chip is a very promising approach because it not only considers the cell systems, but also provides the hemodynamic conditions that are more practical. And they are also suitable for looking at the effect of drugs on the placental cell function. Ex vivo placental perfusion systems are very valuable, but they are typically very difficult to set up. The success rate is somewhere around 20 and 25%. So every 4 setup, you get data only from 1 setup. And typically, they also tend to overestimate the placental uptake, mainly because these perfusion mediums do not contain blood or proteins which minimize transfer from the perfusion side into the placenta. And also there is physical adsorption of very lipophilic chemicals onto the tubing that are used in these perfusion systems. Animal models of pregnancy provide mechanistic information. However, there are some difficulties in species difference in both drug exposure and response, as well as physiological differences in pregnancy between animals and humans. Clinical studies are therefore the ideal way to obtain data, but typically one needs to get data from a large number of subjects in order to generalize. And it's also difficult to look at long-term studies clinically. When we look at fetal exposure, typical approaches include looking at scar tissue concentration, typically done at one time at delivery. Concentration in the cord blood in reference to the maternal blood at the time of delivery. Commonly, the venous blood is used. Occasionally, arterial blood samples are used. When we get both of them, we may be able to understand a little bit more about the fetal contribution to the metabolic capacity of the drug. Most of the studies look at total drug. Very few studies really look at unbound drug, which is the more important component to measure. And the concentration that is measured in the cord blood depends on the timing of the drug dosing with reference to the sample time, and also the analytical methodology that's used. Other specimens that have been used also include amniotic fluid, meconium, but these are fluids are sources which are not available at multiple times normally, and they are one-time information. Placenta is also a common source of looking at concentration of the drug. One can look at placental to maternal or maternal blood or plasma ratio. Typically at the time of delivery, very rarely we get the opportunity to take samples when there is a termination of pregnancy. Concentration in placenta are very dependent on the site. There is site-dependent distribution of the drug, so one needs to be aware of what site is taken as a sample, and also assay methodology influences the concentration measurements. In the neonates, blood or plasma samples taken from heel stick can provide concentration information and therefore perhaps half-life. Other specimens such as hair, nail, and meconium also have been used. Maternal milk is another source where we can look at maximum concentrations in the maternal milk, area ratio of the milk to plasma, and relative infant dose is something that can be calculated, and also another parameter called UAR, upper 95% area ratio. These are measures of potential exposure in the neonate. Computational approaches, such as physiologically based pharmacokinetic modeling and population pharmacokinetic modeling, are valuable in pregnancy. They can predict the time course, not only the total drug, but also the unbound drug. We can get the area under the total and the area under the unbound drug, and the ratio of the neonatal area to the maternal area. Currently, they are not as extensively used as they can and will and should be used, but they are being increasingly used, and there will be more discussion on this later on. In the simplest sense, physiologically based pharmacokinetic modeling take into account the anatomy, physiology, as well as the pathology, incorporates the drug-based physicochemical and other in vitro parameters, develops a concentration time profile in a non-pregnant, normal, healthy adult. Then one can incorporate pregnancy-dependent variables and therefore be able to predict what happens during pregnancy, and similarly what happens during lactation. One can predict the optimal maternal dosing and measure the drug exposure in pregnancy. One is able to predict the fetal exposure. One is also able to predict the drug dosing and infant exposure through feeding milk. Essentially, these model systems allow one to not only look at the concentration time profile in the blood or plasma, but also gives you an opportunity to look at relative concentrations in other organs in the mother as well as in the fetal compartment. In addition to exposure and response, one needs to obviously look at safety. It must be performed anytime it's possible. A lot of times we don't take advantage of the fact that we have the placental tissue, and we should be able to look at the impact of the drug treatment on the placenta and placental function. Studies could be done on the immune cells, especially the fetal immune development and neonatal immune ontogeny. Commonly, measurements are done in birth-related outcomes, prematurity, number of C-sections, birth weight, and few studies have addressed physical development, growth, mental development, neurological, and renal outcomes, especially with some of the immunosuppressive drugs we use. Nephrotoxicity is an issue. Effect on the postnatal infection, myelosuppression, and response to immunization are some of the things that should be followed. Now let's focus on few of the drugs and see what kind of information is available. Tacrolimus, which is very near and dear to us from Pittsburgh, has a molecular weight of 804. It has a log p-value of 3.3, fairly lipid soluble, but it's very highly bound to the red blood cells. When placental perfusion studies were performed, the accumulation in placenta was 100 times more than the perfusate concentration. However, there was no fetal transfer, perhaps because tacrolimus is a substrate for P-glycoprotein. However, this 100-fold higher concentration is an overestimate of what happened because there's no red blood cell in the perfusion medium. More drug now is allowed to pass to the placenta. In a typical placenta, when you get the placental tissue and measure the concentration in the placental tissue, the ratio is somewhere greater than 3, in most cases somewhere between 10 and 20. And again, the concentration ratio depends on the sample site. There is uneven distribution of tacrolimus within the placenta. The cord blood to the maternal blood concentrations are generally low. This is consistent with the in vitro placental perfusion study. Infant exposure through the milk is also very minimal, given that milk to blood ratio is very small, 0.2. Milk to plasma ratio is a little higher, mainly because most of the drug is sequestered in the red blood cell. The relative infant dose has been calculated to be less than 1% of the maternal dose, and there is negligible ingestion of tacrolimus from milk in the neonates. An interesting study looked at decline in the concentration of tacrolimus in the newborn who were breastfed compared to those who were bottle-fed, and the decline was similar, which strongly suggested that whatever drug that comes through the breast milk, which is consumed by the neonate, doesn't really get into the systemic circulation, which is a critical and important observation. So this is a study that was performed looking at the pharmacokinetics in pregnancy. On the left panel, we have the concentration in the blood at steady state, concentration in the plasma, which is in the open squares, much lower concentration in the plasma compared to blood. The concentration in the milk sample is somewhere closer to the plasma, but much lower in the whole blood levels. The study also simultaneously looks at the unbound concentration in the milk and unbound concentration in the plasma. Very few studies really look into unbound levels, and it's important for us to look into this. In general, tacrolimus is relatively safe. There has been no pattern of malformation, normal development of the fetus, no major negative effect. There have been reports of intrauterine growth retardation, prematurity, and low birth weight. Early in this use of tacrolimus, when higher doses of tacrolimus were used, we did observe transient hyperkalemia and increase in serum concentration in the newborns, which, however, resolved within 24 to 48 hours. These babies also had relatively normal GFR, and so the incident that was observed in these patients is essentially dose or concentration related. There are few studies which looked at neonatal immune cells. At high concentrations, there is some dose-dependent inhibition of cytokine production, but really without any impact on the innate immune response. There is really not much data on the functional immunity of neonatal immune cells. However, pediatric transplant patients who do get these drugs don't really have any immunological issues. There is also adequate immunogenicity after vaccination in these patients. So in general, there is satisfactory growth and development, normal physical and mental development in patients who are on tacrolimus. Cyclosporin, again, a molecule with lipid solubility and highly bound to red blood cells, does transfer through the placenta. The placental concentrations are much higher, 5 to 10 times higher than the maternal blood concentration. Then you look at the umbilical cord concentration. So if you look at the maternal blood, somewhere at about 100, the umbilical cord concentrations are much higher than the maternal blood. However, the cord blood concentrations are typically lower than the maternal blood. What's also interesting is in these patient samples, we also observed not only the parent drug, but also the metabolites of cyclosporin. Cyclosporin is also relatively safe. There has been no embryo toxicity or fetotoxicity. There hasn't been any pattern of malformation. No difference in the IQ in these patients compared to non-transplanted controls, and no long-term neurocognitive effects. As reported with tacrolimus, there have been reports of intrauterine growth retardation, prematurity, and low birth weight. Infants born typically tend to have normal renal function, psychomotor development. Both in animal models and at higher doses, there have been some fetal kidney toxicity reported, but it's totally dependent on the higher concentrations used in these studies. In general, there is no overt clinical immune deficiency in patients when they deliver these babies. In contrast to cyclosporin and tacrolimus, mycophenolic acid, which is highly bound to plasma proteins, but is relatively less lipophilic in nature, is an inhibitor of IMPDH activity. In experimental animal models, it has been clearly documented teratogenic effects. Clinically also, cleft lip and palates have been observed. 32% spontaneous abortions, 27% structural malformations or ear deformations have been reported. Higher incidence of these are observed with later cessation in mycophenolic acid. So longer the patient is on mycophenolic acid during pregnancy, higher is the extent of incidence. This really documents that the effect is more likely due to the mycophenolic acid that's used. So mycophenolic acid must be avoided during pregnancy, and there is not much data on how much of this comes into the milk. Prednisone and prednisolone are typically used in transplant patients. This does cross across the placenta, but it's metabolized by the placental enzymes, and there is minimal fetal exposure, less than 1 compared to the mother. Very low amounts of this drug is detected, and the RID, or the relative infant dose, is reported to be anywhere from 1 to 5 or less. And so typically, this has been not shown to be teratogenic. Occasionally, cleft palate and cleft lips have been reported. Again, these are related to higher doses of prednisone, and typically these days we tend to use dose minimization. Only low doses of prednisone is used, and also we tend to take the patients off of steroids. And so effects of steroids, if there is any, is only related to the higher dose of steroids that are used in the past. Azathioprine is another drug. It has a low placental concentration, low fetal exposure, low concentrations in the milk. Therefore, the RID is less than 1%. There hasn't been any major teratogenic effect. Normal development has been reported. With higher doses, again, there has been some myelosuppression that's been related. There is very limited data available for everolimus and sirolimus. And so the general recommendation is try not to use during pregnancy. I wanted to wrap up with some of the confounding and limiting factors when we look at clinical studies and clinical data. We always need to keep the nature of the original disease in mother in mind before we attribute an observation to a given drug. Comorbid conditions in the patients, such as hypertension, renal dysfunction, and diabetes, could have an impact on the newborn. That needs to be considered. Transplant, we use combination therapies, and so it's very difficult to attribute any event that's observed to a single baby. In case of mycophenolic acid, however, it's been convincingly documented by studies where the patient was on, became pregnant more than one time, and one time they were on mycophenolic acid, the other time they were not on mycophenolic acid. So one can convincingly say the observed effects are due only to mycophenolic acid. There are some issues in terms of obtaining cord blood samples. Studies sometimes report cord blood levels and compare them to the maternal trough concentrations typically reported. They don't simultaneously measure the maternal blood levels. And also, when the cord sample and the maternal blood sample is taken in reference to the drug dosing is very critical when we look at the ratios. And again, as mentioned earlier, only total drug is typically measured, then we need to also be measuring unbound concentration. In vitro placental tissues, we typically tend to use normal placenta, and which could be different from the patient placenta. There are differences in ex vivo data and patients, primarily because of the fact that ex vivo perfusions are single dose studies, whereas in vivo you have multiple dose at steady state. And also, placenta does tend to accumulate a lot of the drug, and the extent of accumulation in placenta is dependent on the time and the gestational period. The composition of milk, when we think about drugs in milk, composition changes over time, and there is sampling time that needs to be considered, and the volume of milk, theoretically, it's assumed to be a given constant when one calculates relative infant dose. We also need to do more short-term and long-term studies in neonatal immune system because we currently have limited data in this space. In conclusion, healthy moms lead to healthy babies lead to healthy society. Pregnancy is a major stress. Postpartum depression is something that we cannot avoid. We need to consider that. That impacts not only the mom, the fetus, as well as the neonates. Typical recommendation in transplantation is at least to wait for 1 year post-transplant, be with a stable allograft function, no proteinuria in a kidney transplant patient, no recent episode of rejection or infection, well-controlled medical conditions such as hypertension and diabetes. In essence, the maternal risk of any graft loss is very low due to pregnancy, and so transplant patients can and should and must have feel free to have pregnant conditions. Neonates, there has not been any reported increase in malformation in the patients on cyclosporin, steroid, and tacrolimus. However, mycophenolic acid should not be used in transplant patients. There have been reports of prematurity, low birth weight, but all of this seem to normalize within 2 years after birth. And so whatever observations were observed predominantly happens in the early transplant period. When one looks at milk exposure, these drugs tend to have limited milk exposure, and therefore they have correspondingly small relative infant dose. Finally, to wrap up, as a society, we have the responsibility to support the efforts towards healthy moms. We need to provide better access to health care for all the moms. From a health professional responsibility, provide education and subjects to the subjects and collect as much data as we can. The responsibility of the patient is to ask questions and facilitate data generation by participating in clinical studies. Funding agencies. understudied population. We need to have more funding to study drug use in pregnancy. We also need coordinated national registries which collect data. Bits and pieces of small registries don't serve anyone. Finally, I always leave any session I get an opportunity with a life lesson. We are all very fortunate to be where we are on what we do. We need to acknowledge and appreciate that. Not everyone in this world is as fortunate as we are. We need to resolve to do something for those who have not and make a difference. We have ignored pregnancy long enough. It's time now. Let's act. Thank you.
Thank you, Venkat. I'd now like to introduce our next speaker, Edwin Lamb, who is a senior clinical pharmacologist at Johnson & Johnson Innovative Medicines R&D, supporting maternal fetal immunology drug development.
Thank you very much for this opportunity to talk up here. Oops, sorry, you guys hear me okay here? And good morning to the folks on the call as well as in live session here. Just try to figure out how to work the slides. Perfect. So in continuation to Venkat's discussion, I'll be talking about the placental transfer of immunosuppressive biologics, particularly giving a broad review of the current clinical pharmacology landscape in hopes to kind of bring light to what has been currently done so far up to date. I'd be remiss to not talk about transplacental transfer, which has already been discussed by Les right now, our resident placentologist here. But for IgG transport, transplacental passage of immunoglobulins is mostly mediated by FcRn, as we've heard, the neonatal Fc receptor. Now, what we see here is, that IgG1 is mostly preferentially transported across the maternal fetal through cord blood compared to all the other subtypes, IgG2 as well as IgG3 and 4. Interestingly enough, through work of some of the colleagues here, we know that FcRn is really only the predominant receptor that's responsible for IgG transport compared to the other Fc receptors, such as Fc gamma receptor. And then interestingly enough, there's pretty much ongoing debate as far as where placental expression goes of FcRn and the level of transport. But for the most part, there's no association between placental FcRn expression and maternal to fetal transfer of IgG. In general, in utero transfer of therapeutic monoclonal antibodies are postulated to be facilitated by placental FcRn, while maternal FcRn is what's responsible for maintaining IgG half-life. So think about how the prolonged extended half-life of most biologics is due to the recirculation of maternal FcRn IgG. We also know that maternal fetal IgG transport increases with gestational age. Transport increases throughout gestation, exceeding maternal IgG towards term, as evidenced by the fetal-maternal ratio here. This has been shown before, but really you see this exponential type of curve occurring during the second to third trimester of pregnancy. Interestingly enough, that fetal-maternal ratio is expected to be somewhere less than 0.1 during the first trimester, as documented by some of these early studies from the 90s, showing that fetal-to-maternal ratio and transfer during that time is really minimal, if not none. So what does this mean in terms of transfer of biologics, particularly biologics that contain Fc portions of IgG1 or IgG2 or 4? What we get is really this type of profile and drawing the concern from throughout gestation of maternal transfer via the placental syncytium over to fetal circulation from first trimester to second and third trimester, and what is postulated to be what exposure looks like for monoclonal antibodies. So we asked really the question that given that most biologics are engineered with various IgG subtype constant regions, what is the current evidence surrounding the in utero exposure of particularly immunosuppressive monoclonals? And surely, timely enough, this paper was actually published by Gent and colleagues over in the Netherlands, but there is some evidence that suggests modest to lower serum monoclonal antibody exposure during pregnancy. What Gent and colleagues did here was evaluate a few, by all means not extensive, monoclonals that are out there on the market, and showed that at least the case for vedolizumab, natalizumab, and ustekinumab, that modestly lower serum concentrations were observed across the different trimesters from first, second, and third across pregnancy. So the question really is here, is transplacental crossing of immunosuppressive biologics occurring, and if so, is it occurring at similar rates to native IgG, as we saw in the previous slides? So what I wanted to offer is that showcase here is that most biologics with immunosuppressive properties are in fact IgG1-based mAb structures, with the exception of infliximab, which is chimeric, and then certolizumab, which has a pegylated Fc. The mAb structure here mostly are IgG1. There are some that are IgG2 or 4-based that are currently marketed out there. But the evidence of transfer, as witnessed by the fetal to maternal ratio, is often used as a surrogate of in utero exposure throughout gestation. As we heard from Ramon earlier, there are limitations that surround that fetal to maternal ratio, particularly due to sampling time points where we fall along the concentration time points. But you can imagine in a very busy setting such as labor and delivery, that the capture of this information is oftentimes very messy. So a lot of these studies are very prospective, observational in design, some of them being case studies in general. But in summary, when we look at where these fetal to maternal ratios fall as it compares to IgG1, we're somewhere along that line of 1 to 1.5 times is what we see for when we near term for infants. And of course, another limitation with the fetal to maternal ratio is we're limited to at-birth concentration for mom and baby pairs collected in order to gauge that level of exposure. So what we see is that informative clinical PK data has mostly been observed with a very limited number of immunosuppressive drugs. Data that is available out there come in the form from the anti-TNF alphas, the anti-integrin and cytokine monoclonals, which currently has been used to help contextualize most biologics with immunosuppressive activity. We see mostly maternal, fetal, cord, venous paired blood samples, which is used to pair or estimate in utero exposure. And we use the estimate of the fetal to maternal ratio to help gauge that level or the magnitude of placental crossing, especially near term. Using the fetal to maternal ratio, we oftentimes use that value as well as exposure to estimate time to clearance. And we use population PK/PD approaches. That's one way. We heard PBPK before as well. That could also be another possibility to use as part of our tool and arsenal in estimating in utero exposure. We also see that postnatal infant collection allows estimation of the extent of persistence. So aside from the magnitude of in utero exposure at term, we also want to know the persistence, how long drug persists in the exposed infant postpartum. So we see in studies sampling beyond the birth collection, so that is beyond cord blood in infants, sometimes at 4, 8, or 12 weeks. And that, in fact, is very informative, especially if you pair that together with population PK/PD approaches. And these, as I'll show later on as case examples, have addressed questions such as timing of live vaccine administration or draw on exposure-response relationships for developmental or infection adverse events. What are some limitations that we do see, and as we've heard and will hear later on in the talks from other speakers, is this relationship between the exposure and the perhaps target engagement as well as receptor occupancy within the infant is definitely of interest, but is often limited. We don't have that level of mechanistic interpretation between exposure as well as the infant, in the infant and their downstream infector impact. Think the exposure-response relationship of, let's say, an anti-TNF, what that downstream effect is mechanistically. And we're only really limited to the level of clinical response and observations, such as developmental outcomes and infection outcomes, mostly with these immunosuppressive biologics out there right now. So I wanted to talk about a few case examples. First one being, well, actually four drugs here: vedolizumab, ustekinumab, infliximab, and adalimumab. What some of these authors did here, and these are mostly produced from academic centers, is evaluate the exposure and time to clearance of drug. What we often intuitively think and see here, as demonstrated here, is that drug generally correlates with duration since last maternal dose. So that is, the time to undetectable levels generally depends on the timing of the previous dose at delivery. So we see when days from final drug dose to delivery for the first plot on the top left corner to the infant delivery level, see this correlative type of trend for vedolizumab as well as ustekinumab, translating to the time to infant clearance and as a function of infant delivery level there. Now, what's good about these studies is that they actually took samples not just at the cord, maternal to cord blood pairs at delivery, but they also sampled postnatally as well. So you can see that these samples were taken to determine undetectable levels postnatally as part of infant follow-up, which helps build this nice population type of profile to understand the persistence of drug after delivery. The same case is to say for infliximab, designated as IFX and ADA, and not to be confused with immunogenicity. This is adalimumab. The correlation between the concentration in mom as well as concentration in cord blood, and how that was used to determine drug concentration as well as persistent in the infant postnatally. And I'd like to remind the viewers here that the fetal to maternal ratios really are varying between the IgG types, the drug types here, but for the most part are still somewhere around the variability of what IgG fetal to maternal ratio transfers are. Carrying forward the case examples with infliximab as well as adalimumab, these study designs helped enable birth as well as postnatal blood collection, which ultimately was used to help inform clinical decisions to estimate infant clearance. Now, they used population PK approaches to help to be implemented using these sparse samples collected to address questions such as time to undetectable concentrations at birth, postnatally as well, which helped enable timing of live vaccination. So using the observations here, a model was fitted to for both infliximab as well as adalimumab, and then generalized to what anti-TNF alpha levels would be at 3, 6, 9, and 12 months. And you can see this is a really cool our Shiny app that was designed by the authors here, which incorporated one cord as well as one infant blood sample to help estimate as well draw on the level of variability of where infant levels would be to allow either undetectable or at least safe concentrations to allow for live vaccination. So taken together with these study designs of not just sampling at birth, but also sampling postpartum in the infant, helps enable modeling programs to estimate at least where infant clearance and persistence would be of drug in the exposed infant. Another consideration, aside from the sparse sampling designs, is considerations on the mechanism as well as the compound in itself to help guide study designs. And I use certolizumab as a key example because certolizumab, pegylated certolizumab that is, has a missing FC region, or FC-free region better yet, that is not expected to undergo FcRn-mediated placental transfer. Now, knowing this mechanistically as well as your compound in itself helps minimize the number of infant sampling, so reducing the infant sample burden postpartum. And, over here is a typical study design that is often implemented, not just for certolizumab here, but for most monoclonals in terms of studying effects clinically, but also drawing PK samples as well. You could see the case for certolizumab of no placental transfer as demonstrated by infant levels at delivery, week 4 and week 8, relative to mothers here. I wanted to also mention that through this review here that studies that are evaluating in utero exposure of immunosuppressive biologics are often, again, limited to the birth collection. Prospective cord blood collection is most often collected at delivery to estimate maternal fetal exposure. And then you can see the various different types of matrices, amniotic fluid, placental tissue that are used to estimate at least where we are in utero. But if the opportunity does present, and we actually presented this at our the most recent SMFM conference this year, to sample opportunistically within in utero, we often take that opportunity to also collect PK as well. And this was done through our nipocalimab program, looking at drug concentrations through chordocentesis. And chordocentesis is a medically necessary procedure needed, and at the same time, when taking those samples, we also had evaluated drug concentration as well. So this definitely was informative to help us understand, at least during the 3rd trimester where we sampled here at gestational week 31, where drug concentration falls. And as you can see with the fetal maternal ratio here, less than 0.1, it's expected that no transfer is occurring during that time period. So I wanted to end the talk by summarizing that studies still remain limited, but the body of evidence is certainly growing. And I say that because when we look in literature right now, a lot of the work that it's being done, it's all within the past 5 years. So most of the clinical evidence gap generated are definitely limited to post-marketing prospective PK studies. Very seldom do we have observations from clinical development programs pre-approval, but we certainly do expect that to change in the near future. We know that placental transfer of monoclonal antibodies are mediated by FcRn, with preferential transfer for IgG1 compared to all the other IgG subtypes. Most of the biologics are IgG1 based constant regions, have fetal maternal ratios that are similar to native IgG. Definitely take considerations for fusion proteins with non-native IgGs such as certolizumab pegylated. Most clinical pharmacology data is collected from prospective post-marketing studies, as we talked about before. What we see in the current landscape is that the time to clearance is dependent on the time to last maternal dose and subsequent in utero and postnatal infant exposure. We do know that there are limitations with interpreting the fetal maternal ratios, mostly that these are ratios that gauge the magnitude of transfer mostly to infants at term. And most clinical experience comes from our anti-TNF alphas currently. Study designs, if we are to design studies to evaluate concentrate PK as well as PD, should consider the maternal neonate pair samples collected at birth, but also longitudinally postnatal sampling to help use as part of a population PK approach or other types of modeling approaches to help estimate the time to clearance as well as persistence to address clinical questions such as timing of live vaccination. And of course, as shown previously, use of opportunistic samples as part of clinical care during gestation, for example, at the second or third trimester, to evaluate drug levels in utero are often very, very valuable samples. Lastly, I suspect that we're probably going to be talking about this a little bit more throughout the remainder of this workshop, but considerations for pharmacodynamics as well as target engagement and exposure thresholds. We don't have or have very limited evaluations accounting the relationship between infant exposure and immunosuppressive biologics and their downstream target engagements and outcome. So with that, thank you for your attention, and turn it over to you.
Thank you, Edwin. I'd now like to introduce our next speaker, Uma Mahadevan, who is Professor of Gastroenterology and Director of the Colitis and Crohn's Disease Center at the University of California, San Francisco. She's an expert on inflammatory bowel disease in pregnancy.
All right, good morning, everybody. So, which is the forward button? Should be this one, right? Okay, here we go. All right, so it's my pleasure to address the audience today, and I have been learning a lot from the first few lectures. It's important to remember that a lot of treatment of pregnant women is driven by fear and not data. And the impact of saying, oh, there's placental transfer, that leads to women being taken off their medication. So I would just say to everyone out here, when you have data, you have to put it in context so that the immediate response is not to take women off therapy. These are my disclosures. And I want to start by saying women have always paid a tax for their reproductive potential. And while we have come a long way, in 1977, the FDA policy excluded women of childbearing potential from clinical trials. And while we've talked about the steps that have come since then, and in 2022, the guidance to industry for diversity plans to include pregnancy, things have not changed that much. For example, women who are pregnant, who become pregnant, must drop out of a clinical trial. Even if they're doing well on the medication, even if other medications that are commercially available don't work for them, even if we know safety of that class. So if you have the 10th anti-TNF that you're testing in a clinical trial and a woman becomes pregnant, she must drop out. And often that data is lost. I think that is the lowest hanging fruit for understanding preclinically how people do, is those women should stay in the study, and whether or not you continue the medication, and in most cases you should, they should still be followed prospectively instead of lost and then 2 years later try to figure out what's going on with them. In clinical practice, women of childbearing age, not pregnant women, women with reproductive potential are not given JAK inhibitors, though it may be the most effective medication for them. This is a result of fear that maybe they'll get pregnant and maybe there may be some harm. And then medications with well-established safety records, like anti-TNFs, are discontinued in pregnancy now. 68% of women who stopped their TNF did so on the advice of their rheumatologist. So these are the doctors telling them to do this. And what's the importance of treating immune-mediated disease in pregnancy? Disease activity is the biggest driver of adverse outcomes in pregnancy. Women with inflammatory bowel disease, compared to the general population, have an increased risk of spontaneous abortion, preterm birth, small for gestational age, hypertensive disorders of pregnancy, including preeclampsia, postpartum hemorrhage, and a 44% rate of cesarean section, most of them elective out of fear of disease. And stopping a biologic, which again, out of fear, you're on a biologic, it's stopped in pregnancy, or it still is in many, in rheumatology and psoriasis, less so in IBD. But when you stop it, you can see in the bottom here, reducing or stopping leads to an increase of disease flare. And disease flare, again, is the biggest driver of adverse outcomes. Many of my colleagues who are rheumatologists, and I apologize if there are any rheumatologists out here, but they say, oh, women with rheumatoid arthritis get better in pregnancy. There is not a single study that shows that. In fact, this study from the national inpatient samples shows that women with RA were more likely to develop complications of pregnancy, both during pregnancy and the postpartum and in their neonates. The American College of Rheumatology conditionally recommended continuing TNF during pregnancy despite available safety data, and the voting panel agreed that if the patient's disease is under good control, these medicines can be discontinued. This is now. But in this article of a prospective registry from rheumatology from Sweden and Denmark that looked at 1,700 patients with rheumatoid arthritis, there was an increase in preterm birth and small for gestational age in RA compared to the general population, and that odds ratio increased to 3-fold with active disease. So there is data that active disease increases harm, not just in IBD, but rheumatoid arthritis as well. We know that there's a strong role for inflammation in pregnancy and in pregnancy outcomes. So the significant increase in pregnancy and neonatal complications is closely linked to disease activity and inflammation, and stopping these low-risk medications and steroid-sparing therapies lead to increased suffering for the mother and postpartum flares and worse outcomes for the infant. And as Venkat already said, healthy mother equals healthy baby. So what are some of the study designs and limitations? And these have been brought up before. Pregnant women are not included in clinical trials. There's unmeasured confounding in uncontrolled studies, as we all know. And disease activity impacts the decision to continue or discontinue therapy. It's not random. The choice of therapy is not random. It is linked to their disease, how severe it is, and what they have. There's low event rates for these adverse events. So if you have a series of 100 patients or 1,000 patients or 10,000 patients, you may not pick up the signal. The types of studies that are used for the most part are large data sets, so a bird's eye view, and the highest quality studies are large population-based studies from countries in Scandinavia usually, where they have longitudinal assessment, parent-child linkage, and a good assessment of diagnosis and pregnancy outcomes. However, these are limited by a fair assessment of medication because they can only measure prescription and not whether the patient's actually taking the medicine, and a very poor assessment of disease activity and more granular data. Insurance claims data has a lot of problems, as anyone in EPI knows, including a lack of clinical data, fragmented care, and absence of key data. Registries like Piano, which we'll talk about, has more granular data, but it's prospective data collection, but it's limited by funding, objective markers of disease, and it's finite relative to the career of the researcher. Pharmacovigilance data limited by the patient and provider reporting. People are more likely to report a complication than a healthy pregnancy, and you also often get incomplete information. And case series, again, small numbers, biased reporting. I'm going backwards, sorry. Okay, so what is the existing data? I want to tell you a little bit about Piano, which is Pregnancy and Inflammatory Bowel Disease and Neonatal Outcomes. This is a prospective national registry of pregnant women with inflammatory bowel disease, started in 2007. I am the primary investigator. And we divided people into 4 groups: the unexposed, which could include people on steroids, 5-ASAs, and antibiotics, azathioprine or thiopurine group, biologics, and combination therapy. And we defined exposure as use any time within 3 months of conception throughout pregnancy. And we compared the offspring of women exposed to a medication to women, offspring of women with IBD not exposed. We looked at multiple different outcomes, including pregnancy and neonatal outcomes. We administered questionnaires each trimester of pregnancy, 3 times in the first year of birth, and then annually, and we're continuing to follow these patients out to 18 years of age. So here's some of the data that have been published. Corticosteroids. I often hear from providers that, oh, I'll just stop their medication, and if they flare, we'll give them steroids. This actually leads to increased rates of preterm birth, low birth weight, and NICU admission. And of course, the use of steroids is closely tied to disease activity. It's hard to separate the two, but the whole point is that you don't want disease activity, you don't want steroid use, you want them to be on a steroid-sparing effective therapy. The primary results of Piano were published in 2021 in Gastro. We looked at 1,490 prospectively followed IBD pregnancies. 379 were not on the drug of interest, 242 were on thiopurine, 642 on biologics, primarily TNFs, and 227 were on both biologic and thiopurine, so about 1,000 biologic-exposed pregnancies. We found no increase in birth defects, spontaneous abortion, preterm birth, low birth weight, or infections in the first year of life, but did see increased infections with preterm birth. We saw an increase in spontaneous abortion with disease activity, and we used the Validated Ages and Stages questionnaire to look at developmental milestones and saw no reduction in developmental milestones. We measured placental transfer, as Edwin talked about, and we measured maternal and cord blood or infant blood on the day of birth. The highest transfer was with infliximab. The lowest, of course, is certolizumab, which doesn't have the FC portion. But vedolizumab, despite being an IgG1 antibody, had a lower level in the infant than the mother. So when this data first came out, the immediate reaction was, oh, we should stop the biologic early. So in Europe, they stopped. Europe has a more of a glasses half empty look at medications in pregnancy. U.S. tends to be more glasses half full. So they decided to stop at 22 weeks gestation. And that was in their official guidance. And it was not until 2 years ago that that was changed to match U.S. recommendations because their own data showed an increase in disease activity and worse outcomes with doing that. But the concern was if you have this placental transfer, if you have therapeutic levels of drug in the infant for the first several months of birth, do they have higher rates of infection? And we showed in Piano that there was no increase in infection at 4 months of age and at 1 year. And we looked at infection rates relative to the level of drug in the infant at the time of birth. And there was no association with drug level at birth and rates of infection. So based on that, now we don't stop the biologic at all during pregnancy. We continue it throughout. A systematic review and meta-analysis looking at 8,000 women with IBD showed who were exposed to biologics showed no increase in infant infections, infant antibiotic use, or non-infection-related hospitalization. So again, confirming this data, showing that these biologics do not cause harm. This data from Antoine Meyer, who uses the French national patient sample. It looked at women exposed to anti-TNF or thiopurines and showed no increase in the risk of early life malignancy in children. Another question that comes up. We ask about infection, we ask about immune suppression, we ask, does that lead to malignancy? And so far, at least in this very large number of 3,700 thiopurines, 3,400 TNFs at 3 years of age, going out to 11 years of age, no increase. Again, very reassuring data. From Piano, as mentioned, we looked at developmental milestones out to 12 months, out to 4 years, showed no decline in developmental milestones. We actually showed that patients on TNF had statistically superior developmental milestones in every category compared to the national average and compared to even within Piano. Not to say that TNFs make your kids smarter, but the whole idea of controlling inflammation is what allows these kids to lay down their neural pathways properly. What about the newer biologics? So we published in Piano our data on ustekinumab and vedolizumab, again showing no increase in harm both for the pregnancy and for infant outcomes. And then Antoine Meyer, again using the French database, looked at 398 vedolizumab pregnancies, 464 ustekinumab pregnancies. Again, no increase in harm for all of these important outcomes. But as Anthony Sciali, who's not here, will say, it's not just congenital malformations. What else can happen with these medications? So working with Susan Fisher, who's a placental scientist at UCSF, a question was raised about vedolizumab. Vedolizumab inhibits alpha-4 beta-7, which can inhibit MADCAM, which we showed was involved in the process of placentation. So if you inhibit MADCAM, are you going to have issues with placentation? And this was just a pilot study looking at two samples. The first one here, the patient also had pulmonary hypertension. You can see this is a normal placenta at birth. You can see how this looks distinctly abnormal. But the second patient was born 39 weeks gestation. Mother was completely healthy with her ulcerative colitis, had no other issues. Again, abnormality compared to a normal placenta. So are there other things we're missing here? We are conducting a larger study now with multiple biologics. The question, it is probably not the vedolizumab is my hypothesis. This is more a result of inflammation, having inflammatory bowel disease, but it'll be interesting to see what these placentas look like when we finish. But maybe this is why these patients have higher rates of preeclampsia, higher rates of hypertensive disorders of pregnancy and preterm birth. It may be actually related to the impact of inflammation on their placenta. Small molecule. So I feel very comfortable when a new biologic comes out to continue it in pregnancy. I feel reassured by the minimal to lack of transfer in the first 14 to 16 weeks of gestation. But small molecules will transfer, and tofacitinib showed teratogenicity at supratherapeutic doses. Upadacitinib showed teratogenicity at the doses we use in humans. Of 30 milligrams daily. So that does raise concern. There is now some data, again, from clinical programs. This is 85 maternal exposures, 99 paternal exposures of tofacitinib, showed no increase in live births and pregnancy loss or birth defects. Same for, this is in press, drug safety, looking at upadacitinib, 128 maternal exposed pregnancies, 80 of which from clinical trials, again showed similar rates of live births, spontaneous abortion, induced termination compared to what is expected. And now, what about breast milk? I know we're not talking too much about breast milk, but in Piano, we did collect breast milk samples and found the amount of transfer was really minuscule. But all of the biologics, including certolizumab, had transfer. We found no increased rates of infection or impact on developmental milestones with patients who were breastfed while the mother was on a biologic. What is the impact? Again, we talked about vaccines a little bit. If these patients have detectable level of biologics for the first 6 months of life, will they have normal response to vaccines? We looked at Haemophilus influenza B and tetanus, a poor man's B and T cell test, and found that the rates of response were similar to patients infants of mothers with IBD who were not exposed to biologics. That was reassuring. We had 40 inadvertent rotavirus exposures in our TNF babies. They did just fine. This has also been shown in European data as well, and I want to make sure that you all are aware of the study from Lancet looking at rotavirus vaccine, and this is a prospective study of infants exposed to biologics. And they gave 168 biologic-exposed infants rotavirus vaccine. As you know, rotavirus can only be given within the first 3 to 4 months of life. After 6 months, it's not given. So if you say no rotavirus in the first 6 months, the baby never gets it. But they found no harm. They said lymphocyte subsets and the safety of live rotavirus vaccine are not affected by in utero exposure to biologic agents. So at this point, we are letting our patients on TNFs get rotavirus vaccine. You can argue in the United States in most areas because of herd immunity, rotavirus vaccine may not be that important, but in other parts of the world it is, and it's fine to give it to patients exposed. BCG vaccine is different, especially if it's an anti-TNF, because anti-TNF does have a higher rate of TB having to do with mechanism. So there was one death in a European study of disseminated BCG in an infant given the vaccine at 1 month of age. But BCG vaccine can be given after 6 months of age. So rotavirus is fine within 6 months. BCG, we still recommend after 6 months. MMR in high-risk populations can be given at 6 months. So why do the Europeans, the Asians, the Americans all have very different guidelines? This May, we all got together, did a grade and RAND process, and had a global consensus conference to have one standard for pregnant women globally and to help spread the word. Our recommendations: continue 5-ASA, continue sulfasalazine, steroids when necessary, stop methotrexate, continue thiopurine. The FDA recently came out with a warning about intrahepatic cholestasis, which surprised all of us, but measurement of liver enzymes and metabolite levels, consider split dosing, continue TNF on therapy. So the U.S. and Europe agree. We will not be stopping TNF early. We will continue it on schedule. We'll continue vedolizumab on schedule. We'll continue ustekinumab on schedule. It's okay to start these in pregnancy if they're not on it. Biosimilars, so the Europeans didn't even understand why we felt we needed to put this in the consensus, but this is a common question that comes up in the U.S. Biosimilars can be continued in pregnancy. We consider their safety equal to the originator drug. New anti-IL-23 therapy, even though it has not been well studied yet, we feel based on mechanism and monoclonal antibody status, mirikizumab, risankizumab, gazilkumab can be continued. Small molecules should be discontinued. And particularly for the JAKs, though, we said that unless there's no effective alternate therapy. So I have had patients go through pregnancy on TOFA, on UPA, because there truly was nothing else that worked for them. Inactive vaccines should be given on schedule. We suggest live rotavirus vaccine can be given to children exposed to anti-TNF and recommend BCG be avoided in the first 6 months. That's it. Thank you.
Thank you, Uma. Okay, so we now have the last speaker for this session before we take a break. I'd now like to introduce Laura Bozzi, who's an Associate Director of Benefit Risk Assessment at Johnson & Johnson, where she leads clinical and safety teams in patient preference studies and benefit risk assessment.
Thank you. I'm Laura Bozzi, and I'm honored to be here today to talk about the benefit risk conceptual framework for in utero exposure to immunosuppressive medications. Listed here are my disclosures. So infant exposure to biologic has led to concern as to whether live vaccinations should be delayed due to concerns about reduced vaccine effectiveness. But by delaying these vaccinations, this may put the child at risk of infectious disease. Now, some have argued that to alleviate these concerns, possibly the pregnant person should stop taking these biologics during pregnancy. But as we previously heard, it can put the pregnant person at risk by not taking medications that they desperately need to serve their autoimmune conditions. Additionally, little is known about whether biologics cross the placenta or in the breast milk, as evidence is limited to case reports. So our study team took a step back, and we wanted to assess the trajectory of the pregnant person and the infant. So first, we have a person of childbearing potential who is diagnosed with a condition that warrants a biologic. They begin taking the biologic, and then they become pregnant. The question becomes, do they continue taking the biologic through pregnancy, or do they discontinue? Then once the baby is born and they decide to breastfeed, if they had stopped taking the biologic during pregnancy, do they restart it during lactation? And if they had continued it during pregnancy, do they continue it during breastfeeding? And now lastly, you have an infant who's been exposed to a biologic, whether it's in utero or breastfeeding, you delay the live vaccine or not. And so there are multiple decisions that come in over this time frame, and we have two parties here, both the pregnant person and the infant. So our goal was to develop a conceptual framework for exposures to biologics to help have an evidence-based approach to support pregnant person and their infant in safety and benefit risk to help form future decision-making. Now, biologics was a motivation for this work, but I have generalized this talk today to include immunosuppressive medications where applicable. So this problem is so complex that we needed to dissect this individually. So we created a conceptual model to help us illustrate the different scenarios and what questions we needed to answer. Now, there are not just multiple decisions at given points in time, but rather a series of decisions that are nested within each other. And that's why we needed a nested conceptual model. So this figure is adapted from our publication earlier this year in Clinical Pharmacology and Therapeutics. So again, first we have a person of childbearing potential, and they become pregnant. Do they continue the medication or not? That's the second scenario. Third scenario now, they're breastfeeding. Do they continue the medication during breastfeeding? And then lastly, you have the infant exposed, whether it's in utero or breastfeeding, do you delay vaccination? So the focus of our talk is scenario 4, but we have to acknowledge that it is within the context of how they're exposed, and in this case, it's within scenario 2. So to tackle this problem, we used the Pharma-BRAT framework. The Pharma-BRAT framework was developed by industry under the guidance of the Pharma Foundation to structure and conduct benefit risk assessment. And so for this conceptual framework, we just focused on the first 3 steps of the Pharma-BRAT framework. The first being defining the decision context. Inside the decision context, we have several key principles of the treatment, comparator, decision makers, indication, time frame, and subgroups of interest. We then need to identify outcomes. So we use a visualization as shown on the right called a value tree to help key stakeholders align on endpoints that we would use in our benefit risk assessment before we conduct the benefit risk assessment. And then we would need to identify and extract possible data. So for each of these scenarios, they have their own decision context and their own value tree. So for scenario 2, the treatment here is continuing biologic therapy during pregnancy compared to discontinuing treatment. And we have 2 important people that are part of this population. You have the pregnant person and the infant. Usually when we talk about benefit risk, we're just dealing with one person being the patient, and now we have 2 entities. This conceptual model was made for any autoimmune condition that warrants a biologic. Now, the framework can be tailored to an individual indication, and on the value tree I will present shortly, you can vary the endpoints based on the molecule as well, so we can tailor it to small molecules. But for today, I'm presenting our example on biologics. The time frame here is conception to infant 1 year of age, and that is because we want to see the effects through the infant's first year of life. And there's so many decisions that come into this that we need to consider during pregnancy and after, but really the primary decision maker here is the pregnant person and their physician. There are also several subgroups of interest, including disease severity, rate of transplacental passage, and medical obstetric history, which includes the timing during pregnancy. Now, when looking at the value tree, again, it's important to remember we have 2 entities. So we have the potential benefits to the pregnant person and the infant, and potential risks to the pregnant person and the infant. Now, for this value tree, this is for scenario 2. So this is the full value tree. So we're looking at the decision of continuing medication during pregnancy versus discontinuing. These endpoints listed here were identified through an in-depth literature review conducted by our study team, and then an iterative process with key stakeholders in immunology, gynecology, neonatology, and epidemiology to identify the endpoints that we thought were appropriate for biologics. Again, this was made with biologics in mind, but you could alter this for a given disease condition. This is already conceptual for a vast array of conditions. And also, you can tailor this based on the given drug or mechanism of action. So when focusing on potential benefits, when you're continuing a biologic during pregnancy versus discontinuing a biologic during pregnancy, there are potential benefits to the pregnant person, including symptom control and improvement, and improved pregnancy outcomes. And there are also potential benefits to the infant in terms of improved health status, with reduced chance of preterm birth and improved quality of life. And I think we heard several times today that a healthy mom can lead to a healthy baby, so that's very important. However, continuing a biologic during pregnancy also poses potential risks. For the pregnant person, these potential risks are really just consistent with what we typically think when we're talking about biologics. So usually we consider serious infection, hypersensitivity, or risk of malignancy. The potential risks to the infant are serious infection, congenital malformations, and vaccine-related complications. This last potential risk of vaccine-related complications shows how scenario 4 of possibly delaying a vaccine fits into this scenario 2 and how these are nested within one another. Now, remember, each scenario has its own decision context and its own value tree. So now this is scenario 4. So an infant has been exposed to a biologic. That could have been in utero or lactation. And we want to consider whether we should be delaying a vaccination. So today, for the treatment, we're thinking of attenuated live vaccines on schedule versus delaying it to 12 months. The population here is infants exposed to the biologic, whether in utero or breastfeeding. The indication we chose here was for rotavirus prevention. We chose rotavirus vaccine because it's one of the few live attenuated vaccines that are given in the infant's first year of life in the U.S. The time frame here is birth to infants 2 years of age, and that's because if we're going to delay a vaccine to 12 months, we wanted another year of follow-up on the infant. For scenario 4, we view it really as a policy-related question, which means there's a lot of different decisions and decision makers. But the primary decision maker, again, is the infant's caregiver and the infant's clinician, but they would be informed under regulatory and clinical guidelines for vaccinations. And the subgroups of interest really rely here based on exposure, so whether in utero alone or breastfeeding alone or both in utero and breastfeeding. Here is the value tree for administering the rotavirus vaccine on schedule versus delay. For rotavirus, the vaccine is usually administered at months 2 to 3 for the infant, so that would be compared to delaying it to month 12. So the potential benefits of vaccinating on schedule include reduced chance of rotavirus in that time period from 2 to 12 and reduced severity of rotavirus from months 2 to 12 in terms of non-fatal dehydration and infant death. Now, vaccinating on schedule also comes with some potential risks. As we mentioned, vaccine-related side effects that can include intussusception and hypersensitivity, and also vaccine-related viral infection from months 2 to 12. So this nested conceptual benefit-risk framework for the use of biologics was established for preconception, pregnancy, postpartum, and infant time periods. But now that we have a conceptual framework to work with, we would really want to quantify these benefit-risk endpoints. And usually in benefit-risk, we actually handle this with clinical trial data. In this case, we've been looking to real-world data to see if we can solve these types of problems. So in the case of scenario 2, benefit-risk assessment can partially be quantified through claims or registry data through mom-baby data linkage. However, mom-baby data linkage can be very complicated, quite complex, and often lacks long-term follow-up in the infant. And also in claims and registry data, as we know, sometimes there's missing or incomplete info when it comes to certain endpoints or unmeasured confoundings. But for scenario 4, in scenario 2, we can partially quantify this. For scenario 4, where are we going to get the data? As we've heard earlier today, it's really challenging to find out once you have an infant exposed to a biologic, you've tracked someone through pregnancy, now you have an infant exposed, and now we need to find ways to understand whether we should be administering this vaccine on schedule or delaying. And so it's very limited in terms of claims and registry data. Clinical data would be great, but it's hard to get everything we need in all one place. So we really feel that a multifunctional stakeholder approach is needed to understand these problems. And by working together with folks from immunology, gynecology, epidemiology, and especially patients and parents, I think we can really find ways to develop this data and move forward together to quantify the benefit risk to help infants exposed to these immunosuppressive treatments and really find the best way forward for the mom and baby. And so we propose the creation of a consortium, we call it BRIGHT, but in order to really answer these real-world problems with real-world data, A consortium will allow for the standardization and acceptance of the benefit-risk framework while also tailoring it appropriately to each disease area and each mechanism of action so that we can appropriately answer these questions in terms of benefit risk and conduct evidence-based analysis to answer these important questions for infants exposed to the immunosuppressive treatments. So I hope this is something that we can continue to discuss throughout this meeting, and it'd be great to meet all of you in the room today to discuss further. I want to thank you for having me today. I also want to acknowledge our study team. There's a few of our co-authors in the room as well, along with mentors and colleagues who've supported this project, and they're members of the Johnson & Johnson Pregnancy and Lactation Advisory Group who also helped us advise on the different concepts that we've presented today. Thank you.
Thank you, Laura, and thank you to all our speakers. Unfortunately, we've run out of time, and so we're not going to have time for questions, but please feel free to ask the speakers if you have any questions during the entire day today and tomorrow. So it's time for a break, and we're going to have to cut back the time because we are running a little bit behind schedule. So please take a short break and try to be back here by 11:25. Thank you. Okay, so welcome back, everybody. We heard a lot of really important information this morning, and so now I welcome you all to our first panel session on current clinical and safety considerations. We have one panel member who was going to join us online. Do we know if he's able to? Or? No, okay. So we had Kevin Alt, who is an academic OBGYN, and he was going to join us virtually as part of this panel, but unfortunately, due to obstetrical emergencies happening at his institution, he had to cover for his colleagues. The life of an OBGYN, a lot of us are familiar with that, so we're sorry that he couldn't make it today, but we'll go ahead and proceed. Thank you. So to start off this panel, I'd like to invite each panel member to introduce themselves briefly, provide a little bit of information about their background, their expertise, and what they do, and then also to reflect on what we heard so far this morning, if you have any kind of preliminary thoughts that you'd like to share. Maybe we'll start at the end. Okay, so yeah, we'll start that way, and then we'll proceed this way. Thanks.
Hello, good morning, afternoon, good evening to everyone. My name is Maria Fernandez. I'm here representing Eli Lilly. I'm a drug development company representative, industry representative. I'm OBGYN, that's my background, but I've been working in the drug industry for the past 7 years. I just wanted to start saying that the ideas I'm going to share today comes from my background and not necessarily from my employer, that is Eli Lilly. So, first of all, thank you very much for inviting me for this panel, for this really, really interesting and important topic. I wanted to highlight the importance of gathering good data on use of immune suppressive drugs during pregnancy. We all heard here today, often immune suppressive drugs are necessary for patients with autoimmune diseases, with patients that are are transplant recipients and have other health issues. And sometimes, most of the times, there's a really high pressure because they don't really know the effects, and it's always a challenge, right? I also want to say that, we have, now I wanted to bring this to this team, that the ICH is the International Council of Harmonization is now gathering and putting together this guidance that will support the inclusion of pregnant individuals in clinical trials, especially for drugs that are expected to be used during pregnancy and for drugs that with a perspective of benefit. And this guideline is going to try to bring the tools to enable this clinical trial, this study, right? So I just wanted to say that we had amazing talks today. I think that I'm really excited to be here, and looking forward to the discussion.
Thanks, Maria. And we have Latay next. No, sorry, we have Vani next. Yeah, thanks.
Thank you, Leila. I'm Vani Vannapagari. I am the head of epidemiology and real-world evidence at Weave Healthcare. Weave Healthcare is one of the companies that works, we are the only company that works exclusively on treatments, prevention, remission, and cure for HIV. And I'm also the industry co-chair for the Antiretroviral Pregnancy Registry, the APR. The APR has been in existence for over 30 years now, monitoring for the early signals of birth defects, and it's an industry-wide collaboration across with 27 pharma companies. So a lot of my experience and expertise has been coming out of that. And the morning sessions have been fantastic, and starting from the commissioner highlighting the need for us to come together to generate the data. It's not just about generating the relevant data, but accelerating the generation of the data. In the HIV field, in the antiretroviral field, previously, it used to take about 11 to 12 years for data to come out, but now that time frame has shrunk to about 5 years for the first PK data to come out. From the time a drug is approved, that's still way too long. WHO about 2 years ago has set out a call to action to various stakeholders to accelerate this generation, and we at Weave Healthcare have come up with a framework as to how we can achieve that. It's not that every single drug in development can be developed and the data acceleration for use in pregnancy can happen, but the due diligence and consideration has to happen. We are now requiring our drug development teams who are planning for first time in human trials to come up with a pregnancy data acceleration plan. So in that sense, we are moving in the right direction, and I look forward to having further discussions.
Thank you, Vani. And we have Natalie Hayden next. Thank you.
Hello, everybody. My name is Natalie Hayden. I'm absolutely thrilled to be here. For those who don't know, I am a patient. I was diagnosed with Crohn's disease in July 2005 at age 21, 2 months out of college. And I went on to work in television news for 10 years and suffered in silence. And when I left the news business in November 2014, I vowed that I would share my love for storytelling and make a difference and be the voice I so desperately needed to be on diagnosis. And since that time, I've had 3 healthy children, all exposed to Biologic. My kids are ages 7, 5, and 3, and I'm a patient advocate for inflammatory bowel disease. I have a blog called Lights, Camera, Crohn's, and I work diligently every single day of the year to be the voice that this community needs. And when I see the work that each of you are doing, the research, the time, the energy, the genuine compassion and empathy for the patient community, it does my heart so good. So I thank you all for the important work you are doing. I can't tell you enough, as a woman who is giving my child biologic exposure and feeling the children kick in my belly, third trimester, and I'm injecting something into my leg, how emotional that is for patients, and what goes into every single decision that we make that you talked about in your discussion, that risk-benefit. It's imperative that this is something that is thought about and you are giving such careful consideration to. And I can tell you from a patient perspective that even as a mom with children that have grown up through the years, every single decision I make is because of that thought process. And I can't thank Dr. Mahadevan enough, because the piano study in particular really is paving the way for future families and taking out that gray area for women when they have to think, do I stay on my therapy? Do I give my child immunizations? So many decisions are being able to be made confidently now because of studies like that one.
Thank you, Natalie. Next we have Latay.
Good morning. I'm Latay Bradford. I am a family physician at the University of Maryland Capital Region Medical Center in Largo, just over the Beltway. And I am hoping to be able to provide some perspective from a clinician who makes decisions on a daily basis about these benefits and risks for both patients, the pregnant person and the fetus. As many of you know, pregnancy is a condition full of excitement and angst all at the same time, and there are many difficult decisions that are being made, whether or not it's taking medication for an immunocompromising condition or mental health diagnosis or substance use disorder. There are a lot of decisions that have to be made, and moms want to do what's best. They come to me as their trusted healthcare provider, and they want to understand what is best for me and for this developing fetus. And so it's really important that I, as a clinician, be armed with the data to suggest, the evidence to suggest best practices in clinical medicine. And so I really appreciate this forum, this workshop. Thank you so much for all of the work that you all are doing. I just want to kind of take off my research hat for a minute and just leave on the clinician hat and say that we are really at the crux of trying to both provide evidence-based guidelines and provide equitable health care. So I'm so glad that the commissioner mentioned the intersection of social determinants of health and biological determinants of health, because whether it's health literacy or it's inequities in maternal and infant outcomes, all patients deserve to be receiving the same message. We all need the right evidence. We need the right talking points and the right data points to present patients with best practices across the board, whether they're in California with Dr. Uma or over here in Maryland. So I really appreciate this forum. I'm so excited about the data that's coming out of this discussion, and really look forward to learning and growing together.
Thanks, Latay. And Uma?
Hi, I'm Uma Mahadevan. I'm a gastroenterologist at UCSF that specializes in IBD and pregnancy and drug safety. And I think my main comment is that there are a lot of limitations in getting information to providers, information to patients. Most GIs don't even go to national meetings anymore. How are you going to get this information to them? How are you going to get them to participate in putting in data for clinical trials or for understanding pregnancy safety? So I think there needs to be a need for creativity. AI may be the way, but natural language processing right now is not there. But we do have EPIC as a very powerful tool if it can be connected across centers like the University of California has done through California, but that's really, a mile wide and an inch deep, and it's very hard to get the data you need. And so I also think that you'll find the most enthusiastic group are actually the patients, as you heard from Natalie, and going directly to patients to collect data. Now that, for example, from Piano, we have electronic case report forms. We verify with the doctors if there's something unusual, but moms know there are complications. what their kids have, and they're actually a very reliable, enthusiastic source of information. And I think when you look at drug safety, that's like a group you should not ignore.
Thanks, Uma. Okay, so I'm going to start off by asking a few questions, but it's always more interesting, of course, when we have audience participation, and it's a nice small room here, so please feel free to ask our panelists questions, but I'll kick it off with a few questions. So is there value in pursuing additional safety evaluations in infants, and what are the unmet needs? And in no particular order, whoever feels that they would like to share their thoughts, please go ahead.
I'll say a resounding yes. There is value in the work around understanding the effects of these medications in utero. As I mentioned, moms want to do what's best for their babies, and physicians want to do what's best for both of our patients. Unfortunately, you may be surprised to hear that patients aren't always reassured by outcomes in monkey babies and rat babies. That really doesn't provide them any consolation. They want to know what's going to happen to my human baby. And so there is a lot of value in the clinical studies that are being done, and including pregnant women in clinical trials. A lot of the unanswered questions are around the particular drug itself, whether it's a biologic or corticosteroid or calcineurin inhibitor, whatever the drug is, but the dosing, the timing of the drug in pregnancy, the duration of therapy in pregnancy, and then the subsequent outcomes. So not just is the drug still present, but does the presence of the drug in my baby's blood translate or correlate with clinical outcomes? So those are questions I see, Dr. Uma presented very eloquently on the piano trial, and so we really appreciate the work being done in that area. I think there's still a lot of unanswered questions from the patient's perspective, and we collectively as clinicians have to get together, as she mentioned, disseminate the information so that we can be providing the same message across the board.
I'll add that while we look at infections as an outcome and hospitalizations as an outcome, that's a very blunt instrument. I think we need to look at T and B cell development in these infants, exposed to a JAK inhibitor, exposed to a TNF in utero. Does that impact how their immune system develops over time? We did that very preliminarily in piano. I didn't present that data, and it looked okay. Thiopurine's less so, but it was very small, and I think that is definitely an unmet need that an immunologist needs to tackle.
I will say from a patient perspective, you want to see the numbers. You want to see the studies that are being done and the work that's going on to prove that what you're doing, staying on your medication, staying on your therapy, is not going to harm your child in any way. Because I think any mother out there, any parent in general, you don't want to do anything. You are as selfless as you possibly can be. And it is an emotional burden on the parent to feel like they already have a chronic illness that there's no cure for, and they feel guilty as it is for staying on their medication. But it's also about reminding the patient that they are actually being the best mom possible by staying healthy for their child so that they can bring them safely into this world. So the data that can be created, the safety studies that can be the research, everything is so appreciated, because when you are told, oh, this will be fine, you'll be all right, the patient wants more. We want sustenance. We want to see the numbers. We want to see the research. So when you can tell a patient, hey, there's this study out of France, I want you to look at this. This is how many people were looked at. This is the response. This is how old these babies are now. This is how they're doing. And part of patient advocacy, what's so important about it is that fellow IBD moms, we can be the proof. So we can say, look, I stayed on my anti-TNF till 37 weeks with 2 of my children and 39 weeks with my oldest, and all of them are pictures of health. They were studied, 3 different studies. I studied with my first child was followed until he was in kindergarten. And I just do that out of the goodness of my heart because there was so much gray area back in 2016 even. I wasn't a patient advocate back then. I didn't know any fellow IBD moms. So I try to show people this is how you can participate in piano. You can really pave the way for future families. And this is all just out of the goodness of your time and heart because you've been there before and you want to make it better for your daughter or somebody else's daughter or somebody who's pregnant right now so that they don't have those same fears about safety.
Yeah, and to add to that, from a drug development perspective as well, we need good quality data to be added to the labels, right? Because not having the data generates a lot of uncertainty, and for both the patients and for the prescriber as well. So I think that, and also how we can mitigate the risk, right? If we know the data, we can have a plan. We can put a plan in place. So I think that's really important.
Great, thank you. So I'll go on to my next question. So what unanswered questions did you have? What knowledge gaps exist about the health and safety of the fetus or infant when a decision is made to start or continue medication during pregnancy?
So as was mentioned before, having a good patient-centered risk-benefit discussion really necessitates having foundational knowledge, right, the data, because how can you have informed discussion without the data? So let's start there. But moving forward, I feel like a lot of the discussions about impact and real-life clinical outcome is what patients are most concerned about. The drug concentrations and, I think the nitty-gritty of the science needs to be translated into clinical outcomes for patients to understand. And I just, again, want to mention health literacy because in communities like the one that I serve in, where there's language barriers and cultural differences and varying experiences in healthcare, we often, label communities as having low health literacy. Well, that's not their problem to navigate the system. It's our problem as healthcare providers to help them navigate the system, meeting them where they are. But I can't do that if I don't have the evidence to go to. And it's very hard finding 10 different ways to eloquently say, I don't know. Patients start catching on, right? So I think some of the unanswered questions are just still along the lines of, given an in utero exposure, what are the potential risks to the fetus? How long? What's the odds of having an adverse outcome? Will it affect their neurological development and, behavioral, educational potential and opportunities? But all of those are obviously downstream from these very early trials and studies that are being conducted now.
Well, I would ask Maria and also Marie Thiel, who are here, part of ICH. I'm often an author on these clinical trial outcome data, and every single one, the patient is dropped. They have to go track them down. Often you don't know the outcome. You don't know the details. So this is critical preclinical data. Why are these patients being let go? I mean, I think that's such a huge opportunity. If they get pregnant in a trial, whether the trial makes you drop the patient, but why do you stop following them?
So from the drug development perspective, if a patient gets pregnant during a clinical trial, we follow that patient until the birth. And usually there's maybe until like 1 year of age. So they're not really dropped. We don't really lose this data.
But in every single study, so many of those patients, is data not known, data. So they were dropped.
So there is, how can I say, we try as much as we can to follow up, that pregnancy until the outcome, okay? But we know that there are, how it is when a patient gets pregnant. We have so much to do, and sometimes it's hard to keep, going back to the site and following up with the PI. But that's usually what we do because it's part of the pharmacovigilance, right? However, as I said, with this new guideline that is trying to be in place. We want to really close that gap, right? We want to make sure that we get as much information about the patient being exposed to a drug as we can, because it's really important data to be captured, right? Okay, go ahead, Mary. Do you have a question?
Yes.
No.
Okay, is it better?
Yes.
Thank you, Uma and Maria. So I may not answer exactly about the ICH guidelines, but more the way we approached it, and we started approaching it at UCB. And I think this is now more and more what pharma is thinking about. There's a big project with Transcelerate as well. Oh, by the way, I'm Marie Thiel from UCB, for people online. So what we have now is in our common protocol template, we actually allow women who get pregnant in our clinical trials to continue. Of course, it depends on the preclinical data that we have, on the animal data, so it's not true for all of what we do, but at least that's an option depending on what we have. And I think this is an important point, and they can continue in the trial, stop their drug, and re-enter later on, because as you know, a lot of these women may actually not continue their pregnancy voluntarily or not, and we want to give them this option to continue. It gives us an option to look at the continuity and the disease activity as you stop treatment, as you continue for these women who decide to stop. So we're giving this flexibility now. You can imagine that it makes the analysis of the data very complicated, so it's not that it can happen all the time, but at least we made it a template. We made it in our protocol template now. So this is something that I think we're going to see more and more coming, so that we can continue to follow better these patients. I want to say though that, and we've conducted many advisory boards with patients, and Natalie, maybe it's also a question to you, is that for these prospective studies, a lot of the patients do not want to continue. And so I think it's also this mindset that we've had for decades of you're pregnant, you do not take anything, that even if the physician, and Uma, you mentioned physicians want sometimes to ask to stop the treatment, they're not aware of the data, but even if they are, we've heard a lot of patients saying, I want to stop. So it's a true paradigm shift. There's a lot of education that we need to do. It's not possible to do it for all products, but when it's possible, this benefit-risk assessment of what is the risk of stopping the drug is something we need to evaluate better and share better with physicians, but also with patients. I don't know, Natalie, if you have something on that.
Absolutely. So the family planning discussions are imperative. So patients shouldn't be talking about what they're going to do on therapy after they've already conceived a child. If you're a provider and you have a woman who's of childbearing age, you might not want to insinuate that they should become a mom, but say, if you plan to be a mom in the future, I just want to inform you that the medication, the therapy you're taking is safe and effective in pregnancy. We'll cross that bridge when we get there. I know my provider, back when I was still having children, she told me if my anti-TNF failed me, what my next biologic would be, what the game plan would be. We always had a plan in place. And I think it's also important for patients to speak up and be their own best advocates to their providers and say, how can I prep my body best for pregnancy? I know I had bowel resection surgery in August 2015. I was engaged to be married, and I was finally in remission after a decade of aggressive Crohn's disease. And I thought, now's my time. I have to capitalize on this remission and get pregnant. So I talked with my GI in November. I was getting married in June. She said, Natalie, we're going to prep your body. We're putting you on a prescription prenatal. We're putting you on folic acid. I'm going to do a colonoscopy a month before your wedding. We had an entire game plan in place, and that made me feel so confident going into motherhood that my body, she came in after my scope and she said, Natalie, you have the green light. And that's what she did for each of my children. And I knew going in, I'm keeping on my medication, I'm not stopping my injection. It wasn't ever this, what am I going to do? I'm 6 weeks pregnant. Do I stay off my meds? Do I go on them? So having those conversations early on makes a world of difference.
I always say nobody gets more unsolicited advice than a pregnant woman. So.
I think another set of unanswered questions is around coexisting comorbidities. So oftentimes moms are facing more than one diagnosis. They're on more than one class of medication. And in my practice as a family doc, I don't want to take for granted that everyone understands. Family physicians take care of the entire family across the lifespan. So I have newborns, I have elderly patients, and everyone in between. And when I take care of pregnant persons, sometimes they have chronic hypertension, gestational diabetes, or chronic type 2 diabetes. And so understanding the impact of their coexisting condition along with an autoimmune diagnosis, and that treatment is also a black box at this moment where we don't quite understand the effects of fetal development with hypertension and ulcerative colitis, or the treatment of hypertension and a biologic. So I do think that including patients in trials that have more than one diagnosis is an important part of our investigative studies.
Oh, no. Okay, so we've received a question online, and I'll ask our panel members to try to address this question. So could you please comment on the difficulty of obtaining serial blood or tissue-based assessments in neonates and infants who are generally healthy, but whose mothers may have been exposed to a drug or biologic during pregnancy? Depending on the study, infant sampling is not always embraced by patients, and reasonably so.
So in piano, if they were exposed to a biologic and had detectable levels at birth, we would check again at 3 and 6 months. And so it is definitely a commitment from the mom. I'm fortunate at UCSF we have pediatric phlebotomists. So there's a pediatric clinic where children go, and so they're used to drawing children. I think that is important, but all of these patients are seeing a pediatrician or a family medicine doctor who would be drawing blood potentially on infants. So it's not easy. Interestingly, at birth it's easy because it's in the hospital, and the mom often doesn't see it. 3 and 6 months is harder. 1 year is really hard because when you try to do the T and B cell study, a lot of it has to do with the mother's commitment. Often they want to know that the biologic is cleared from the blood. So part of it is, what does a mom get out of this? We do share the result with the mother, and then they know. So some of it's reassurance. But I think definitely having good pediatric blood draw is the key, and for the mother to understand and buy into the reason for testing. I hope that answered the question.
I guess I'll just add from the patient perspective, because I was one of those moms who didn't want to see my baby get a blood draw after 3 months old. It was just hard to witness, and it was a lot easier in the hospital when I had my C-section and they took the cord blood and they took his blood and they took my blood. And as I took them to the pediatrician, I just didn't want to see them go through that. So it is important, I think, as you said, to just say you can see how much is left in their blood as well. And I never thought about it in that way. And I think just explaining that to patients and showing them what those numbers actually mean, because I remember, and changes were made after it, as after my youngest, who was part of piano, I received the numbers in an email, and it looked daunting to me because I didn't know what those figures meant. They were just numbers on a screen, and they looked worse than they were. And I said, I need some more explanation. What does this exactly mean to me? And I know that Piano shifted how they communicate those results to patients, and that makes a world of difference too, because we are not, for the most part, medical professionals, and we're giving our time, our energy, our blood, sweat, and tears to these studies, but it's difficult for us to sometimes understand the results of what we are receiving. So understanding and using that health literacy, to your point, everybody has a different degree of what they understand. So just remembering that when you pass along that information.
Anybody in the audience who would like to ask the panelists a question?
I don't have a question, but I have the same experience as you have, Uma. In our placental transfer study, 99% of the samples were collected from the baby. At birth, of course, easier at birth, and then at 4 weeks and 8 weeks. And we only lost 1%, which was, I think, just hard to draw. We did a heel prick. There's nobody more altruistic than a pregnant mother or a new mother. Really wanted, as you explained, Natalie, to better understand and know for the future and for the future, her own future and the future of others. So very similar experience. I don't think this is a huge issue. For us, we had nurses. It wasn't done at the hospital. We had nurses going home and taking the blood from the baby to avoid the mother traveling. But very similar.
Okay, thanks, Marie. Okay, Venkat?
I have a comment and a question. Thank you, Natalie, for being a patient advocate. We need a lot more of you. In our clinical experience, at least, patients are very willing to participate in clinical studies as long as you tell them why are we doing it and how we are going to do it. I think we need to do more of that. I think one of the issues that we face as medical professionals, we don't do enough commitment to collect data and share data, perhaps because of the limitations in time, limitation in the expense that's involved in collection of this data. My question to the panel will be, once we have study observations or clinical data, there was a study which said it takes about 15 to 18 years before it comes to practice as in a routine clinical. That is ridiculous. I think the question is, what should we do? My thought is, I think the professional societies are not doing enough. They need to do a lot more to translate the clinical observations, clinical data that's available. You don't have to wait till an individual person reads about it. I know Uma brought up the issue of an average gastroenterologist may not be going to a meeting and learning about it. I think somebody like a professional society need to take charge and get the exposure to the rest of the medical profession so we can take advantage of the knowledge we have. Thank you.
I can comment on that. I think one of the ways we've done it, it's much, much quicker in GI, at least in IBD, and one of the ways is consensus statements and guidelines, which come out every few years. For example, we recommend that all women with IBD be started on aspirin based on the Anchor study to reduce preterm eclampsia. And that went, but not all OBs know. So we as GIs are telling our patients, and then their OBs are like, you don't need aspirin. And so also communication across disciplines is very important. And so trying to have cross-disciplinary combined guidelines is something of importance.
So at the time of a drug's approval, there is very limited data to no data at all, the human data that is included in the labels. Generally, label has the non-clinical toxicology, animal data. As one of my mentors says, data in one pregnancy is more than data in 0 pregnancies. So for a physician in the clinic, having data even from a handful of pregnancies is really useful. So one of the things that we're pushing for is not to wait until enough data accrues in large, sufficient numbers to make conclusive, definitive conclusions, but rather keep publishing and keep communicating the data as those get available. And of course, the guidelines, especially in the HIV area, the perinatal guidelines for ARV treatment and the pediatric guidelines, they are constantly updating the guidelines. So for the pharmaceutical companies, the process and the time it takes to update the labels with data, and that data has to be sufficient. We can't just keep updating them. That takes time. But the guidelines can update, based on the publications, based on the data available. They can review that, and they can update the guidelines quicker than the label updates.
Two other thoughts that I have, and I completely agree that much of our academic research never really gets disseminated and translated into the communities that would most benefit from it. What if we were to take more of a community-based participatory research methodology where from the beginning of study design and conception, community members are engaged and involved? And then what if on the back end, funding mechanisms required data dissemination as one of your metrics? Because right now, publication is what's awarded. Publication is what's incentivized. But if data dissemination and community translation were incentivized, maybe we could shift the model of how research, what the research outcomes are from just publishing in a nice journal to actually making a difference where it matters.
Michael Keller, Children's National. I'm a pediatric immunologist. Thank you to all the panelists. A really wonderful discussion. I have one particular question about, and that's with regard to JAK inhibitors. So as a clinician and someone who focuses on immunodeficiency disorders, I use these not uncommonly in pediatric immune dysregulatory diseases. This class of medication really gives me greater pause than many biologics. And we'll probably talk about this later at our panel. But I'm curious about what the panelists think about. Is there still a substantial gap in terms of clinical needs for inflammatory diseases in pregnant women, such that JAK, and we should be thinking more about what are the real risks of JAK inhibitors. As many of you know, many of these, including alpha-citinib, are considered teratogenic in animal models, and there's precious little data that I'm aware of, except accidental exposures in the literature. And if there is a need, how do we really collect that data more effectively?
It's a challenge. So if you look at a drug like an S1P, so the ozanimods, etrazomods, their uptake in IBD is very low. We're never, ever, ever going to get pregnancy safety data on that because we don't use it that much, and there are other agents that are more effective that we could switch a patient to. I think the JAK inhibitors are far more challenging because if a patient's on tofacitinib or upadacitinib, it's because they've already been exposed to a TNF, because there's nothing else that works for them, and so we end up using shared decision-making with the patient of the risks and benefits. I think tofacitinib almost is in the category of a thiopurine where it was super therapeutic doses that showed harm, and at the doses we use in humans may not be. And so I think people feel more comfortable with tofacitinib. Upadacitinib was teratogenic at 30 milligrams at the human dose. I think there, though, those patients, so I could either stop the drug and have the patient have a significant flare and be very sick, because usually those patients are the most severe, or we talk to them about other ways of pregnancy, including surrogacy, or they continue it. And we've had patients make that decision to continue. Anecdotally, across the globe, we've had a handful of patients go throughout pregnancy on these medications, and they've done okay, but it's truly a handful of patients. And the alternative is the mom being too sick to conceive. I made the mistake of, I have a patient, first pregnancy on tofacitinib, she continued it, she did great. second one, she really wanted to come off because she'd been in remission so long. She came off. We could not get her in control through pregnancy, very complicated pregnancy, preterm delivery, all of that. Now she's controlled on something else, but you do harm by stopping. And so the only answer is you just have to we have banded together globally to get cases and put them together. Not sure what else we can do because there aren't that many of those patients.
This is Josh Unatkat of University of Washington. I subscribe to the view of collecting more data. But I'm wondering, and I would like your thoughts, the panel's thoughts about this, is that when we are at the beginning phases of collection and the publications are put out there, is there a danger of getting a false signal because of confounding issues, And what does the panel think about how to deal with these confounding issues that might be producing a false signal? And how can we better, in a sense, control for these confounding issues as we collect the data? So there's a plus to collecting data, but there's also, I see, a potential negative to collecting data and publishing, little amount of data at the very beginning stages, And all these studies are going to be prospective or, retrospective analysis of data. They're not necessarily always randomized controlled. It's really hard to do. So just some, thoughts on the panel's part about this problem that we could potentially run into.
Yeah, I can answer that. So my background, I'm drug safety, and I'm also a gynecologist. And so I've seen both sides of the discussion, right? So when we say that we want more data, we say quality scientific data, right? So you need to be really careful what to put in from an industry perspective. I'm not only talking about disclosure, I'm talking about label changes, right? We have to be really careful to only add something to the label of a drug after have done, that signal has gone through all that stages that we, for sure, or at least we have a higher probability of causality, right? So I think that all the observational studies and animal studies, they're amazing to raise safety signals, but they're not enough. We need to continue to look into until we have we'll never be sure, right? But at least we have a great chance of causality to be established to add something to a label. Yeah.
No, I understand that. I'm just thinking about some researcher out there who does, puts out a publication, and then that becomes like, the focus point, in terms of, because there's very little data, and that's the first publication out there, and then it sort of, it becomes an issue of sort of unlearning that data point, that may be a false signal.
Yeah. So we have a couple of examples in the HIV arena. One of them was based on animal studies, and more recently based on a cluster of, and a potential signal which happened during an interim analysis, and then, everything exploded. So nations stopped rolling out their HIV treatments based on this, cluster of the events reported. What we need to do is look at the totality of the data, put everything in perspective. And there are times when we don't have that certainty, that definitive answers. That's when you do need to bring in the community. You do need to talk to the individual that becomes the benefit risk and the uncertainty in the data. That becomes a bigger discussion between the physician and the person. Within the HIV community, what happened was based on a potential signal, when the recommendation came that women of childbearing potential, not pregnant, but women of childbearing potential should not be prescribed that data, the community fought back. And what they said is this becomes a human rights issue because we are not just the vessels. Give us the choice, give us the option, discuss the uncertainty. So I think we do need to take that discussion until we collect the data further and there are answers to these questions. We do need to bring in the community and the individuals and their physicians into the conversation.
Yeah, I think I can give you two examples in IBD. One, when adalimumab first came out, somebody used the FDA pharmacovigilance database and said that adalimumab increased the rates of VACTERL, V-A-C-T-E-R-L syndrome. And you know, the good thing is a lot of people don't read the literature in minor journals, so it did, you know, but you also have the responsibility for the people at the podium to put these things in context. There needs to be responsibility from editors to have an editorial for some things that may change practice to make sure that it's put into perspective. But I do think it's the responsibility of the academic leaders in that area to interpret the data, work with the community, make sure that we understand, because that study was poorly done. You can't, for many reasons we won't get into. Another example is the EMA, which is the European equivalent of the FDA, send a letter to all physicians saying that any woman breastfeeding on infliximab, their baby should not get live vaccine. That baby could be 2 years old. And so that was ridiculous. And there was no data for that at all. And the European community of physicians just ignored it.
I think we also need to institute sort of best practices in terms of this analysis of data, in terms of the retrospective.
That's the hard part, because it doesn't get into the good journals. It gets into a side journal, and then it's on Twitter. You know, it's hard to control.
Yeah.
Those are the perils of the open access journal publications now.
Dinesh Stanislaus, I'm a reproductive toxicologist. Uma, I really liked your talk, and I was kind of curious. I mean, it was very encouraging to see in the piano trials no increase in infections, changes in vaccination didn't have any impact. When would you consider, and I kind of noticed that you looked at up to 18 years old in your...
We're following them out to 18. I think most of our kids are 5, 6, 7, 8. Yeah.
So when do you think is, from a practical sense, from a drug development, I'm from GlaxoSmithKline, a drug developing company. So when would you think is the upper limit, practically speaking, if you're going to look at offspring, which I think is very important for, in a broad sense, if you're going to put it in a guideline or something like that?
I mean, I think if you have to say diethylstilbestrol, right? It could be
multiple generations, yeah.
Right. You never know until you know. Yeah. That's the danger. So I don't think there is a time. I think you always have to be vigilant. But I think within the first 4 years is key for malignancy to the infant, birth defects, increased risk of infections. I think that's where you're going to catch the majority is in the first 4 years, but we don't know what we don't know.
And in the IBD space, I'm not that familiar. Is there concern for autism type effects later on with some of the therapies?
So we actually, so the rate of autism in piano is lower than the national average. We're not sure why.
Okay.
And so again, is there a role of brain inflammation, which is reduced by being on therapy? And so our TNF exposed had lower rates, and there may be some there is based on rat models important mechanisms of neural development related to inflammation in TNF, and maybe there's a role. So not in terms of harm, but we don't see an increased signal.
Thank you.
All right. Well, with that, I'm going to close this panel. So thank you very much for all of our panelists, and thank you to the audience for the engagement and great questions. And it's time for lunch. Please be back here by 1:10 p.m. Thank you very much.
Ask that you find your way back to your seats. We are ending our lunch break, and we're moving on with the meeting. We are starting session 2, non-clinical evaluation of placental transfer and immunotoxic potential. I'd like to invite Dr. Josh Unadkant to the podium as he'll be moderator for this session. Thank you.
Good afternoon, everyone. Thank you, Tamara. I'm Josh Unadkant, professor at the University of Washington. It's my pleasure to chair this session number 2. And I'm just going to keep introductions of the speakers really, really brief so that the speakers have maximum amount of time to speak. And then, of course, we're going to have the panel discussion. So our first speaker is Dr. Nick Ilsley. He's a principal and senior scientist with the Placental Research Group. He's also an emeritus professor of pharmacology, physiology, and neuroscience at Rutgers in New Jersey Medical School, and adjunct professor in other departments as well. So with that, Nick, the podium is yours.
Okay, thank you, Josh, and thank you to the organizers for inviting me. Today, I'm going to talk about relevant in vitro and ex vivo assessments of small molecules and biologics. And I've taken as my remit the examination of various models that are used for the assessment of transfer primarily, although they can be applied to things like metabolism as well. Before I do, I should just note that I have no conflicts of interest to disclose. Today's talk, I want to first take a look at the barrier structure and transport systems of the placenta, and as Les has already done some of that, and others also, I can be brief. I want to describe the current standard assessment models, which some people have already also described. And then I want to spend some time discussing the newer models which have or are being developed. So first, the barrier structure. Well, you're all, I'm sure, familiar with this structure, but what we're interested in here is the villus tree through which the umbilical circulation flows, and the villus tree is surrounded by or bathed in maternal blood. And as you can see in a diagram that's very similar to Les's, this is the cross-section of the villus tree in the first trimester and the third trimester. And all I want to do here is to emphasize what he said, that the placenta is a dynamic organ. And so going all the way through from the first to the third trimester, you're going to see a variety of structural and functional changes in the placenta, which means looking only at term is not going to tell you what's happening earlier on in gestation. So just to emphasize that, I want to just go through the barrier structure. So here's the sort of simplistic maternal blood, fetal blood, and here's the syncytial trophoblast, the multinuclear epithelial barrier layer. But you can't forget that there's an epithelial cell between the maternal blood and the fetal blood. There's an epithelial cell layer. There are cytotrophoblast cell layers, and early in gestation, those cytotrophoblast cell layers may provide a barrier as well. So what we're looking at are several different layers of barrier, which, depending on the sort of molecule you're looking at transfer, may be important. Therefore, looking at one component here may give you the wrong answer to what's happening in terms of maternal-fetal transfer. So here again, the transport systems, again, are very simplistic: maternal blood, fetal blood, and syncytial trophoblast. As Les has already talked about, paracellular diffusion and transcellular diffusion. The paracellular diffusion is pretty limited for a number of reasons. One, because the actual transfer through that structure is limited to 1 to 2,000 molecular weight, perhaps, at most. And because these are multinuclear
syncytial cells, which are very big, the number of paracellular channels is relatively limited. Again, transcellular diffusion limited to things that are much more hydrophobic. There are the usual panoply of drug transporters that people have been showing, but I'd also like to emphasize the potential for other sorts of transporters which are considered less. So, for example, the equilibrative nucleoside transporters, which are found on both sides of the syncytial trophoblast and which can carry JAK inhibitors, and potentially other transporters similar to that, which may also carry immunosuppressive molecules. So what are we going to look at in terms of the current techniques? In vitro, the current techniques involve cell lines, for example, JAR, JEG, BUO, which are all choriocarcinomas. And the pros for those are they're immortal, they're proliferative, they have some morphologic similarities to primary cells, but on the other hand, they're mostly triploid or tetraploid, and there are substantial differences with primary cells in terms of expression and epigenetics. Transwell, people have probably seen the Transwell experiments before, where you have a permeable membrane separating fluid reservoirs, and on that membrane you can grow cells. And that's great as a barrier transport model. It can include extracellular matrix layers, and it can include other cell layers other than the barrier layer. The problem there is that the barrier layer is only as good as the cell type utilized to test that barrier model. So, for example, when people are growing BUO cells across a transwell, they try to get them into a confluent monolayer so that you're looking at transport across that layer. And at the same time, usually, they're also looking at overgrowth of those cells in that monolayer, as I have done myself, unfortunately. So there are clearly problems with that system. Ex vivo, the syncytial trophoblast membrane vesicles, microvillus membrane vesicles, basal membrane vesicles. These are great. They're easily prepared, they're easily stored, and they have a faithful membrane representation. The problem is they're just a single membrane, there's no regulation, there's no metabolism or interaction with intracellular components. So they're a very, very crude system, but helpful. Primary cells, cytotrophoblast. They're great, they're diploid, their gene and protein expression is representative of the in vivo membrane environment, and they can be differentiated into the multinuclear syncytial trophoblast cells. Except that usually when you get them, if you're getting them at term, they're non-proliferative. They're genetically dissimilar because they're coming from different individuals, so that may or may not be an advantage or disadvantage. They're quite often, even when differentiated into syncytial trophoblast, rather poor barrier layers, if a barrier layer at all, in 2D culture, and they're pretty limited in terms of duration, perhaps 3 or 4 days. Ex vivo, one of the commonly used techniques is to use explants, and they're great. They've got full structural and compositional representation. There's a potential for superfusion of the explants, but there's some serious problems. There's no fetal circulation, they can't be manipulated, and frankly, they degenerate, and they start degenerating as soon as they are removed from the tissue. So while people have tried to use them and still do use them, I think they're probably unaware of some of the problems. Lobule perfusion is the sine qua non of these sort of measurements. You have full structural and compositional representation. You have maternal and fetal circulations, and it's possible to use samples from pathological pregnancies, which is a great thing. Unfortunately, the lobule perfusion is a black box system. It's limited to the analysis in general of what's coming out of the fetal or maternal circulations. It can't be manipulated in any meaningful way. It's of limited duration, and it's limited to the 3rd trimester. So what this means, essentially, is that we're going from the very simple to the more complex, but the more complex we get, the less ability we have to look at that system and look at the components within that system. So what I want to talk to you about is some more recent technologies, trophoblast stem cells, trophoblast organoids, and placenta on a chip. Trophoblast stem cells are the basis of a lot of the things that are beginning to be done now. And they were derived in 2017 by OK and colleagues in Japan, and they took first trimester villus cytotrophoblast cells to generate a cytotrophoblast-like stem cell, which was a proliferative line with characteristics very similar to the primary cytotrophoblast. They can be differentiated into extravillus trophoblast cells and syncytial trophoblast cells using specific cytokines and pathway inhibitors. They have transcriptomes and methylomes, which are very similar to their primary equivalents. They could be stably manipulated using siRNA, CRISPR, and so on. And they could be stored frozen and used for multiple passages. So they're great in many ways. However, these were derived from first trimester placental tissue, which is not always available to people. However, trust people to come up with a solution. So now you can get trophoblast stem cells from term villus cytotrophoblast. Again, it's manipulation using a variety of cytokines and pathway inhibitors. You can get your induced trophoblast stem cells, which will go for multiple passages, but can also be differentiated into into extravillus trophoblast and into syncytial trophoblast. What this means is now we've got a system where the starting material is readily available at term. You get proliferative cultures. You get a high degree of similarity to in vivo primary cells. You get the possibility, if you want to, of making stem cells from pregnancy pathologies of one sort or another. The real problem here is that trophoblast stem cells are maintained within a specific culture system only by employing that specific range of cytokines and pathway inhibitors, or at least that's the problem at present. Nevertheless, these are much better models than the choriocarcinoma and transformed cell lines that have been used up until now. Placental organoids are a three-dimensional ex vivo culture model that can be used to study a variety of aspects of placental development. And as you can see here, what you're doing is plating cytotrophoblasts within a matrigel dome here, and you're growing up these structures. And you can see the structures here diagrammatically. And what you'll notice is, and what is repeated here, is unfortunately in the initial versions, you got the cytotrophoblast on the outside and the syncytial trophoblast on the inside, which is the opposite of what occurs in vivo. In vivo, you have the maternal blood, and the syncytium is in contact with the maternal blood, and the cytotrophoblasts are underneath. However, again, methods have been found to reverse this. So in this case, or what we're looking at here, above is the normal, or the original version, STB in which case the inside, the green, is a syncytium, and the outside, the red, is a cytotrophoblast. Down here, it's been reversed. The green is the syncytium on the outside, and the red is underneath. Similarly, you can see a reversal here. The syncytium on the inside, and then down in this one, the syncytium on the outside. So now we've managed to derive techniques for reversing this system. So now the syncytium is in contact with the external medium as it would be in vivo. So let's go on from organoids to perhaps the most interesting set here, and that's the placenta on a chip. So what you can see here, well, first, I suppose, is you can see the size of the chip. It's not exactly a huge object, but you can see the existence of two channels. And so what you have are two channels, up, one above, one below here, with a membrane in between. And that membrane can be seeded with a variety of cell types, so that you can get a membrane here with perhaps an extracellular matrix, basal lamina, or endothelial cells, syncytial cells. In other words, you can begin to get something that resembles the diagram I showed you earlier. And one of the advantages here, well, there are multiple advantages here. The initial placenta-on-a-chip models employed cell lines like BWO to mimic the syncytial barrier. But the more recent models now using various trophoblast stem cells, which can be differentiated into the multinuclear syncytial trophoblast. And as people have found, when you are doing that differentiation and you're doing it under flow conditions, interestingly, the barrier becomes a much better barrier than it does under static conditions. So what we have in this case is a model that can also incorporate other layers, extracellular matrix, endothelial cells. And so we've now got a model which has multiple cell layers, has maternal fetal flow, is stable, can be used for a useful duration. It's capable of manipulation. You can manipulate the cells before you put them in there. And again, we can use carriers with this. You can put in proteins or blood cells into the flows that you're having either side of that chip. And finally, here's a little bit on the placenta on a chip and biologics, because again, the placenta on a chip lends itself to looking at biologics. Now, this diagram was not derived for the purpose of a placenta on a chip illustration, but again, what you're looking at here is what you can see on the placenta on a chip. A syncytial layer, a barrier of some semi-permeable barrier in the middle here, and your endothelial cell layer. So we know that this can work, especially if you can add a tag to the IgG. So now you have a system where you can look at endocytosis, exocytosis, degradation, and endothelial interaction. So we now have placental barrier models. We now have placental barrier models which we can use, which are much more complex than those individual components, but they're in the components in these we control, we can manipulate, and we can look at and change. So the challenges now are to improve these models with more components of the villus structure. We can put in cytotrophoblasts, we can put in immune cells, and really, I suppose the biggest challenge is to develop conditions which ultimately do not rely on playing with specific cytokines and pathway inhibitors to control the nature of the culture. So the future, I think, is that we've got new models that more faithfully reproduce the complex features of the human placental barrier. Rather than a black box, however, which is incapable of being manipulated, the elements in these models are defined, controlled, and can be manipulated to test specific characteristics. And the combination of these methods with new methods of in vitro, ex vivo, and in vivo assessments will provide the means to determine the transfer and metabolism of drugs and biologics. And I'd like to thank you.
Thank you, Nick. Our next speaker, I believe, is online. Is that right? Do we have confirmation? Yes, we do.
That is right.
Next speaker is Rohan Lewis. Glad to hear your voice, Rohan. Rohan is a professor of placental integrative physiology at the Faculty of Medicine, University of Southampton. And I'm going to hand the podium over to you, Rohan.
Excellent, thank you. Can you see that fine?
Yes, we can.
Excellent. Okay, sorry, I've got Is that working? Sorry, I've just got here a slight problem with the. Yep, I'm sharing. Okay, sorry. Okay. Okay, so today I'm going to talk about in silico assessments for small molecules and biologics across the placenta. So the placenta determines fetal exposures. As we've heard a lot today, we have some things that are transported across the placenta from the mother to the fetus, nutrients and some drugs. Some things that are transported from the fetus back to the mother, which including wastes, toxins, and drugs. There's things that don't cross the placenta, and things that don't cross at all are mainly the very big molecules. Then there's some things that cross by diffusion, like gases, and some drugs. But the placenta controls what goes across the placenta. And what goes across the placenta is then what the fetus becomes exposed to. So understanding this process is really important. So I was asked to talk about in silico approaches to study placental transfer. And basically an in silico experiment is one which is done on a computer. So there's lots of options here. A few of them include algorithmic approaches, bioinformatics, computational modeling, which I'm going to talk about most today, and machine learning, which I'm going to talk about a little bit at the end. So what can you study in silico? Well, you can study people, you can study animal models, you can study in vitro models, including all of those that Nick's just been talking about so well. And you can do things like predicting novel drug-protein interactions and predicting genotype drug interactions. And you may wonder, you may wonder why we want to do modeling if you can get actual data from experiments. Well, I think modeling can test the biological understanding of the system. And if we make mistakes about our biological understanding of the system, this is going to affect the predictions we can make about fetal exposures and how accurate these are. It's also useful for understanding model systems like those Nick's just been talking about. And a lot of these, the transfer, some of it's going to be relevant to what happens in vivo, and some of it's going to be a property of the system. And modeling can help tease out what's what in that situation. You can also cheaply and quickly make predictions for multiple conditions. So doing experiments can be expensive. Modeling can be really cheap and quite fast. So you can change multiple conditions. And then the point of this is trying to understand where the system behaves in ways you wouldn't expect. And then you can go and do experiments focusing on those areas. It allows you to explore questions that are difficult to assess experimentally. For instance, different stages of gestation. We can make a model and we can adapt that model to reflect the transporter levels in mid-gestation. We can predict what might be happening here without having to do an experiment, which might not be feasible or ethical to do. And models can also help design the right experiment. So if you know where rate limiting steps are, you can design the experiment so you collect the data points at the right point and you get the most information. Okay, so computational modeling, which is my main area of expertise. So I think there'll be a lot of talk at this meeting about physiologically based pharmacokinetic modeling. And I don't quite do that. What I do is a much more focused approach, just looking at the placenta. So we treat the mother and the fetus in a very simplified way, but study placental function in much more detail than some of these models. Not all of the models, but some of the models. So here's an example of a compartmental model for transfer of cortisol. So that was how we conceived what was going on. We then tested that in the perfused placenta system. And our initial models didn't represent the perfused placenta very well. So we developed a model a bit further, changed some of the parameters, because some of the things we didn't think were important turned out to actually be quite important in determining how cortisol got across the placenta. So the model you can develop iteratively so that it reflects the biology. And then, of course, you have a model which is much better and could be, of course, be put back into a physiologically based pharmacokinetic model. So we've modeled the transfer of quite a few substances across the placenta. And the ones I'm showing here, the amino acids, fatty acids, cortisol, glibenclamide, metformin, in all of these cases, what we thought were the rate limiting steps for transfer as shown on the second line here, when we did the modeling, it turned out that something else was more important or equally important in determining what went across the placenta. So for amino acids, it turns out metabolism was also very important. For fatty acids, metabolism is the primary determinant of their transfer. For cortisol, turns out the rate of diffusion of cortisol across the placenta is actually equally important as 11-beta-HSD2, the enzyme which breaks it down. For glibenclamide, we thought it was membrane transport. Actually, the real determinant of transfer was the albumin concentrations in the maternal and fetal circulations. For metformin, we thought it was transported across the placenta. But really, in terms of going across the placenta, what's determining the transfer is diffusion. There is, of course, still transport in and out of the placenta, just not across the placenta. So these models help us understand what's actually the underlying biology, and that then gives us a much stronger basis onto which to make predictions about fetal exposures. And modeling is also really useful to understand model systems better. So here we've got what's essentially a transwell, but there's flow through the upper chamber and flow through the bottom chamber. What you can see here is that we can model the patterns of flow. So in the upper chamber, primarily going across the middle, and it's reasonably evenly distributed across the top. But in the lower chamber, the flow is primarily actually going around the bottom of the chamber, but only a little bit going underneath. And the way that you're getting flow in these models is going to affect the transfer you observe. And through using modeling, you can essentially account for what's due to the transfer and how flow is affecting that. And if you can take out the effect of flow, essentially, you can make data that's much more comparable to other systems. For instance, in vivo. And modeling can help design better experiments. So in this experiment here, I won't go into great details, but it's basically measuring metformin transfer across the perfused placenta. So in the first half of the experiment, we put the metformin into the maternal circulation. In the second half, we put it into the fetal circulation. And halfway through each block, we add additional metformin as a blocker. So the data points are the points, and the lines represent 3 different models we tried to fit to the data. And basically, as you can see, the models fit the data pretty well, except in this area here. But the way we designed this experiment, there were no data points in that region, so we couldn't observe that phenomenon. So because we hadn't modeled in advance, we had to go and do a completely new set of experiments where we put data points following the change in metformin concentration. And you can see you get the peak as the model predicted. And then these peaks are just other substrates of the transporter we think is mediating this to show that it is in fact that transporter. So by modeling it in advance, we could have designed a better experiment and saved ourselves a lot of time and money. So now I'm going to talk about modeling placental antibody transfer. So as we've been talking about the barrier, so you've got the maternal blood, the syncytial trophoblast, and the fetal capillaries. So here's the syncytial trophoblast, and antibodies go across by transcytosis, as several of the speakers have presented today. And then you also need to get transfer across the endothelium. So we developed a model framework to represent this, and this includes the binding affinities for IgGs to the receptors. We've got different receptors in the different barriers. As you see, compared to a pharmacokinetic model, we've got much simplified maternal and fetal circulations. But of course, this is designed to represent a placental diffusion, where you've just got a maternal and fetal reservoir. But you could, of course, put this model back into a pharmacokinetic model once we'd tested it in the perfusion system, and hopefully it would then be more reliable. Of course, we informed this model using single-cell RNA-seq data, which other people had performed. But you can see there's the FcRn, which everyone's been talking about. There's also other receptors, which are really highly expressed, particularly in the endothelium, which may be important in mediating the transfer. So other forms of in silico data can inform the development of the model. So looking at infliximab transfer, and we collaborating with a group here where we got the original data underlying their publication in 2020. So what you can see here is the maternal reservoir concentrations. It's essentially the maternal circulation and what's being transferred to the fetus. Now, as a biologist, when I look at this, I think, we've just fitted a line. It's not very, very informative. But actually, those lines aren't just fitted. They're not correlations. They're based on all the mechanisms of transfer I showed you in that model. And based on that fitting, we can then do sensitivity analysis. So we look at all these different factors, and we can try and understand where they become rate limiting. So for instance, if we look at the blue line, FcRn, the model predicts that if FcRn concentrations dropped, transfer would drop, but that if the FcRn concentrations increase further, there's actually going to be no benefit to transfer of IG or infliximab across the placenta. And then you can do more complex sensitivity analysis where you look at multiple factors at a time. So in this case, we've got endothelium FcRn and syncytiotrophoblastic FcRn. And what the model, at least at this early stage, is saying is that the syncytiotrophoblastic FcRn is really the rate limiting factor for transfer rather than the endothelial FcRn. This model compares the different transporters that the RNA-seq showed, not transporters, sorry, receptors, which the RNA-seq showed were expressed in the endothelium, or the two highest level expression. And it suggests that Fc gamma R2 beta is probably more important than FcRn in the endothelium. Although I should say at this point, it's quite early in our modeling process, so we're still testing these. Okay, so what do we gain from modeling? So we check our understanding of the biology of transfer. We can understand the experimental system better, whether this is a human, an animal, a flow cell, or an organoid. And once we've tested the model, we can predict how clinical conditions might affect transfer. So briefly, I'd like to talk about machine learning. So machine learning can be very powerful. So what we've done here is we've trained a machine learning algorithm to understand the relationships between structures in the placenta. And then we've given it the shape we show in blue, which is a real fibroblast for a section that it wasn't trained on. And then asked it to predict what the tissue around that looks like. And if I was there in person, I'd ask you to guess which was the real one. The real one is in fact B. But I think if you look at A, it's fairly convincing. I think there's a few things you think maybe aren't quite right, but to the untrained eye, it's a TEM image of the placenta. So machine learning is very powerful, but it does have a lot of limitations. So I don't think it's useful for predicting placental transfer at the moment. I think the real problem is that you need an awful lot of data to make machine learning algorithms transferable between sites and between patient groups. And also, because machine learning algorithms are a bit of a black box, you can't predict whether they're predicting something meaningful. If they're looking at a scan, for instance, and there's a date on the scan, and all the sickest patients come in on a Tuesday, then the machine learning algorithm will pick up based on the date they come in, who's more sick. So, and that's of course not going to be transferable to any other hospital or any other patient group. So for now, I don't think they're going to predict transfer across the placenta, but where I think they can be really useful is predicting novel drug receptor interactions or drug transporter interactions. And that they, of course, can be put into our models to help us understand the other systems we use to study the placenta better. But if you look at the literature, there's certainly people publishing on this. So I think, watch this space and don't underestimate it. Okay, so in conclusion, I think in silico is good for understanding the biology better, understanding data from experimental systems better, and also being able to relate the data from one experimental system to another experimental system better. It's good for designing better experiments, but I don't think it can replace actual data. And I don't think it does replace actual data. I think it complements the real experiments that we're able to do. But once we've developed good, for instance, computational modeling, which is the approach I use, we can then simulate maternal and placental states to predict where atypical transfer may occur. And I think it's where we've being able to predict what might be unexpected in terms of, drug interactions or different physiological states in the mother and how that affects transfer across the placenta that we really need to understand. Thank you very much. I'd just like to acknowledge my collaborators, including Bram Sengers, who actually does most of my modeling, and our funders. Thank you very much.
Thank you, Rohan. I hope you can stick around for the panel discussion for later. Our next speaker is John DeSoso, and John is a principal scientist at Exponent, a scientific and engineering consulting company, and I believe he's also presenting online. John, can you hear us?
Yes, can you hear me?
Yes, we can hear you.
Okay.
Are you able to share your slides at your end?
Sure, be glad to. Okay.
Excellent. We can see your first slide.
Okay, good. Should I start? Can you hear me?
Yes, go ahead and start, John.
Okay, fine. Well, that's great. Well, thanks for a chance to come and talk to you. I'm supposed to talk a little bit about in vivo animal assessments. And I have to say that the first two talks you heard in this session are really excellent and are showing you where we are going and where the future of placental research will be in 10 or 20 years. But we're still on the march to get there. As was mentioned earlier, I am a principal scientist at Exponent, and for some reason my slides aren't moving. Why are they not moving? Let's see. Oh, here we go. So my disclosure is that Exponent is a science, engineering, and technology consulting firm, but I have no financial interests or other interests that would form a conflict. There was no external funding for the preparation of this presentation. So what I want to talk about briefly is, what is a placenta? Just to remind ourselves what we're talking about. Talk a little bit about something we haven't heard about yet so far, which is the inverted yolk sac placenta, which will have a role to play in what we're going to discuss a little bit later. And then talk about the placental transport of immunoglobulins in rodents and humans, in which the geography is different. And then talk about a little bit about which safety tests can inform us about placental function and research. And lastly, determine whether or not there is a best animal placental model for humans. So a placenta is any apposition of embryonal to parental tissues for the purpose of physiological exchange. It was defined as this by Mossman back in 1937 or 1939, and it is a really important aspect because it tells us, first of all, what a unique organ it is. It's composed of tissues from two different individuals. There's no other organ in the body that does that. It's a temporary organ. It only lasts in humans for about 8 months. In rodents, it's for a matter of 10 days or so, but it's obviously of critical importance. And as has been pointed out by numerous previous speakers, the morphology and physiology evolved dramatically with the age of the placenta. So the maternal fetal transfer is the major function of placentae, but that doesn't mean that all placentae act or look the same. The efficiency of this transfer is affected by a number of things, including the circumferential aspects of the placenta base, modifications of the interface to increase the surface area. There's a lot of modifications in the diagram to the right over here. You can see the pig, it just has a number of folds to increase the surface area. If you look at the human, which is down in the lower right, it's the villous tree. And in rodents, it's even more complicated with a labyrinth, which has got even more surface area. So there's a lot of ways to increase the amount of surface area, and the amount of surface area will, of course, affect the rate of absorption across the placenta. Is there another ways that you can increase the rate or change the rate of crossing? And that is to decrease the number of layers and the thickness of those layers between the maternal and fetal blood. And as been shown in numerous of these other talks, we've got a number of layers in between them. They're shown here in this little diagram. On the left side, there are 6 layers between the maternal blood and the fetal blood. In all cases, the fetal tissues are on the top and the maternal tissues are on the bottom. So on the far left, that's what the pig has. There's all 6 layers. As you move towards the right, more and more layers of the maternal tissues disappear until finally you wind up with maternal blood bathing the trophoblast layers, which is what happens in humans and in rodents. So that's the chorioallantoic placenta. So that's another mechanism whereby you can change the transfer of materials. Another thing that has been pointed out before are the ontogeny of various receptor and carrier molecules in the placenta, which has an ontogeny similar to what you see with enzymes, and they increase in their size and number throughout gestation. So having said that, we want to again remind ourselves that regardless of these morphologic differences, of which there are many, the role of all placentae is this maternal-fetal exchange. And the differences in morphology that we're talking about are important, though, and we have to think about them when we're taking into consideration how we're going to extrapolate to humans, and that will be most apparent if we just talk about what the timing of appearance of things are in the embryo. And so this is a diagram that I use, I call it a roadmap of embryonic development. And on the left-hand side, what you can see is where fertilization takes place. And then as you move to the right, the embryo begins to differentiate. And eventually, by the time you get to the far right, it's ready to be born. And each of these territories is demarcated by the letters A, B, C, D, and E, in which I have divided up from my own thinking what's going on. We've got pre-implantation, followed by gastrulation, followed by formation of the germ cells, major organogenesis, and eventually development in the fetal period. The sequence of events is the same for all mammals, but the difference in timing is quite different. As we'll see in the next slide, you go from fertilization to birth in hamsters in about 16 days. In humans, it takes around 266 days. In elephants, it takes around 440 days. So there's a big span in terms of these differences. So when does the placenta actually show up? Well, it turns out the placenta shows up sometime during organogenesis. And you say to yourself, well, isn't that interesting? There's a lot of things that have happened prior to that. How does the embryo of a rodent work with all that, especially if we take a look at what has to happen in the rodent, and that gets us to the inverted yolk sac placenta. So this is a table which basically shows what the timing would be for each of those intervals of A, B, C, D, and E. And the hamster's at the top, and if you look at the far right, you can see that its parturition gestation length is 16 days. I put humans at the bottom, where it's 266 days. And as you look at that, you say to yourself, okay, when does that choriolanthog placenta develop? And that's written in red. And as you can see, for the rodent, it shows up around 11 and a half days. There's only about 6 days left before organogenesis ends, and only about 10 days until the fetus is born. Whereas in humans, shows up about the end of the first month. You've got another month to go for organogenesis, and a total of about 8 months with the placenta in place before parturition. So it's a very big difference. So the question is, what happens in the rat prior to that 11 and a half days? They've got a lot that has to get done. Things are going very rapidly. How does it get nutrients there? It just can't be sitting as a ball of cells that's getting things by diffusion. And so it has another placenta called the yolk sac placenta. And the yolk sac placenta is important and will become important when we talk about the different kinds of testing a little bit later. And briefly, what it is formed from a derivative of what would be the gut tube. And so this is a diagram that shows a rodent conceptus. You can see the yolk sac is depicted in yellow. It is connected to what would be the gut tube of the embryo. The embryo is the magenta area. Notice the little green thing, the allantois. That allantois will eventually form the stem to form the chorioallantoic placenta. So it's how it gets its name. But we're talking about what happens now with the yolk sac. And in rodents, the yolk sac expands dramatically as it's formed, so that the yolk sac becomes in apposition to the maternal tissues. And so it is a fetal to maternal apposition. So it's a placenta. It lines up against that maternal tissue. And then what happens is, now think about it. The yolk sac, just like the yolk in an egg from a chicken, the absorptive epithelium is going to face where the yolk is. So the absorptive epithelium is on the inside portion of that, let's see. This area here is where the absorptive epithelium is. And so that's the absorptive epithelium is here. You say, well, how are things going to get from mother to baby? Well, what happens is the yolk sac in the rodent eventually decompresses. so that it collapses. And as it collapses, now we have both layers, the layer on the posterior wall here and the anterior wall are facing the maternal tissues, and the one on the anterior wall dissolves and wind up then with the endodermal lining of the yolk sac now faces directly against the maternal tissue and can absorb the materials. So this is a rather large organ in the rodent. This collapsed balloon isn't just an umbrella. Right here, it looks like it's just a little T-shaped sort of a thing, but actually it becomes quite large. I'll show you a slide after this one that shows you how big it is. To remind you what's going on, it is the transcytosis model that's already been described for the for immunoglobulins. Basically, there's going to be an imbibing of pinocytotic imbibing of materials. They get attacked by lysosomes, shown here. The lysosomes degrade it, and the digestin either diffuses directly across the membranes, or you can get the vesicle can bind to the wall of the basal portion of the endothelium and release materials that way. So that's very similar to what we saw for the IgG and the other slides. This is a photograph that shows you what that yolk sac placenta looks like in a rat embryo. This is a day 13 rat embryo. You can obviously see the head and the rest of the body. And this translucent tissue here, where we've taken away the uterine wall, shows you where the yolk sac is. You can see the blood vessels in it. So this is a large structure, and it remains working. It's working in the rat embryo up until at least day 19 of gestation, and possibly all the way through until the end of gestation. So it's an active part of the nutrition of the embryo, even though there is a second placenta, the chorioallantoic placenta, which is also going to become operating about day 12 of gestation forward. So one thing is, if people say, well, what about what's going on in the human yolk sac? Well, the human yolk sac is important, and it's a very large structure in the young embryo, but it never surrounds that human embryo, and it does not invert. And so this next three diagrams will show you a human conceptus. Here's the yolk sac, here's the embryo, and then after a few days, you can see that the embryo has begun to develop a little further. The yolk sac now looks like a balloon, but you'll notice it's not adherent or does not touch the uterine wall. And by the time you get to about the end of the 6th week or so, it becomes just a very small vestigial organ, and it is no longer even much of a structure to talk about. Now, is there pinocytosis going on in the yolk sac in the human? Yes, there is. Is it enough to support the human embryo? No, it's not. And is it going to be involved in transfer of materials from mother to baby? No, it's not, unless the material could perhaps first diffuse into the amniotic fluid, and then secondly, diffuse from the amniotic fluid into the yolk sac. So it's highly unlikely that would be a very important route of administration or route of transfer in a human. However, we have to keep that pinocytosis in mind as it is important. So placental transport of immunoglobulins in rodents and humans, the mechanism has been described earlier, and it is going to be the same, but the location is very different. So as you already know, we've got the antigen binding portion of an immunoglobulin plus the FC stem, and the FC stem is what is going to be picked up by the FcRn receptors. So in a human or non-human primate, here's the structure of the embryo. Here's the yolk sac. Here's the developing chorioallantoic placenta. And if we look at this in a little bit of a blow up, you can see that you got the villus tree. The villus tree is bathed in maternal blood. If you look at it a little bit more closely, you can see that the series of steps we talked about, where we have the material picked up from maternal blood. The pH is around 7.3 or so. Eventually, the endosome gets attacked by lysosomes. The lysosomes change the pH down to about between 6, maybe around 6.3 or so. But as that happens, the FcRn receptor it manages to grasp a hold of the IgG and protect it. And honestly, it's interesting because the IgM and IgA are picked up the same way, but they get destroyed because they don't have that receptor. And then when the endosome eventually binds to the opposite side of the cell, and it opens up, the pH rises to around 7.3. The receptor releases the immunoglobulin, and it crosses into the fetal compartment. Now, rodents do not have FcRn receptors in the sissio-trophoblast. That's very important, but they do have it. But in their case, it's in this very important yolk sac placenta. So the same structure, if you look at this, you have a blow up of this, you see the same structures, the same series of events is going on here. In this case, the the maternal tissues are at the bottom. You can see the materials are being picked up here into these yellow cells. Those are the endodermal cells of the yolk sac, and you have the same steps of pinocytosis, binding to lysosomes, acidifying the endosome, and then eventually the release on the other side of the placenta. So, now, importantly, what that tells us is, from an experimental standpoint, is that we have the process is the same in rodents and in humans, but the location is different. So it's important to keep that in mind. Now, one of the questions that have been looked at for quite some time by a number of pharmaceutical companies. There's been a lot of data generated by Chris Bowman's lab at Pfizer and by Graham Moffat's at Amgen, Susan Westmoreland at AbbVie, and folks at GSK. And we've been able to show that there is IgG transport throughout gestation. And I'm showing it here in terms of this graded green bar. And I compared, in this case, the mouse, rat, rabbit, cynomolgus monkey, and humans. And you'll see that the dotted bars on each of these dotted boxes represent the periods of organogenesis. And a few years ago, we knew quite well that during organogenesis, there was transfer of immunoglobulins in the mouse, rat, and rabbit. It was shown more recently that it was present in the cynomolgus monkeys, and it was questionable about the humans. Well, recent data that's being generated by AbbVie shows that it's definitely occurring here in the humans. This bar, this diagram was made up about 6 years ago, and I would say now that the green ought to be down around here in humans and probably about the same place in rodents or in monkeys. And so the idea is that IgG does cross, it crosses well, and you'll notice that it crosses greater at the further into gestation you get, and that's because the number of FcRn receptors increase, as does, in this case, the surface area of the placenta. Now recall, in the mouse, rat, and rabbit, all of that is going on in the yolk sac placenta, whereas in the monkey and the human, it's going on across the syncytial-trophoblast. So if that's all true, then what do all these animal tests we do, what do they tell us about what's going on with the placenta? And there's really, of the ones we do, there's really only 3 tests that actually involve the placenta. There's the embryo-fetal development test, which obviously we start treating after the animals are mated. And in that case, the difficulty is that while we might be able to find out about some things that would cross, it could cause malformations or death, we really don't have any. The test looks at fetuses just before they would normally be born, so we don't know anything about behavior or postnatal development. There's a multi-generation reproductive toxicity tests. They're rather complicated, and the data that we get from those is not really pointed at the placenta so much as it is at other aspects of development. And rather than spend much time talking about those, the more pointed or the more practical type of test that would tell us about that is the pre and postnatal development test, which is what we'll talk about in the next slide. And so in this case, this is a diagram that shows what happens in those tests. And in those tests, a female rat is treated beginning on the time of her implantation, around gestational day 6, and she continues to be treated with whatever the test compound is until the end of weaning, which in this case is PND20. And that mode of exposure means that the fetus, which is shown here in the stripe bar, gets an indirect exposure to whatever she was exposed to when she was carrying the babies. And that would be where our FcRn receptor would become important. And you say to yourself, well, that's good, but what's that going to tell us about what's going on here? One of the difficulties with studying, for instance, the brain and development of the brain is that the brain in rodents develops quite dramatically during the first 10 to 12 days postnatal. And the interesting thing about this is that in rats, and also in mice, the absorption in the small intestine is very different from what it is normally in an adult or in a human or a monkey. And the difference is that during those first 10 days, the villi, developing villi in the intestine of the rat, transfer materials strictly, completely only by pinocytosis. So it's almost like the process that we saw in the yolk sac placenta. And what's interesting about it is that in the rat, the cells that do the pinocytotic transfer have FcRn receptors, and IgG is transferred in the milk in rats. And so they would continue to get this exposure in an indirect manner during that period of time. So it doesn't just stop right here. On the converse side, if you're talking about a rabbit, rabbits do not have that FcRn receptor in their immature intestine, and so for them, the gestation end of gestation would be the end of the period of time when you would get that exposure. Now, what's interesting about this test and what it tells us, what it allows us to look at in terms of the IgG, is that now we have animals that are grown up to weaning. They're kept alive for a number of weeks to do testing on it, and some of them are allowed to have their own offspring. And so we can see whether or not it affects mating, whether it affects neuroanatomical or behavioral changes, whether or not there's differences in the F2 offspring. And so this is a very complex and helpful kind of a test, and it is especially true if we after we look at these mid to late gestational exposures, that we can reveal the adverse effects that would not be seen in the EFD study. Also, we would be able to look at what, if there's TK involved, we can figure out what kinds of real life exposures animals would have and use those exposure levels in the in vitro and in vitro models, so that the concentrations of materials in the cell systems are not super pharmacological, and so we're not confounding some kind of an effect that was just because we've got an exaggerated pharmacologic effect rather than something that would be due to the compound that would be seen in the real life. So after talking about all this stuff and the differences and some of the similarities, is there really a best animal placental model? And so I prepared this a couple of comments first. First of all, the in vivo models incorporate ADME and vascular alterations that aren't yet possible in vitro, but as has been pointed out by Nick, one of the problems with that is it is sort of a black box, because while it incorporates all those things, you really can't adjust those on your own. You have to just look and see what you see. Although this, I was told not to talk too much about non-human primates, I do want to make a couple of comments, in that among the apes, probably the best placenta would be the chimpanzee, because it's nearly identical to humans. The problem, of course, is that it's ethically untenable. We're not going to use chimps. They're intelligent, and there's very few of them, and so on and so forth. But among old world monkeys, the most commonly looked at, or most deeply looked at, is the rhesus monkey. And it's got problems too. And the problems are that they're discoid, they have a bidiscoid placenta. So they've got two placentae, each that are on either side of the fetus, but they have superficial implantation. So the invasiveness is not as good as it is in humans. And because of the superficial implantation, they're very prone to abortion. And that can be from any kind of stress. It can be from stress in the animal room, and it can be from the stress of being treated with an exogenous agent. So they're not really a terribly good model for this, even though they've got a placenta that looks somewhat like ours. With respect to other animals, this is a chart that's got 6 species on it. The human is on the left. Then we look at mouse, rat, rabbit, guinea pig, and mini pig. And I've got a number of characteristics of the animal species, and this including the size of the females, the gestation length, the number of litters, and so on and so forth, the neonatal weights. And you can see they're none are really great in terms of being comparing to humans. Then we get to the placenta. So all of them are discoid placentas except for the pig, which is diffuse. With regard to a placental invasiveness, most of them are hemochorial, but humans are hemomonochorial. The only hemomonochorial placenta in here is the guinea pig. The rat and the mouse are hemotrichorial. and the rabbit is dichorial, so they're somewhat different. And of course, we talked about the pig before being an outlier in the sense it's epithelial chorial as well. In terms of the surface area modifications, the only one with villi is humans. The mouse, rat, rabbit, and guinea pig all have a labyrinthine type of arrangement. And with the mini pig, as we saw in the earlier thing, they just have folds. So those aren't the same. Now we get to the actual boundary layer. The boundary layer in the placenta for all of the animals except for the pig is in trophoblast, so that sounds pretty good. But if we're talking about for the site of the FcRn, the only one that has the FcRn at the trophoblast is the human. For the mouse, rat, rabbit, and guinea pig, is the visceral endoderm of the yolk sac. So again, it's in a different location. And with the mini pig, we're back to the cytotrophoblast as well. So when you take all of this into consideration, you wind up with I think the following things. We've looked at the placentae. We found they've got different kinds of anatomies, but they have a common function. We've shown that the rodent inverted yolk sac placenta enables the maternal fetal exchange in early gestation. It's also the site where the FcRn is located. That in the safety tests, probably the PPND is the best to be able to investigate any health effects from immunoglobulins. That is to say, health effects looking downstream other than just abortion or fetal death. And if you got TK involved in those studies, it gives you this, it's going to inform the dose setting for the in vitro tests. And I think that when we get finished, and I was thinking about this again when listening to everybody else's talk, and I still think this is a good thing. The placental research, I think, is best considered as a tapestry. We're getting all these diverse tests and various systems, and they all have something to add. They all have their problems too, but you're getting a little bit from all these things. And as this moves forward, I think our understanding of this very complex organ and its role in maintaining fetal development and health is going to become more and more prominent. And I would just say again that there's just no single best animal model at this stage. And that's it.
Thank you, John. We also hope that you will stick around for the panel discussion that's coming up shortly after our next presenter, who is David McMillan. He's a toxicologist in the Division of Pharmacology and Toxicology for Infectious Diseases here at the FDA, and is assigned to the Division of Antivirals. So the podium is yours.
Hello. Thank you for the introduction. Now for something completely different. How do I... Mm-hmm. That work? There, it's working now. Okay. Well, thank you for having me. I appreciate the invitation to speak to you today. So I'm here to give you an overview of the non-clinical guidances pertinent to developmental immunotoxicity. And I'm also going to be going over a case study that is related to this at the end of the talk. So from a non-clinical perspective, there are a handful of guidances that are that apply to the assessment of immunotoxicity. There are 8 of them within CEDAR. Some of these are dedicated immunotox guidances, and some of these are guidances that pertain to other areas of toxicity assessment and that have some sort of immune component to them. Regarding in utero exposure and developmental toxicity resulting from in utero exposure and placental transfer, there are really only 2 that I can really reference that are applicable here, and that's S5R3 and a guidance that we had published in its final form last year, non-clinical evaluation of the immunotoxic potential of pharmaceuticals. So for S5R3, this is the primary guidance for the assessment of developmental and reproductive toxicity. So this is the guidance that is the primary one for the design and implementation of these non-clinical DART studies, as we call them, or developmental and reproductive toxicity studies. This applies to small molecules, biologics, including vaccines and novel excipients, so it's a joint CEDAR-CBER guidance. And as it pertains to developmental toxicity, all we can really say is that specific immune endpoints are not typically included in these studies, and if you have a concern, then it would be prudent possibly to include these endpoints when you're designing these studies. The guidance that really says a lot more about this is the one we had published last year. So this is a CEDAR guidance that's intended to supplement S8 and S6 for small molecules and biologic products, respectively, and to replace the old 2002 CEDAR Immunotoxic guidance, which is now out of service. This applies to small molecules, oligonucleotides, and CEDAR biologic products. It provides guidance on a number of areas relevant to immunotoxicity assessment, although in terms of, as it pertains here, there is some guidance on implantation and early pregnancy. I know it's a little outside of scope of what we're talking about. But here we're talking about standard, basically we're saying that the standard non-clinical fertility and embryo-fetal development studies are generally adequate to assess the risk in terms of effects that a drug may have on implantation and gestation. But if we think that there's a mechanism for a drug that may be incompatible with fertility or pregnancy, then a weight of evidence assessment may be more appropriate. Regardless, fertility studies are generally not warranted for anti-cancer drugs, according to ICH S9. Regarding developmental immunotoxicity specifically, this evaluation relies on more of a weight of evidence assessment of multiple factors. If we think that a drug may adversely affect immune development and the existing data do not adequately characterize the risk, then you might want to further evaluate this risk by potentially conducting a non-clinical study. This study could evaluate immune endpoints in the offspring of treated females via a PPND or ePPND study. This is pre-postnatal development or enhanced pre-postnatal development when it pertains to non-human primates. If that study does not adequately characterize the risk, then you might consider doing a juvenile animal study where the juvenile animals are directly treated with the product. And I realize this is a little outside of scope for this session. Regardless, again, these are typically not warranted for anti-cancer pharmaceuticals. And it's not advancing again. Not really sure what to... There we go. Thank you. So should a study be warranted to assess this risk, then the species and the endpoints that you're evaluating need to be appropriate and scientifically justified. So this could include enumeration of specific cell populations via immunophenotyping, measures of immune function, for example, through a T-cell dependent antibody response assay, which is a general measure of immune function in animals, and assessments of anatomical integrity. So if there are any physical differences in structure. As there are species differences, we have to consider that extrapolation across species, and especially ages at this point, is very difficult. It really depends on what organ you're looking at and what endpoints you're looking at. And I was actually just talking to someone about it this morning, about something completely separate from this. So regardless, you really have to keep this in mind in terms of when you're designing the study, what you're looking at and what animals you're looking at, what timing of age that you're looking at as it pertains to the population that you're interested in. Regardless, you don't want to be using animals unnecessarily, so you want to make sure that you design these studies with the input of the reviewing division. So now I'm going to be switching gears and talking about a case study. It's probably what you're more interested in rather than all of the guidances. So I'm here talking about iblizumab, UIYK, or Trogarzo as it's marketed. This is a humanized IgG4 kappa mab against CD4 receptor domain 2. CD4 is the immune receptor on T cells and helper T cells that's responsible for binding to HIV, GP120. It has 4 domains, 1, 2, 3, and 4. Domain 1 is the primary domain that binds to HIV and also is responsible for all of the T cells, all of the CD4's normal immune functions. So iblizumab, I'm going to show you, will block HIV infection without affecting a normal host immune function. This is a CD4-directed post-attachment HIV-1 inhibitor. It's indicated for the treatment of HIV-1 infection in heavily treatment-experienced adults with multi-drug-resistant HIV-1 infection failing their current antiviral regimen. So this is a mouthful, but basically the nickname for this is HIV salvage. So this is a small patient population where previous regimens have failed, possibly due to patient non-adherence or to other factors that may have precluded the ability of those multiple different treatment options from being effective. Dosage is 2,000 milligrams IV, loading dose with 800 milligrams once every 2 weeks thereafter. Licensure was issued in March 2018, and as it pertains to non-clinical assessment for this, it only recognizes human and monkey CD4. So monkeys, by default, were the only pharmacologically relevant species. Regarding the mechanism of action, as I mentioned, CD4 has 4 domains. Under normal circumstances, HIV will bind to CD4, and then there are a series of steps that allow for membrane fusion, which will then allow HIV to release its genetic components into the host cell. But in the case of ibolizumab, it binds to domain 2, so it does not block host immune function, and it also does not block HIV binding to CD4, but it will prevent the downstream steps which will allow them for the membrane fusion. So that's why we call it a post-attachment inhibitor. So from a non-clinical standpoint, so we know that this is specific for human and monkey CD4. There was no off-target binding in any of the non-clinical assessments. We do know it is specific. There were no adverse drug-related findings up to the highest doses tested in the GLP toxicity studies in adult animals. Up to the highest doses tested, there were no changes in CD4 positive T cell levels, and also no changes in other immune endpoints, as was assessed by TDAR assay. So the non-clinical package was considered sufficient to support licensure back in 2018. There was extensive discussion regarding the design of an EPP&D study for this, which was ongoing at the time of licensure, and a PMR was issued to supply the final report. So the design of this, as I mentioned, was subject to quite a bit of negotiation, and the primary concern here was immunogenicity. So you'll notice that there's only one dose level that was selected for this study, and the reason for that was the immunogenicity. If you go any higher, you won't get any exposure because it's all cleared. And any lower than that, there were also concerns that it would be too low and it would be non-informative. So the animals here are cynomolgus monkeys, pregnant females that were treated once weekly, roughly throughout pregnancy, starting from around gestation day 21, all the way through gestation day 160, around parturition. Infant animals were not directly treated, so these were only exposed via in utero. And all of the infants and the mothers were monitored out to postnatal day 180, so 6 months post-birth. There were no findings of any kind of any concern in the adult animals. However, we did see some findings in the offspring that we did not expect to see that were not seen in the adult animals. So here we see that there were decreases, drug-related decreases in CD4 positive T cell counts and increases in CD8 positive T cell counts on postnatal days 14 and 28, so 2 and 4 weeks post-birth. And also there was a decrease in B cells around 2 weeks post-birth. This correlated to ibolizumab serum concentrations on the same days. So if you look at 2 weeks and 4 weeks, ibolizumab is still present. This is when these levels are decreased. By month 3, ibolizumab is no longer detected, or at least at trace amounts. These cell populations by this point had gone back to normal. There was also an assessment of immune function via a TDAR assay, and this was conducted at postnatal day 138 and 180, so 4 and 6 months post-birth. By this point, all of the cell levels had gone back to normal, so we didn't see any changes in the TDAR assay, and that's probably because all these numbers had gone back to normal. You may wonder why we didn't do this earlier on, and typically TDAR assays in infants like this and studies like this are considered not feasible for time points prior to this when they're younger. So regardless, if that was possible, it's conceivable that we would have seen changes in the TDAR assay. All of these findings are one thing, but the other thing that really amped up the concern for us was that one of the treated infants had died at postnatal day 24 of a systemic viral infection with secondary superficial bacterial infection acquired during the postnatal period. So you have to consider that wild-caught monkeys often will be carrying things within their systems that are suppressed by the normal host immune system. When you put them in a sterile environment, it should not be a concern, but if they are undergoing some sort of immunosuppression, then whatever those infections are that are normally suppressed might be reemerging in these animals. So this is not normal. You see this is not something that we would typically see, and if we do see something like this, it's usually a sign of immunosuppression. The animals in this particular animal had decreases in all lymphocyte subsets, CD4 and CD8 positive T cells and B cells, all up to 90-ish% around postnatal day 14. Also decreased cellularity in the lymphoid organs, spleen, thymus, and mandibular lymph node, and body weight was also decreased in the last 10 days of life from postnatal day 14 to 24. So there was not an OAL in infants. So the concern here then became, what do we do with this data? So this is where it came down to labeling and a lot of discussion. So this risk was communicated in the label first in section 8.1 in the risk summary. I have a description of the study here. I'm not going to read through it for you. It's more or less describing what we see. And then similarly in the animal data section, all of this is detailed in the label. And you can find this in the label if you want to look it up yourselves. The box that I have here in red is just indicating that we actually, because we don't have any exposure multiple data in human infants, we can't really calculate an exposure multiple relative to humans. So it's just not possible. So it's not, we can't really tell you like what fold increase relative to humans is really safe. So that's really what the risk here. But regardless, even if it is multiple times higher, there's still a risk of immunosuppression, and that's why this is all communicated in the label. And upon discussing this with our clinical colleagues, this was also considered worthy of putting in Section 5 and is a warning and precaution. So there's a Section 5.3 in this label basically saying that it may, based on the animal data, this may cause reversible immunosuppression in infants born to mothers exposed to Trogarzo during pregnancy. So there's a chance that it may cause immunosuppression within the first couple months of life, therefore plan accordingly. And there's also a section 8.1 where it details this as well. So this is basically a case where we didn't see any findings in the adult animals of any concern like this, but when we looked at the infant animals in the EPPND study, we did see a finding related to in utero exposure, and this could result in possible being immunocompromised within the first 3 months of life. It's possible this could interfere with lymphocyte development during late gestation or in the early neonatal period. It's really not entirely clear. This is the best data we have at the moment. We did not ask for any follow-up non-clinical safety studies. There were immunogenicity concerns. We thought it would not be possible basically to identify a dose where there were no effects. But all of the concerns were communicated in the label. So that's everything I have. Thank you.
Microphone on here. So now we'll move into panel discussion. And I hope both Rohan and John can hear me and will be able to join the panel discussion. I'd like to also introduce Dinesh Stensulov. I hope I didn't mangle your name there. Dinesh is the head of reproductive toxicology at GSK, and so he's going to be joining us for the panel discussion. What I'd like to do really here is to start off with a couple of questions, and then I'd like to have the panelists respond, and then we'll open it up for discussion for questions from the audience, including those online. And I'm going to get some help from Tamara to read out any questions that show up online. The presentations that we had today were really divided into two, really in vitro or in silico systems. So let me start there first and pose a question to the panelists who were addressing the in vitro and in silico assessments in terms of exposure, because most of it was devoted to fetal exposure. Those presentations were. So let me ask the question about how these exposures can inform potential fetal safety issues. you know. And if you could speak about that, and this really question is posed to both Nick and Rohan, and can that information be useful in terms of understanding potential concerns in terms of fetal safety?
Yes.
Put you on the spot there, huh?
Yeah, right. Yeah, we're not normally asked about fetal exposure. We're normally asked, simply how do you determine what gets across? And I think for us, for me anyway, the important questions are becoming, how do we transition from very simple models to complex models? Because the simple models, easy to see, but does it reflect what's actually happening in vivo? Certainly the lobule perfusion can tell us that, but it can't tell us how it happens. So building these new models enables us to look at the multiple steps that eventually can enable fetal exposure. And I think that's the pattern I see building, the ability to be able to generate a complex model, but a complex model where you can, for example, remove a component, a gene, knock out a gene, or at least temporarily bring it down or something like that. And I think the other development that is useful in that, and that it's something that's quite important, I think, and has not been possible prior to this, is to do the same thing, but to do it for pregnancies where there's a pathology of some sort. To do it under conditions, where it's an obese woman or it's a woman with preeclampsia or something like that, and to be able to say, okay, well, maybe it doesn't happen under normal conditions, but now we're able to mimic some of the conditions that might happen in these pathologies. And these pathologies, if you add them all up, these pathologies add up to a lot of pregnant women. So I think, as we build towards that, we're able to actually look much more towards a realistic assessment of fetal exposure.
So what I'm hearing is that, these are models that can feed into perhaps some of the models that Rohan is talking about in terms of perhaps predicting down the road fetal exposure.
Oh, certainly. I think maybe, ask Rohan, but I think, I think the placenta on a chip, for example, are very, very much so. You've got a controlled model which you can then examine and measure and model it in silico.
So before I turn this over to Rohan, maybe just a follow-up discussion point here in terms of what of the models that you were talking about are going to be suitable in terms of not just the term placenta, but also early gestational age, processes that could feed into models that Rohan can speak about as well.
Yeah, I think that there is a problem, and that is getting cells from early placentas. Yes, you can get them from terminations. You can, and it should be possible, although I haven't seen it being done, but in theory, you should be able to get, for example, tissue from chorionic villus sampling that's usually done late first, early second trimester, for example. So there are possibilities for obtaining cells that you wouldn't otherwise not be able to get, or you'd be able to get them, but it would be very small amount, short-term use. Now you can start to use trophoblast stem cell techniques to develop permanent lines or permanent systems that you can incorporate into these models.
Thank you. And the other panelists, please feel free to chime in here. But I also want to bring in Rohan first to comment on the same questions that I posed to Nick. Rohan, can you hear me?
Yes, I can hear you. So I think Nick makes a lot of good points, and I agree with what he's saying. I think you can get things from all these different models that help build up a better picture of how placental transfer occurs, and also how it might occur differently in different people. And I think that's really important, because I don't think, what's going to cause a normal exposure in one person won't necessarily have the same level of exposure in another person. And ultimately, we want to be able to understand this at an individual level. And I think getting a more complex understanding of what goes on, and then using the modeling to try and, because there's so many individual mechanisms we could study, using the modeling to try and understand what's likely to be rate limiting. And that allows us to focus future research on the things that are really likely to determine fetal exposures.
And do you have some thoughts about how to translate that fetal exposure to inform potential fetal safety considerations?
So I think when we've got better models, I think we can identify situations in which the transfer might be different than what we'd expect normally. So, in terms of antibodies, if the mother's got an infection and she's got very high levels of IgG going around, that may affect transfer in ways that we don't expect. And using modeling, you can do a lot of experiments very quickly to predict where the system's likely to behave in unexpected ways. And I think the real problem for a lot of drug transfer across the placenta is, if everything's happening as it happens typically, that's great. But often, if we don't have quite enough knowledge of the underlying biology, or if there's a combination of factors that come together, you can get something very different happening. And I think the modeling allows us to identify where that might be likely to happen.
Dennis?
So, my opinion is that, just modeling and measuring. To answer your question directly, can you get safety data? No. But as John said earlier, if there's no model is perfect, and animal models in silico, in vitro, and I think together you can put together the tapestry, as you mentioned, to understand the safety aspect. We are just measuring, you don't know what the fetal outcome is. But models can help to fill the gaps. But those models should be validated in terms of, is this really a reflection of what really happens? In vivo, in humans, in animals, in the in vivo setting, there's so much resilience that just having a little bit of detection of a drug in the fetus does not mean it can lead to adverse event. So, you have to integrate that information. Modeling will help, especially like what Nick and Rohan are talking about, like in early pregnancies, their exposure, then understanding is the target present in the fetus. Those come from other areas. And the animal data will, as John pointed out, there is exposure there. Then understand how much exposure is there target engagement, does that lead to adverse event? And putting together, you can understand exactly if this dose is given to a pregnant woman, and understanding the presence of the target and other non-targets, you can understand the safety. But simply measuring in an in vivo model, the drug level, you can't get in the safety data.
I'd agree with that.
Yeah, let me bring in John and David into this, and in terms of the same sort of a question with respect to the animal models and how they can inform potential human safety issues.
Yeah, so the entire point of what we do, I would argue, is risk, just as what you're saying. There's a benefit risk to everything that we do, and all of these assessments that we conduct non-clinically is really to inform the clinical risk. So, that's the goal, right? But all of this is really informed based off of basic research. We're not going to be able to make that leap directly from these placental transfer studies. I mean, I don't want to belittle them at all, because everything that we do at the stages that I work at are entirely based off of the early stage experimental research that such as this, and it's all incredibly informative. But it's not something that's going to immediately jump from we're looking at the differences in placental transfer versus species. Therefore, that's going to give us risk. It's much more intricate and sort of long term than that. But it all helps to inform in one way or another, however large or small, eventually the studies and the data that are necessary to make an informed decision regarding approval and clinical monitoring and communication of risks to patients.
Can I bring in John as well? John, you had some comments about this, these issues as well, right?
Well, I think my comments are pretty well stated already by several. I think it's easy to be a Luddite and say, no, we don't want this, all new stuff is all newfangled stuff isn't going to work. And in reality, this is an evolution of things, and we need to, and you got different populations of people. You've got the folks that are regulators. You have to give them data. They just can't make decisions based on you're a nice guy or I like your experiment. They have to really, they've got a heavy burden to make sure things remain safe. From the standpoint of people developing products, they're doing their best to make sure it's safe. And sometimes the data that you get is limited because, as David pointed out in his thing, sometimes you can't dose as high as you'd like to, or you can't dose as many times as you want to during gestation because the animals will die because they'll get a buildup of antibody, autoimmune antibody disease. There's all sorts of things that have to be. So we are moving forward piece by piece, and I think that's the thing. As long as everybody's got an open mind as to what we're trying to do, we'll be able to make progress on this. But the view that we're going to eventually be able to, for instance, have placenta on a chip that will work for safety evaluation, that's a way in the future. It may be possible, but nobody's going to base a safety decision just based on that. It's going to be that plus other information, and I think to me that's what we ought to be looking at, is how we're going to fit this all together and come at it as a collegial sort of an activity.
Thank you. And I see that there are a couple of people who are standing here waiting to ask questions on this very topic. So, Aaron, go ahead, please. I think there's a button there that you have to push up.
All right, how about now? No?
You have to slide it up, I think.
Okay, now? Yeah. All right, is it working?
I don't think so.
Yeah, I don't know if the mic is, oh.
Oh, yeah, now it's working.
Okay.
Yeah.
One of those, it takes a second. Yeah, so Aaron Pollack, I'm with the National Institutes of Health. So a little bit of a background to put a framework forward, then I have a potential goal, and then the two-part questions about the goal. So first off, when we look at the entire general drug discovery to non-clinical development, first in human studies, then non-clinical development to assess fetal exposure and DART assays, right? From the beginning, the drug is discovered and initially developed just so that it's efficacious, right, for disorders. Then the paradigm is generally we're screening to see if that drug that we made, what is the exposure or what are the effects in DART assays, right? So that's not built in up front. So the following, I don't think we could do this for small molecules yet, but my question is really specifically to biologics then. Could a goal be to understand enough about the structure, function relationships of monoclonal antibodies or FC fragments so that we could, once they're discovered, design them so that we are very sure that there's a very low likelihood that they will be transported across the placenta? Because, using protein engineering approaches and perhaps the models you've all described to understand the structure or function relationships of those proteins in terms of transport. So if that is a reasonable goal from a research perspective, what would it take to get there?
Not a lot. is the simple answer. I mean, there are people, I would hate to contradict John, but there are people who have developed the placenta on a chip system for looking at IgG transfer. They're actually looking at it for IgG transfer coupled to things. So they're not even interested in the IgG being transferred. They're interested in what's coupled to the IgG being transferred. But again, it's that's sort of the placenta on a chip system, which is what one of the things they're using, is ideal for that because it mimics the multiple layers. And so given the problem with the IgG transfer or being that it's not just handled by one cell system, but it's handled by at least two cell systems. You've now got a model where you can actually play around with those cell systems and see how well that actually works, what gets transferred, what doesn't get transferred, and so on. So yes, those systems are not in general use now, but I would think another year or two, and you'll see this being published, this sort of thing, and it will be available essentially for use. I mean, there are already companies beginning to develop these things for mass use, as it were. So, this is going to be relatively soon, and I think we can do that.
If your question is about FCR, can you modify a monoclonal antibody to prevent getting into the fetal compartment?
Yeah.
Then you have to ask the question, there are a lot of drugs that are being developed for basically long-acting. For example, just give one injection to maintain that. And that's by tweaking, increasing FCR and binding. So fundamentally, then it's a balance between efficacy and the fact of PD activity. And when you do that, you may be, in fact, theoretically, that's where the models would come and experimental data is important to inform. Will that increase the exposure to the fetus? So you have to balance the two, and it's not that, it's a difficult question to ask. I mean, do you want to have a drug that can give one injection which would last 1 year, or daily injections? For that, for a monoclonal antibody, you need to start modifying those FCR and binding sites. Then it's a balance between that and is that increased exposure, potential increased exposure in the fetus, is it bad? So I don't think it's an easy question, but it's something that has to be experimentally evaluated.
Yeah, so it seems definitely a lot of structure-function relationships to be done. It sounds like the models are maybe there. And I actually didn't think that, you could also flip that of using that as essentially a way to try to enhance fetal transport of something. If you could do the opposite, yes, where you would want a low maternal half-life, but, rapidly taken up. Okay, thank you.
Hi.
Please introduce yourself.
Yeah, thank you. Lynn Yao, FDA. So just to piggyback on, Dinesh, what you just said, it's not actually my question or comment, but, I get that we might want to have longer-acting biological products out there, but we also need to not forget those who are going to get pregnant when they need them. And so even if it would be longer acting, maybe we need to think about formulations and development of products that actually could be used more safely in pregnancy, right? So I think that that's an important consideration, even if, it's not going to be the same product, just like for children. We develop different formulation strengths, different methods of administration. So I think we do have to think about whether or not there's the potential to develop those exact molecules that actually could be given more safely during pregnancy.
And I absolutely agree. I think you need to have an integrated approach. One size fits all may not be the right answer.
Right. But actually, my question, and I'm going to be a little provocative, I think, to the group. It sounds very much like, animal models, not going to get us all the way there. The placenta on a chip, the organoids, that gives the hope and the promise that integrating all of that information will get us closer to models that can actually predict what's going to happen in the fetus, right? What kind of safety considerations they had in the fetus. So knowing that the state right now is that we're not quite there, my question to the panel is there a situation that you are confident that the animal data tell us the whole story? In other words, does the animal data ever get us far enough that we really wouldn't want to have human clinical data?
No. I just agree. I just think there is a utility to the animal models, but there are enough differences. I think most biological scientists are used to dealing with, it's a liver. It's a liver. It's a rabbit liver. It's a rat liver. It's a chimpanzee liver, and they're all the same, sort of. Placenta is so completely different that there is no substitute, as the phrase goes. So I think while the animal work is very useful, there is no substitute. It will require human work of one sort or another.
So if I could ask a follow-on, and thanks, David. And so the follow-up is, if there's no substitute, right, we can't rely wholly on animal data in almost any situation. And we heard our panel in the morning say, we don't know what animal data means. I want to know if I'm supposed to give my patient this, product when they're pregnant or not, and we don't know what the animal data mean. What can we do to better explain that we're going to need the human data, that we can't just rely on the animal data to support, a decision about giving a drug to a pregnant individual or not?
So, that's exactly what we are trying to do with ICHE 21. There is no single animal model that's appropriate, but when you do see, the people who do the studies, who do understand the data, needs to help to explain, translate that into exactly what's real and what can be happening. And then when you see, you can't think of it this way, like a rat or a rabbit, I'm approximating here. The embryo develops much faster than in a human. And fundamentally, we do know that a fast developing embryo can have an impact on if you perturb the biological process. So rats and rabbits may be oversensitizing. So understanding those concepts, understanding the exposure, all the data, animal data can inform on basically how you can approach a clinical trial in pregnant women. And that's exactly what we are trying to do with ICHE 21. Unlike any other system, like you can do, let's say you have in animal studies, you may see liver toxicity. You can do ethically, you can do a clinical trial and see is there liver function enzymes going up or anything like that. But you can't do that. But animal studies are the next best thing. It's a stepping stone towards a clinical trial, in my mind.
Yeah, you can't do away with the animal data yet. Certainly there's the goal downstream that we shouldn't be using animals. There's a big push for this now, and FDA supports the development of these alternative technologies. But just as has been said, these are the best tools that we have now.
Right.
So even though there are challenges and there are limitations, it's incumbent upon us to figure out how to fine-tune those models to best inform the clinical risk or to develop new models entirely. And these things they take time to develop and then to validate and then to implement.
I'm sorry, I know I'm hogging, but I got one other thing to say, and that's because I work at FDA, and I'm struggled in this catch-22 every day, which is that we get an animal finding that's bad, right? Low multiple, looks like it's a bad major congenital malformation in either one species or maybe even more than one species. Then we write these embryo-fetal tox warnings, and we say contraception, and we say, do not use during pregnancy, and then we say, well, then we should get the human data, and then everybody says, well, there's no way to get the human data because what we've labeled says don't use it. So, what I'm trying to do is, spur the conversation for the next now and also for the next day and a half, the next half a day, about what we can do about that, because that's where we're caught right now.
And that's an excellent point. And, I want to sort of build upon that point a little bit as well with the panelists here. In terms of, we've heard that the animal studies will not really inform, in terms of human fetal safety here. But are there things that we could do with the animal studies and also some of the in vitro studies here that we've talked about that could help us refine our approach and have a better endpoint there in these studies that could better inform what is likely to happen in humans? Could we sort of combine those two approaches as well as some human data, to better refine how, for example, are we doing animal studies at a super pharmacological doses, if we knew what the fetal exposure was, let's say this placental efflux of transport of a drug, and the fetal exposure is going to be much lower than the maternal exposure, if we had some information about that, could we refine our animal studies in such a way that we'd have a better experimental design in our animal studies to better inform what might happen at the level of the human toxicity?
Well, the first thing I want to reiterate and to clarify is that the initial question was whether or not we think that the animal data will fully replace human data or something to that effect. And the answer there, I would agree, is probably no. But that's not to say that the animal data does not provide any utility at all. I mean,
and could we do better, in terms of our
yeah, I mean, as I said before, it's the best tool that we have, and so it is informative, and there are limitations, but it's not like, it's the best we have for educated guesses. So, from my perspective, and I'm maybe going a little off of your question a little bit, but from my perspective, it's maybe I should let someone else go first. Regulator speaks last.
Well, I think there's always going to be utility of the animal models. They can feed into whatever we're doing. Frankly, the human studies, clinical studies, can feed back to in vitro. There is it's not an immunosuppressive, but there's an HIV drug which is in common use around the world, which first came up as a problem in terms of potential interference with folate transport, and I think that's been confirmed, and is now suggested as being associated with increased levels of preeclampsia. And I think what's going to happen, or what I know is beginning to happen, is people are going back to human models to try and look at those particular problems. Because, again, for at least the preeclampsia one, there's no point in going back to an animal model. You're not going to simulate that in the animal anyway. So we've got to go back and use a human model of some sort. The better the model for looking at invasion, the more we'll get out of it. So I think there are those sort of things that we need to be able to do, and we can go both ways.
So it's a continuous feedback loop that we need to keep in mind. That we could utilize some human data to better refine how our in vitro and animal studies are being conducted.
I think it feeds into Rohan's models, which I think, again, feed back to what we want to be able to do on the bench, sort of the wet lab.
Tamara, you have some questions online?
Hello, hi, it's Mara Johnson, FDA. Yes, we do have questions coming in online. I did want to get one to the panel, but I'm also reminded that we are coming upon time for ending the panel discussion. So I'll ask the question and then we'll see what we have time for. The question, this is for placental modeling. Are folks also paying attention to the placenta as an endocrine and or immune organ, not just a barrier transporter?
Who would like to say that one?
I mean, when I lecture to students, what I tell them is that the placenta is a lung, a pituitary, it's a drug processing system. It's got neural connections. It's all these things combined into one. It throws out a variety of endocrine substances, growth factors, God knows what, and in gram quantities. So, when you think about how systems operate, you've got to think about all those. We haven't covered what these things are doing to the placenta, but rather what's happening about them when they're transferred by the placenta and how. But a lot of these things will also affect the placenta. And so, once you affect the placenta in terms of structure and function, then you're not only affecting that organ, you're now affecting what happens to the fetus. So I think there's got to be a degree of consideration within that. One of the questions, or one of the things that people are talking about is corticosteroid transport. And there is a line, Les can probably answer this one better, but, there's a line of thought that says that depending on the 11-beta HSD, I believe, expression in the placenta, you can get cortisol transfer or metabolism altered. And that in itself may alter fetal programming. And so all of these things need to be examined. And in some cases, there's no substitute for looking at the human version.
Yeah. Next question, please. Please introduce yourself.
I'm Jeff Roberts. I'm formerly of the Office of Vaccines in CBER. Now I do clinical development for Merck. I know we're running out of time, and this may be the beginning of our next conference, but I hear us saying, are these new techniques that we're developing, animals, good enough? And it begs the question to me, good enough for what? So I want to focus in on the narrow question of good enough to mitigate risk enough to allow studies in pregnancy. That's what we always thought about in the Office of Vaccines. That's what we were up to, is have we mitigated well enough to allow studies in pregnancy? And I would challenge you guys that we're throwing a lot in terms of dose at these animals, almost trying to induce teratogenicity. And we're also sometimes not hewing to the principles that are laid out in the platform technology guidance that I don't know if you guys are familiar with. In other words, we know a lot about some classes of vaccines and drugs, and we could maybe draw back to some of the less some of the in vitro approaches to mitigating that risk of going into pregnancy. So, it sort of goes to Lynn's point. What do we do about these problems? Potentially we draw back a little to in vitro studies when we can, based on platforms that we're familiar with. But more to the point, what do you guys think about the fact that we're almost giving it our best attempt to induce, and see teratogenicity, rather than trying to really replicate what might happen in humans?
You're absolutely correct. Actually, that's a dichotomy between what the guidance is and expectations for getting a drug for authorization, as opposed to what really are we tweaking, testing like that. And I think it's important to be what you said about looking at the platform, if you know the class, if you know what's happening, do you really need to be repeating the same thing? And if you know, corticosteroids are a really good example. If you go and give a corticosteroid to a mouse, you're going to have cleft lips and cleft palates. But is it translatable to humans? No. If you understand that biology, you would know exactly this is not a useful model. So you have to actually take it. I don't look at these animal studies as not being important. I think it's just a stepping stone to understanding what other things that we haven't thought of do we see anything? And is the dose, is the saturation of the target, are we basically doing something that's unrealistic for human translation? And I think that aspect is not coming, and that's the biggest gap we have currently in human risk assessment when it comes to translating from animals to or in vitro to humans.
I think we don't want to be going backwards, as I was describing earlier, of having seen things in the human that we have to now go back and investigate. That's not the right way it should happen. And I happen to know that some of the experiments that were done earlier with that particular drug were done on a model that just doesn't work. So I think you need to get the models that work best so that you can get the best answers out and not have to go backwards and do work on them after you've seen the problems that they've generated in the human.
So a couple points. First of all, my understanding based on my experience is that particularly with labeling, when exposures are over 25-fold higher than what's seen in humans, it's often not considered label worthy. I mean, it's that high. If you got like, okay, yeah, we see teratogenicity at like 84 times human exposure, like, okay, what does that mean? Nothing. Probably nothing. But if it's like 3 times, yeah, okay, that's more comprehensible. So that's the cutoff that was that's been put in place to determine it's sort of a guideline what's worthy of being communicated and what's not being worthy of being communicated. And secondly, I'm at risk of losing my own train of thought here. I guess what I'm thinking, there are labels as they're written now are kind of open to interpretation, and this is kind of alluding to your point and to the previous point. They're open to interpretation. So me as a non-clinical reviewer, I'm seeing these findings. Okay, yes, we have concerns. These concerns go in the label. These get discussed with the clinical staff. Okay, the clinical staff says, okay, well, we are thinking this is maybe a bigger risk or a less risk, and maybe it should be limited to certain populations, or maybe we should be communicated in the labels in certain ways, or maybe this is a concern, and not just for reproductive toxicity, but maybe these concerns are big enough that we don't even want these trials to move forward, or we need to put other limitations in place or so forth. So, but in terms of labeling, like, they may have their own concerns that need to be labeled. And then these things go out into the public, and then doctors will see them and they'll say, okay, well, I don't know if this is really something I feel comfortable prescribing for this particular individual. So there's just multiple layers here that are so subject to interpretation that it can be very subjective to by the time it actually gets to the patients that need it. Now, I'm possibly opening a huge can of worms and getting myself into very big trouble. That's just my take on it, and I don't know that I really have an answer to it exactly right now. It's something that needs to be discussed as a community.
Well, ideally, it'd be wonderful if the physicians all had taken courses in risk assessment and all this kind of stuff. The deal is that, what was it, 10 years ago when we had ABCDX, everybody used that. It was simple, but it wasn't accurate. Then the labels were changed to try to put some of the risk in there so that they could be informed decisions. And the problem is that if the physicians don't have the tools to understand what's written there, then the label could get to be a lot longer as we explain, well, this is what could happen, but it's really not all that important because we saw it in a mouse and it's probably blah, blah, blah. And then nobody would read the label because it's too long. So I think we need to be someplace in the middle, but it's not going to be easy to get there. I mean, really.
One quick question for David. Aren't the embryo toxicity data done before you know what the dose is going to be in humans? That's part of the problem.
Well, the embryo fetal development studies are usually the first of the DART studies that are completed, and usually I think that's before you want to include pregnant individuals, and usually that's like before phase 2, I think.
And usually in drug development, you do have a fairly good idea by the time you do the embryo fetal development studies where the doses are going to be, and you try to make sure you're bracketing and having that. Yes, you may not have the exact dose, but you do have an idea.
Yeah, by phase 2, you should have a decent.
But I wonder also whether relying on just the dose issue is also misleading, because one should think about, what the pharmacokinetics are, right? And the differences, because animals behave very differently than humans. And so we should think about more the exposure at the maternal level, and potentially thinking about what the fetal exposure is likely to be based upon some of these models. You know, so for example, if there's a great degree of efflux of the drug by the placental transporters, you know, the concentration in the fetus in humans is going to be much lower. And so we should think about how can we reproduce those type of concentrations in the animal studies to really get a better handle on, at the equivalent exposure levels, what is the fetal safety and toxicity. And I want to bring in Rohan here as perhaps a final word before we close here, in terms of any of his thoughts, in terms of this discussion about animal models and predictions and potential informative studies in terms of fetal safety.
Well, I think computational modeling can be applied to animals, and I think, as I was saying during the talk, I think one of the really powerful things we could do is try and help use the modeling to understand what's transferable between different systems. So, where you can use something from an animal model which will be applicable to a human.
Thank you. I think it's time for a break. This is not going to be the end of the discussion, so to speak. I'm sure there will be a lot more, so feel free to have discussion with the panelists. And I'm sure tomorrow we'll be integrating some of this knowledge also into other discussions that are going to be happening.
Great. Hello, thank you so much, panelists. Thank you, Jajwant. And we have had such a good discussion with this panel. We are a little bit behind, so what we're going to do is keep moving on to the next session. As audience members need, you can step out, to use the facilities or such, but we're going to keep moving to session 3. Thank you. At this time, I would like to invite Dr. Kelly Stone to the podium, as he will be our moderator for session 3.
I'm Kelly Stone. I'm a pediatrician and an allergist immunologist. I'm here at the FDA, where I'm Associate Director in the Division of Pulmonology, Allergy, Critical Care in CEDAR. The session this afternoon is titled Framing Concerns for In Utero Exposed Infants Based on Available Data. The risk of immunosuppression in infants from in utero exposure. Immunosuppressive products depends on many factors, including the mechanism of action of the specific immunosuppressive drug. The assessment of risks for specific products impacts immunization recommendations, particularly for live vaccines, but also impacts vaccine safety. The goal of the session this afternoon is to talk about available data that may provide some context for assessing benefit risk. Some of this was covered in the outstanding talk this morning on the current clinical landscape. And the way this panel will work, I'm going to ask each panelist to introduce themselves, a brief introduction. I'm going to go through a series of questions, but I will reserve time at the end for audience questions. So let me start here, Jeff.
I'm Jeff Roberts. I was in the Office of Vaccines in CBER for 14 years before I went to Merck about 2.5 years ago, and I do clinical development for them. While I was in the Office of Vaccines, I really enjoyed working with my colleagues in CEDAR on the use of development and use of these products in pregnancy. Maternal immunization was kind of near and dear to the hearts of me, particularly me and Marian Gruber, who was office director at the time. And we've been really gratified to see, for example, Pfizer's RSV vaccine for use in pregnancy to prevent RSV in infants. This was something that we started talking about with the industry many, many years ago. And now I'm internal to Merck, preaching the same thing that Lynn and others and I, back in the day, were preaching, which is to think early about what the end game is going to be with respect to pregnancy and lactation when you start developing these products. What I've found is that the bureaucracy in an institution like Merck rivals the FDA. It's really remarkable. Just to get us to change a simple template to spur our development teams to think about this early in development has been an enormous heavy lift. Eventually, I think we should, together as pharma, or maybe some as ICH, Transcelerate, get together and do this together, and to say industry together should start to think about this earlier on. And I have some thoughts about how we can maybe do that eventually. But I've been doing vaccines for many, many years, so to the extent that we want to take this to a place where we say, what about safety and efficacy with respect to immunomodulatory agents in pregnancy? Happy to weigh in about what that might mean for the vaccine piece of things.
Michael Keller, Children's National Hospital here in D.C. I'm a pediatric immunologist specialist in congenital immune deficiency. So I'm both a user of a lot of these immunosuppressive and biologic therapies in my dysregulatory patients. I also evaluate a lot of infants who are prenatally exposed to these agents. So I'm really here to really provide the clinician aspect, and particularly also to talk a little bit about congenital immune deficiency as it relates to secondary immune deficiency and the parallels.
Great. And Ofer?
Hello, everyone. Good afternoon. My name is Ofer Levy. I am a physician scientist at Boston Children's Hospital and a professor of pediatrics at Harvard Medical School. I'm an associate member at the Broad Institute at MIT and Harvard, and I'm director of the Precision Vaccines Program at Boston Children's Hospital. The PVP is a multidisciplinary academic program focused on discovery and development of vaccines tailored for vulnerable populations. We make use of novel approaches such as systems biology, in vitro modeling of the human immune system, and preclinical and clinical models to advance vaccines that are tailored to vulnerable populations, whether they're very young, elder, immunocompromised, etc. I am also a special government employee of FDA. I serve on the Vaccines and Related Biologic Products Advisory Committee, or VRBPAC, serving on those committees during the COVID mRNA vaccine discussions and authorizations. Of course, my comments today will be my own personal opinions. Finally, in the interest of disclosure, I'm also a co-founder of a company named OVAX, OVAX, that's involved in developing vaccines against opioid overdose, including fentanyl overdose. A pleasure to be here today and to help us think through how we bring precision medicine concepts and principles to the important topic at hand. Thank you.
Great, thank you. As you can see, we have a terrific panel. So I'm going to start out asking the panel to discuss the available literature on outcomes for infants who have received live vaccines, and whether there are potential lessons learned from this literature to inform vaccine recommendations, particularly for live vaccines. And you can volunteer, or I can call on.
Why don't we go in reverse? Ofer, you want to get started?
Sure. So first of all, I really want to thank FDA and all involved in organizing this really important meeting and pulling this together. It's really an important, in some ways understudied, and very impactful area. The days where there were only one or two tools to modulate the immune system are over. I'm also a practicing pediatric infectious disease clinician, and when I'm rounding in the hospital, it is a challenge to stay up to date on every new monoclonal antibody and small molecule that is out there to modulate the immune system. And that's great. It's great that we're developing these tools, but as they're used in diverse populations, including pregnant women, questions are arising as to the implications for the fetus, newborn, and infant. And we've already heard about some of those questions today. As a team preparing for today's meeting, we discussed the value of trying to gather some literature in this area, and Kelly, you've been great bringing us together and bringing some of the literature together for our review. My impression is that there are a smattering of case reports, some of them quite dramatic, with unfortunate deaths after receipt of BCG in infants that born to mothers who received anti-TNF therapies. And I think one of the and then a number of literature reviews, systemic reviews, etc., which are helpful because we want to get past single case reports. I think that there is emerging consensus on, exposure to anti-TNF therapies in pregnancy and avoiding BCG, at least in the first months of life. That's makes sense to me based on what I'm seeing, even though the data still remain limited, but that does seem to make sense. It is a loss depending on where you're at, and there's this whole concept of risk benefit. I believe one of the speakers talked about it earlier today. What does it mean to postpone BCG? Let's take that for a moment. BCG is one of the most commonly given vaccines in the world. I believe over 100 million newborns every year around the world receive BCG. World Health Organization, WHO, now recommends that it be given as close as possible to birth, because not only does it protect against disseminated tuberculosis in early life, but it also seems to have potentially beneficial off-target effects. And in resource-poor settings, West Africa, etc., has shown to reduce the incidence of bacterial sepsis and other infections in early life. So I'm just pulling that up as an example where everything seems to matter. Which vaccine we're talking about, the target population, the exact age, the gestational age of exposure, the age of the newborn or infant when they're immunized. All these details matter, and then where they are in the world. The risk for neonatal sepsis may be quite a bit higher in newborns born in Africa, for example, than it is for the average newborn, full-term newborn born in the United States. So as we make these considerations and recommendations, I think we're going to have to be very mindful to be pretty granular in our thinking. And the challenge with that, of course, is we started by saying there's not enough data, and if there's not enough data and you want to be granular, boy, that's why there's a lot to discuss today. So I think one of the major conclusions will be it matters which vaccine we're talking about, because I'm mentioning some things about BCG, but then when it comes to rotavirus, which was a different live vaccine, we saw a powerful article in Lancet Child Adolescent Health in 2023 by Fitzpatrick, Karina Top, and others in Canada. They have the special immunization clinical network, and they concluded that rotavirus vaccine can be given to infants exposed to anti-TNF in utero, despite the prior recommendations to avoid it. So this is a long-winded way of saying that we can't batch all live vaccines under one policy. It seems like it's going to depend which one, and that all the clinical parameters are going to matter. So I believe that what we need here is a repository, some sort of centralized repository of data and publications in this area, and to keep building into that repository and periodically reviewing it and updating our recommendations. I don't see any other way. Thank you.
Great, thank you for that, Mike.
Thanks, Ofer. Earlier today, Dr. Mondevan did a really wonderful job in summarizing a lot of the literature that we're referring to here. So majority of these are just, either case series, case reports. Coming from the rare disease space, given that many of these had sometimes several hundred patients, seems actually pretty decent. But really to find severe adverse events that are going to be, 1 in 10,000, 1 in 100,000, it's simply not enough data to be able to find those cases. And you really want to have recommendations that you're going to roll out uniformly, either U.S.-wide or worldwide. We need a lot more data. So Officer's exactly right. We really need more reporting. With that said, looking at the data that we do have, including a nice prospective trial in Canada that was summarized earlier today, I would argue that the data is actually relatively reassuring. That with the exception of the case report in England detailing a 4-month-old who died after BCG vaccination, which I would add was not adequately researched. There's a lot missing from that report. There were very, very few serious adverse events that happened after live vaccine exposure in these biologic exposed children. That said, I agree with Offer that really we have to take the vaccine by vaccine. There's a world difference between BCG and Varivax, for instance.
Yeah, just to echo some more that it is very much case by case. Not all live vaccines are built the same. In fact, they're dramatically different. Fortunately, I think for the most part, it's a fairly narrow question, particularly in the U.S. and in high-income countries, which is, what about rotavirus? So I guess what I would add is that it's nice when the data line up together. We see from these clinical data some reassuring, outcomes with respect to rotavirus. I can tell you that also from a scientific perspective, this is what you would anticipate. There's a couple of things about the way a live attenuated vaccine is built in the rotavirus space compared with other live vaccines. First of all, we have tremendous experience with it across the globe. Many, many millions, maybe into the billions of doses administered without seeing any evidence of reversion and behavior like a wild type, virus that these attenuated products are built from. That's one of our main concerns with respect to live attenuated products. So we haven't seen that. That's reassuring. The other thing is the live attenuated products that scare me are the ones that have the ability to invade systemically. And, we're talking about yellow fever. The Ebola product is built on a virus that can be systemic and can be found in joint spaces and stuff like that. Rota itself doesn't ever do that. And so that worries me less. So those kind of scientific priors are lining up with the clinical data overall is really reassuring. So couldn't agree more with where this consensus process landed with respect to use of rotavirus in these particular situations.
All right, thank you. I'm going to go on to the next question, which has to do with secondary immunodeficiencies due to treatment with specific immunosuppressants and available data. Is there additional data in terms of immune responses with specific products that would be valuable? And are there specific immune targets that are particularly impactful for potential safety concerns from live vaccines? I can go to reverse order this time, if you're willing.
The first part's really about effectiveness. It's about the immune response to the vaccine products that potentially all the rest of them, the inactivated, the subunit products, in the context of the immunosuppression.
Exactly.
Yeah. So here, I really am not in the majority. I've found that particularly in the academic community, and to some extent, industry likes to get these studies in labels where we pick an immunocompromised population, and in this case, it could be the infants, the offspring of mothers who are on immunomodulatory agents. It could be, immunocompromised for whatever other reason. And then we study the vaccine in that population because everyone wants to draw broad conclusions about how that vaccine will perform in immunocompromised populations. But the problem is that immunocompromise is such a broad array of different reasons for different. So, for example, the immunomodulatory agents drive anti-inflammatory pathways really hard for very specific ones and result in things that we can't really anticipate a lot of times with respect to immune responses. So we get results of these vaccine studies in some specific immunocompromised population. And what I would say is that really tells us only very narrowly about how that vaccine will perform for that very specific population. So I guess I'm a little more sanguine about doing those types of studies and what kind of conclusions we can draw from them.
Yeah, and I might jump in here and take a middle ground. While I don't think we can extrapolate one-to-one from studies of vaccine safety, immunogenicity, and efficacy in individuals with primary immunodeficiency and directly apply those to infants born to mothers treated with agents that may block the same or similar pathway, I do think that those kind of data in individuals with primary immunodeficiency may help give us a sense of what to look out for as we're gathering clinical data when certain drugs are pushed out to populations, including pregnant populations. The other point I wanted to make is, there are many approaches to inhibit inflammation or immune reactions to either prevent or treat a range of autoimmune diseases. One of them is to inhibit innate immune pathways, and I'm now specifically alluding to there's a growing literature on developing inhibitors of toll-like receptor 8 or TLR8. We know these innate immune receptors, TLRs, and TLR8 plays a special role recognizing single-stranded RNA, such as that from influenza. There's some evidence that toll-8 plays a role in the development and progression of lupus, of systemic lupus erythematosus or SLE, and there are efforts underway to develop inhibitors of toll-8 to treat that condition, SLE. And, we'll have to see if those inhibitors progress to approval or are eventually used in pregnant women. But the reason I'm focusing in on that receptor is there's some emerging evidence that toll-like receptor 8 is especially important in the detection of live microbes. It senses, that the innate immune system differentiates between dead and live threats. And toll-8 is particularly pro-inflammatory as it detects mRNAs from live pathogens. And, we're going to need data, but one might predict that if you inhibit toll-8, you might really fundamentally alter the response to live vaccines. So there are many areas to consider. That, in my view, will be one of them coming up.
Yeah, and with regard to the secondary immunodeficiency patients, patients with juvenile idiopathic arthritis, JIA, patients with inflammatory bowel disease, there's been a reasonable number of studies in pediatric patients who are getting their primary vaccines on biologic therapy. And generally, there too, the data is relatively reassuring. There's some cases where the magnitude of serologic response might be impacted to specific vaccines, like for instance, influenza B in the live attenuated influenza vaccine, inactivated influenza vaccine. But as a whole, though, there's really no indication that there's increased risk of a lot of these infections, these populations, and no real data that the vaccine is ineffective in spite of the quantity of the titers that we're seeing.
I would just add that in all the years that I did this, I can't think of any data that even suggested that there's a safety signal for the inactivated protein subunit, any non-live product. And so, that we always need to be open to that possibility, but I'm not aware of any safety signals there.
All right, let me move on to another question. And at some point, if anyone has questions, I'm certainly happy to break in. But based on what's currently known, provide your opinion on what circumstances vaccination with live attenuated vaccines should be delayed in an infant. And describe what factors would influence the duration of the delay. Discuss risk of acquiring disease from the vaccine compared to the risk of acquiring disease from the environment. So what situations where you would be concerned, and sort of moving on to what can you measure that may sort of inform that?
I'm happy to take a first stab at this. So one thing that we are fortunate in the U.S. to have is newborn screening for conditions such as severe combined immunodeficiency and related disorders of T-cell biogenesis. And these are one of the most severe forms of congenital immunodeficiency that usually are basically a straight shot to transplantation, and would be a very clear reason where a live vaccine would be dangerous. Chronic rotavirus has been well described, unfortunately. Before newborn screening, it's a presenting sign of SCID. And really is not fatal, but is very, very troublesome. So we occasionally will see TREC-positive infants that have been prenatally exposed to maternal immunosuppression. So thiopurines are pretty well described in this regard, particularly when infants have TPMT mutations. But that would be kind of, especially when we see them and determine if they're truly T-cell lymphopenic, that would be, to me, a very clear indication that you really need to think about delaying live vaccines until the patient is further worked up and until you determine whether or not they will resolve, which, again, in most cases, if it is exposure to immune suppression, should resolve between anywhere from 4 to 6 months after birth.
Yeah, this is a great advance. I totally agree. I mean, I think, sometimes live attenuated vaccines were the vehicle to make the diagnosis of some of these specific immune deficiencies. Wasn't uncommon. You make it to age 1 without any real pathognomonic infections, and then MMR, you have a really bad outcome, and that's diagnostic essentially. So, did you want to?
Terrifyingly, not to get sidetracked, but we even see in some combined immune deficiencies that may not present even in teenage years. We can see chronic rubella as a presenting sign in some of those patients. But those, luckily, will be caught on newborn screen even now.
I'm going to pile in and recall an evening many years ago that I was on call in the emergency room at Boston Children's Hospital with Dr. Gary Fleischer, our former physician-in-chief, and we saw an 11-year-old girl with a maculopapular rash and a pneumonitis and respiratory distress. And after a lot of workup of her lesions, it turned out that it was vaccine strain varicella virus and from the vaccine. And it turned out that we worked with the immunology division at Boston Children's to diagnose the first case of invariant NKT cell deficiency. We published that case report. So as people are saying, live vaccines can be probes of the immune system, and immunodeficiencies have been discovered by observing difficulty of rare hosts to control live attenuated vaccine replication.
Yes, I would just emphasize again something that you raised, Ofer, which is benefit risk, and just remind us all that it really comes down to that. We still have licensed in this country a smallpox vaccine that is very dangerous, that even in the most healthy, immune competent adults can cause systemic disease that can be life-threatening. And yet, the label says, essentially, if you face smallpox, it's worth it. you know, so it really comes down to that, and there may be some difficult decision-making, particularly with the travel vaccines, because many of them are live attenuated, and you really want to protect your infant if you're, you know, traveling in some situations, and it's going to be case-by-case, benefit-risk.
And to that end, just because this often comes up in terms of what is really the value of the rotavirus vaccine in the U.S., where we so rarely have actual rotavirus-associated deaths. I did want to point out that, I mean, still 200,000 ED visits, 50,000 hospitalizations related to rotavirus a year. So there's definitely, it's definitely still doing a great deal. And so definitely avoiding it is not without major consequence.
Completely agree with that comment. Thank you for that.
And then let me ask, is there any data that would be helpful to better inform immunization recommendations for infants exposed in utero to immunosuppressives? What type of data would be helpful in making those determinations?
I think want every type of data. I mean, these are really important. So we want clinical data, we want immunologic data, we want systems biology data, we want in vitro data. We heard earlier today about in vitro systems. Let's not forget that FDA Modernization Act 2.0 was signed into law on the 29th of December 2022 by President Biden, and we view that as a very important development because it encourages the use of human in vitro models and artificial intelligence, machine learning in support of drug and pharmaceutical and vaccine development. And you know, there's a lot to learn about how the growing pipeline of immunomodulators affect the pregnant mother, the fetus, and the newborn. And I would hope that FDA would require of sponsors, manufacturers, to gather more information when they do their pivotal studies, including phase 1, 2, 3 studies, and including studies in vulnerable populations such as pregnant women, so that more information is captured and we learn more. If in vitro systems would have predicted a safety signal, that's critical. And for people who tell me, well, that's very expensive to do all that additional work, is that any more expensive? It's less expensive than having to do large clinical trials of every type of immunomodulator in every subpopulation, et cetera. So I think developing non-clinical and preclinical data sets that help us better understand which immunomodulators under which circumstances lead to which kind of risk will be very important moving forward.
So for Mike and I debated this a little bit, sort of pre-panels. I'd be interested to hear your feedback here, but to me, some of the data that can be most informative is not simply take Tdap and go into a specific immunocompromised population and see what happens with Tdap, but instead to use some model antigens in specific immunomodulatory or immunocompromised populations to understand exactly those populations and what their deficit is, to probe it completely. There are a set of like bacteriophage neoantigens that no one ever sees, so that everyone's essentially naive. Those are T-cell dependent antigens that can tell you about how the immune system is performing, along with a battery of other things, including challenging with polysaccharide vaccines that are T-cell independent. You can learn a lot about a specific immunocompromised population, and I think that data has a lot of value toward telling us, in general, what recommendations we should be making with respect to the performance of the other products.
And you feel, you mean in relation to a product that's known to inhibit through its mechanism of action a certain pathway to draw a relationship between that and somebody with a primary immune deficiency in that same pathway? Is that what you're alluding to?
Yeah, and or to study the primary immune deficiency with this panel of tests to fully understand what the defects are and how profound they are and how maybe they have interplay with each other.
Yeah, I think that's valuable research. I think that will add to our mechanistic understanding. I'm all for it, and I agree with you. And I feel like at the end of the day, we will also need just clinical data from the actual scenario where the pregnant woman is exposed to one of these immunomodulators and the infant is followed, because we need to tie it all together in the end to a clinical endpoint.
Mike, any comments?
No, I agree. I mean, use of, we definitely, neoantigens, including for that matter in the U.S., things like typhoid vaccine or even rabies vaccine are useful and potentially could help to have a clean system, clean kind of readout in terms of what B and T cell responses look like on different immunosuppressive agents.
Dr. Nelson?
Skip Nelson, Johnson & Johnson. Interesting idea. I guess my question is how much of that work could be done in adults? I mean, to what extent could you expose them to product X and then do these antigen probes? Because it'd be kind of hard to do that kind of study in a child. So, I mean, what are your thoughts? Is that enough to find out that information?
Yes and no. I mean, I think you can learn a lot in adults. That's where it's been done, obviously. But as you know, the infant immune system, and I shouldn't even be commenting on this on a panel that Oprah's sitting on, is profoundly immature in some ways, and that's where we really need to know what that interplay is between, say, a mom who had an immunomodulatory agent during pregnancy and the immaturity of the infant immune system, how those work together to give you a specific picture of immunocompromise. And so these types of model systems like bacteriophage to probe the immune system in that setting would be tremendously useful. On the other hand, these are very difficult systems to do, difficult studies to do. And there's no world in which we're going to be sticking infants, 18 times in the context of these types of studies. So it's very difficult. I recognize that.
Yeah, I will point out that you're absolutely right that the, from the moment we're born to the moment we die, the Greek philosopher Heraclitus of Ephesus taught us the only constant thing in life is change, and the immune system is no different. And we have published a number of studies showing really very dynamic and robust cellular molecular trajectories over the first even hours, days, weeks, months of life. If you survey the cells and molecules in the peripheral blood, and we've developed approaches, we call them a small sample big data approach. We could take less than half an ml of peripheral blood, less than half an ml, and extract transcriptomic, epigenetic, proteomic, metabolomic, and other types of data, and witness the ontogeny, the change in the same individual across time. And those changes are profound in early life and mirror the susceptibility, particularly in early life, to intracellular pathogens. So there's a lot to learn about that dynamic immune system and how it relates to health and disease and how it's impacted by immunization, or in this case, how it's impacted by in utero exposure to immunomodulatory agents. There's a lot to learn here, but also tremendous potential to have a much deeper understanding of trajectories predictive of health and disease in early life.
Great, thank you. For Tamara, do you have a online?
Yes. Yes. Hello? It's on? It's working? Okay. Yes, we have one question from online. It asks, would vaccine titer testing with pneumococcal testing be preferential to tetanus or others in evaluation of humoral response in infants?
This is where vaccines get so complicated. I mean, with some vaccines, we have a quote-unquote correlate of protection that we can trust in any situation. We know that if you have a titer of such and so against surface antigen for Hep B, that you will absolutely be protected 100%. In contrast, for PCVs, for pneumococcal disease, that correlate is much less well established, and therefore the value of measuring these titers is lower. So I think any data is valuable to bring to the table, but it has variable value in terms of what it can tell you about protection. So you have to be careful, I think.
Hi, I'm Uma Mahadev, and I'm a gastroenterologist at UCSF. And my question is, what is it that puts the infant at risk, if anything? Because in the Lancet study, they did immune phenotyping of these babies that were to infliximab in utero, and they were all fine. We sometimes look at drug levels of the biologic. That doesn't seem to have an association. So, for example, when we had a global consensus, and one of the things we learned from our colleagues in Colombia is that the rates of TB are so high there that they want to give BCG at 1 month of age or 2 months of age. They can't wait till 6 months. And so they were trying to stop the infliximab early to have lower levels in the baby, putting the mother at risk. But is it the level that matters? Because it's not clear that it does. And so that is my question. Do the drug levels even matter?
Yeah, it's a great question. I think the short version is we probably really don't have that data. But when I read that study, my take home really was that most infants exposed really are not having any issue. So why is it we're seeing some that may pop up on newborn screen or on lymphopenia? There's probably something intrinsic in terms of it's probably not just the drug, it's probably also the child.
All right. If there are no other questions, we're 2 minutes over. So I'd like to thank the panel for a terrific discussion. Thank you, Ofer, for joining us virtually. It's nice to see you on the screen. And thank you all.
All right. Thank you. Thank you, everyone, for a very informative Session 3 panel. We are at the end of today's sessions. So I do want to thank our speakers and panelists for informative presentations, sharing the current science about placental transfer, capabilities of non-clinical methods, available clinical data, and knowledge gaps to understand the potential clinical impacts on infants exposed in utero. We will reconvene the workshop tomorrow at 9 a.m. with a focus on clinical study design, future directions, and next steps. Thank you so much for attending.